Nice diffuse flow. Let's stand by for a few minutes and see what Jacob thinks. I don't think this is going to work, but we'll just get a second opinion. And then the next thing we'll do is move to the Chimlet 1 marker, and there should be more clams there, and they're also going to do some super sampling next. So we'll just hang out here for a few minutes, see what the biologist says.
Return home and then just do it over. Yeah, that was a big process.
Some cool diffuse flow.
It is PART 2.
Can you turn the lasers off for a minute? Thank you.
Oh, it's easy to say. Oh, if you just click in there, you go control. Where is that? If you just you can control A, you don't have to do that.
I don't remember. Chimlet. Chimlet 1?
Is that what you said we're going to next?
Yeah.
Do you know where it is relative to us?
I don't. I see it on the list there. Biologist friend.
I don't know where Chimlet 1 is.
What's that?
I said, well, I don't know where Chimlet 1 is. Rhode.
Oh no, I was gonna talk about clams.
Okay, we'll talk about this.
We're at clams 1 right now, but they all look dead.
They should
We've
do.
got clams 2 or 3 that were alive, but
In fact, they don't even all look like clams.
Are they mussels?
Anyways, whatever they are, they're dead.
Okay. We've already collected mussels here. Looks like there's no clams. So the next thing, okay, if you haven't already collected 15 clams, go to near
Go near
Chimlet 1.
1.
And that's where all the super sampling, or around where the super sampling has to be done as well.
Great.
So I think next for you is super. Both mussel collections are done. They said if you need to, you can put the clams in both bio boxes. It's okay to split
Sure,
them
they're
up.
very large.
All the IGTs are taken. So this is done.
Have
So
you found the marker?
do you
For
remember?
Chimlet 1? It's not in there.
Well, so
There should be
Chimlet 1
beginning.
display range
What's the
bearing
coordinates?
percent 200.
You know, buffers. You can keep those on.
243
Right.
Wow.
How far? That might be a different Chimlet 1. That's a Western Iguanas, right?
No, yeah, I can't answer that.
And we want to be at Eastern. I mean, sorry, that should be a statement. I believe that it's on the upper left-hand side of your screen. Wait, my purple one.
This is if I
It's a different Chimlet 1. Somebody might have overwritten it.
yeah.
there's the.
Okay, can I give you some coordinates? Give you some coordinates and see where they show up?
Here, let me marker.
Yeah. All right.
Alright, shoot.
Okay, now one order. 2.10398.
We're at Schlam's 1.
Minus
There's no Schlam's.
91.92653. 92.
We did some stuff.
653. See where that gets you.
More muscles. Budget floors.
It should be at the base of the V is where I remember it. Okay. That's where it should be.
Have we named it?
It should be called Chimlet.
Well, it's just MKR right now, yeah.
Yeah, he's right there.
All right.
Chimlet right underneath it. That's where he should be.
It might just be called Chimlet.
It's just, he's just.
Was it hiding?
It's spelled out.
Instead of
Oh.
earlier
Tricky.
details. Alright.
When you're ready, we can head there. Okay. Clams is the only thing left on animals. We're gonna do a lot of supering around Chimlet 1.
And this transect, it looks like we're just supposed to follow the directions and make it up. Oh, you're supposed to go east. I guess that's pretty
Did we
definite.
have mapping time?
Okay.
You can turn the lasers back on too.
Cool. Follow the land.
Super head transect. Okay.
Yeah.
Is that what those
So muscles, supers, and then I spent a fair amount of
Then you
time
just
in
have to-
geology.
Or
So
not.
there's time at the end.
Close up those
around.
maps and then
I don't know what
Try
you're
again.
talking about.
you said you spent a fair amount of time in your mind. You didn't sample you wanted.
I got 2.5 good cores. We got really good talus, and Amy seemed not that interested in inactive chili, so I didn't grab that many. Yeah, I'm happy with the talus. I'm happy with the 2 cores that we got, but there's still empty tubes if there is extra time to
Sure.
core.
Sure.
But we'll let Amy do that tomorrow.
Okay. And then we still have, so I mean, the super will take a while. And the clams will take a while. But chances are we'll end up either coring for you by the end of our watch or possibly doing these.
I don't think you'll be
Unless.
coring. I think
Okay. Well, if you. Go
there's
ahead.
BBC filming.
I was going to ask if there's a time that you come in just typical 0600.
Sorry, say that again.
Dan's going to come in just typical 0600.
I think that's the plan, and I don't know where the lander is. I don't think it's been moved.
Okay. So we will.
long day.
Oh.
I'm sorry?
That lander's been down there for like 3 days. That camera's long.
Alright, I do want me to
It's
get
only
you.
got like 24 hours of
Oh,
time.
right. What about the lake?
The lights will work.
Cool. He's like,
Well, I think-
found it.
He was interested in vehicles.
Probably not even.
Oh, cool. Okay.
Yeah.
Yeah, and I think Jill still wants to send the IGs back up. So I
Okay.
think for your
I
watch,
don't wanna give you.
supers, clams,
Head
the
lander.
lander,
But do you want the one?
Yes.
and then the
Okay,
next
that's
watch will be filming.
Great. That's a pretty familiar routine for us.
Okay.
A phone crash.
This is messy, but up to date.
Gorgeous.
What matters.
Where does this one spot?
Thanks, guys.
Thanks, Karen.
Thanks for all the support.
We just saw bulbous spot. Six. All friends.
Good morning.
Depends, what are you offering?
Yeah, I'm snack.
There's one cookie.
I can literally offer you a crust of bread.
Oh,
was on this planet.
This is what's the Donner Party. Well, this is the salad cookie.
That is.
Told you. Chalkies. Very salad cookie. That will not stop. It tastes less salad, of course.
What are we doing?
Yeah.
Oh. You sure want to switch out maybe 2 hours? Totally up to you.
Maybe.
It's a maybe. Alright. Pretty straightforward. We're gonna Chimlet 1. And we're gonna look for clams because they struck out on clams, so they say. And then
Except, but not right around Chimlet 1 is where they don't want clams from.
we live in a world of paradoxes, Rachel. Yes. But not anywhere. We're supposed to go to Chimlet 1
Okay.
and then avoid collecting clams from the place we just collected clams, even though Chimlet 1 is the only place we know there are clams.
Awesome.
Do
So we're just
do
going to
we
Chimlet 1.
know if they want these clams? If
We just have to be 10 meters away from the Chimlet.
So position
If we're
ourselves.
less than 10 meters away from the Chimlet, wait, I think that's the cutoff. So 10 meters or more.
That's what Corey told me.
Yeah.
Okay.
10 indoor clams around Smoker 1. Most of our clams. This is possibly not the fully updated version because I just read it online. At any rate, haven't already collected any clams. The first 2 sites collect 15 clams from anywhere, but yeah, hang on. I think they clarified in a revision. Make sure we're on the same page. Oh, this is what's this way. It's not here. I don't.
Near Chimlet 1, but not markers animal 2 or animal 1 or claims 5.
There it is.
Okay.
It is in fact more. Yeah, there it is. So there you go. So we're gonna do that first, whatever that is, and then we're going to run this west-east transect with 4 supers in it. The super transect.
The super transect.
Super transect starting at Chimlet 1. And at that point, we'll probably just go get the lander, and then we'll go back to bed. Ooh, these are clams. Okay.
unknown vitality.
Yeah.
Inactive clams.
Hmm?
This is what Lander was.
Yeah. You're close.
Still got some 20 meters to go. 15 maybe.
Wait, just so I know, are we left?
Yeah.
Thanks.
What's that?
We're 15 meters away.
From the marker that you just found, but yeah.
Alright, well why don't we pull up to the marker, like right on top of it if you don't mind. Let's try not to crush this chimlet, which we may already have harvested previously. Yeah. Oh, it's like meeting an old friend though. That's it, yep. So now what would be nice is I'm going to ask you to enable 2 other markers on, ideally on Doug's chart if you don't mind, Drew.
Okay.
If you're able, are you able to do that? Maybe Doug has to do it. There we go. Cool. Yes, there should be a marker. You ready? Okay. Stand by. Standing by.
No, same thing that you do over there. You just drop the marker
No. I'm trying to have him pull up some existing markers.
Oh, I'm sorry.
That's all right. And I take it that the list of markers available on both consoles is the same. Oh man.
Nope. It is not.
Okay.
If you
Well.
get a coordinate, we can make it happen.
Okay. Stand by for the coordinate. Meanwhile, yeah, we just want to be on top of that chimney.
What do we want to call this one?
We're gonna call this first one S0563-Animal2.
S0563
Animal 2. Great. Ready with the latitude. All right. 2.103938. 03938. That's correct. Negative 91.926405.
91.926405.
Okay. Stand by for the next one. There are 3. Okay, no, there are only 2. The next one should be called S0564
S0564
Animal 1.
Is this animal numeral 1 or ONE?
Numeral, please. Latitude 2.104066.
1040.
Correct.
What was the last?
66.
104066.
Minus 91.926529. Okay.
926529.
Yep. Okay, so great. Now,
Is there a third one?
no, that's fine. That's it. Sorry, there were 2 names for the second one. Now what we're gonna do is you have to turn over. But well, if you want to,
We were just
if you want to. Morning, Ben.
I think we just accidentally got deleted one day and then they just copy and paste the document. That is absolutely what happened. 100% what happened. Yeah, we're the lost. We are the daughter part. We are the daughter part as well.
Today's
We're headed right here where we're at. So we're on top of Chimlet 1. You can see it there. We are looking for a clam site that is not either of those 2 sites that you see on your chart, and the Animal 1 or Animal 2. So what I'm proposing we do, I'm open to other ideas also, is that we get up above the chimney with some decent altitude difference. Tilt the camera down a little bit and spin around and look for clams that are not on the bearings of either of those 2 sites. I guess the way the yeah.
Do a 180 and go across that one.
I think probably better than a 180. Well, it would be a 180, but I think perhaps we should do a 180 from that heading to that heading.
So you want to head this way.
Are you how well are we able to azimuth the place?
Change our heading.
Yeah.
Pretty good.
Okay. So why don't we swing around so we're 0, 0, 0. Unless that's more turn than you want to put on. But yeah, so basically we point north, and then we make a 270 to the left. And we may stop and ask you to actually. Now that's a great clam bed right there.
Yeah, but I don't.
But it's probably too
that's
close to the
too
chimney.
Close to the chimney.
Okay. Yeah. So go ahead and keep swiveling. And we'll just start at that 0, 0, 0 heading, and we'll methodically work our way around. Also a phenomenal clam bed, but I'm afraid that it's really close to the previous sampling site. Okay. So once you're pointed north, let's swing around to the left again. And you go ahead and call out if you see a promising spot too. Do you see where our previous markers are on Ben's display?
Yeah.
Yeah. So go ahead and slowly rotate to the left, please. And we're going to come all the way around basically to a 0, 9, 0. Yeah, I will.
You
Thank
go
you.
a little bit faster. Mayhaps in the distance. Maybe
Yeah,
clams.
there's a marker there, which worries me.
Well, let's not do
Yeah,
that.
keep going.
Keep going.
I don't know exactly which marker that is, but I don't trust that
You
clam
won't
bed.
trust it.
And we have a whole other 180 degrees of arc to look at here. Yep. Okay. That they look kind of dead, but let's get these nice square in our view, and maybe I'll zoom on them real quick. Yeah. Can we? Yeah. So we'll hold once this little clam field is centered. Zooming. They look pretty dead. Do you mind rotating a little bit further? I can also swing the camera, but you'd probably rather I don't. Right, makes sense. Could we center up on this, center up this bit, please? That's good. Thank you. There's a vent in the way. Some of those kind of look alive,
Some of them.
but it's not that great. Let's keep going to the left, and let's see if this vein of clams leads off into the distance anymore. The chinlet. Well, maybe we need to pull over. Well, I don't want to cook the vehicle, I guess.
That's a pretty live patch.
Yeah, it looks pretty good from here. It's also pretty close, which isn't. But I see more in the distance. So why don't we steady up so that this is our heading, and then go over there. Yeah, I mean you can basically center this vertical section here. The patch in the foreground looks live. The patch in the background looks kind of dubious. This is good. It's a good thing. And let's go look at these first, please.
Okay.
Yes, please.
I'm going to message the group chat and say
Mm-hmm.
that we did not see. Because there's okay.
That we did not see what?
We did not see clams at. Clams 1 was dead. Actually, dead clams. Is that the?
They may not have been. It's a shell patch would be the best way to put it. I think more mussel shells.
Yeah.
Um, okay, so once we get out of some of this shear, yeah, that's what I was going to ask.
Do not sample any of these near Chimney.
Okay, these are alive.
They might be too close to the chimney.
They are too close to the chin.
Why do you want to go up to the chin?
Well, yeah, they are. We want to be at least 10 meters, I think. Let's take a look at this more distant patch here. Just basically keep your heading. Yeah, it's sort of a tall order. We want to be 10 meters distant from the chimney and 10 meters distant from either of these. I think we can pull it off though. So, I mean, this is a nice live patch. We are only just 10 meters distant from the chimney. So, or sorry, 10 meters distant from animal 2. Were you going to request that an animal person comes down here and
Yeah. What
verifies
about
our site?
clams 3, 5, and 2? I just don't.
What have they been? I mean, I'm just following directions.
I know,
but
but
that's a good thought.
I know that the last thing we need to do is like
Spend a bunch of time
clams
collecting
that
clams
nobody
at once. Yep. This is a nice looking patch, and it's. I mean, if we follow the clam trail up and to the right, I think we'll be well in the clear. Do you mind just heading a little over here, seeing where that takes us on bird's eye, please? Well, yeah. You're working on this.
I'm
Cool.
messaging the chat.
Awesome. Thank you. Okay, these are so looking good. Yeah, I think that this would be a good patch to set up on, but we're also going to stand by for advice from people who actually need these samples. And maybe we can give them like 5 minutes or something like that. 10 minutes to respond. Meanwhile, I'll zoom around and look at stuff. So to catch up, anyone out there watching on the interwebs, thanks for joining us. This is the Dawn Watch, quite probably the last Dawn Watch, which means, now that I've said it, that it will be most likely the penultimate Dawn Watch of this cruise. You're in the control room with the Donner Party, D-A-W-N-E-R Party. We've made it this far, and as yet, no one has been eaten. That's Haley at Logger 2. Rachel and I will likely be trading off lead, watch lead duties today, pending awakeness. At the sticks, you have Alex and Ben. Right now, we're on the hunt for clams that are close to the last clams we collected, but not the same clams.
Maybe we can do supers in the meantime.
Yeah, if we're waiting on a response, well, yes. In fact, I'm a little bit surprised. That's a very good point you make. We should probably do these supers before we start tearing up clams.
Yep.
So we're gonna pivot, and please drop a marker here. Let's call it. Let's call it clam 6. People evidently think they have seen a lot of clams at this site. Maybe they did.
Or were they mussels?
No.
We are lost.
Do we have that marker?
Lost
Cool.
hopes and dreams.
Okay. Clam 6, definitely live clams. So once we have the marker dropped, we can swing about and transit back to Chimlet 1, please. 10, 15 meters. Chimlet 1 was where we just came from, that's right.
What? I heard my name.
No, you're good. Stay on target.
What is my prime directive? Turn on the super sample where please superpower on. Okay. Are you ready to move, Jacob?
We are moving back to Chimlet 1.
Oh, but it says east from Chimlet 1 at marker super 1.
Right. So we, I guess they've left us markers, which I didn't note, but we need to start at Chimlet 1 first.
Whoa.
Let's.
We need to go to super marker 1.
Do you have that up on your display, Alex?
Super marker.
Yeah, I may have to.
There's so many markers.
I may need to lay it in from the list here. It should be
It
to
is
the east.
Yep.
Clear this.
And it says right at the edge of a mussel bed where the mussel bed ends and bare basalt begins.
Yeah, it's looks like it's really to the northeast, east-northeast.
Got it.
We've got, that's at 60 degrees now.
Yeah.
16, we're at 16.
Nice big arc. Are you? Oh, I see. You pulled around. Okay. Yeah, we just need to go to this other point. Sorry, the revision.
Now we're going to go 14.5 meters, almost directly east to the
Okay, we're at the Chimlet now. Cool. It just says where the mussel bed ends. We're not looking for clams at the moment. These clams don't look very happy. Super 1, yeah. Okay. When I call out when you're ready, Rachel. We can probably deploy the temp probe and the Super Wand.
Do you want to land?
Right at edge of mussel bed where mussel bed ends. 4 super samples at varying distances. Do we know how high we want these from, Rachel?
That's a fantastic question.
I'm going to checking the objective sheet. Wait, which is disappeared. Do you have an objective sheet over there? There it is. Thanks.
I assume the seafloor is fine?
Right at edge of mussel bed. I think we should land. Thank you. Yeah.
Do we see the end
Of the mussel bed?
of the mussel bed?
We see some dead clams. May I tilt up?
Okay, we're looking away from the.
We're looking away from the chimlet.
Okay,
We could back off a little bit if you want visual confirmation. You want us to back up?
yeah, just a little bit.
Can we just come out? Or you won't run into anything?
Go up. Chimney's behind.
Sure.
Okay, so
Stand by. We're gaining some altitude.
Want to go back to the chimney?
Not all
No,
the
no,
way.
no, no, no.
Just push aft like, I don't know, 5 meters.
we're just double checking our position.
Right at edge of mussel bed. Bare basalt ends. Are we north of the chimlet? Well,
What dive is this?
yeah, we're a little bit north. I mean, we're a little bit north. We're northeast of it. Well, I'm confused by the fact that they said.
575?
Sorry, I think there's probably something wrong with the marker. Yeah. So Rachel, we've
Yeah.
backed up, better part of 10 meters and haven't seen any mussels, but also the marker is not due east of the chimney.
I think what's more important is that we are like just past
We
this.
better keep that down.
So going back distant from like basically returning where we just were. So instead of backing up, going forward and going just a little bit past this bed of broken up shells.
Okay, because there are no mussel beds here.
Well, I don't think that matters so much.
Okay, because I thought we were going for. I mean, you know more about this sampling campaign than I do, but here it says right at edge of mussel bed, where mussel bed ends and bare basalt begins. And
Well, this is like the outer part of the transect, so like
this is the outer
yes.
part. I thought is our point of reference here for the inner part the chimlet, because it kind of looks like. I'm just trying to parse this out with you.
Just
Yeah, take your time.
Looks like just due south of us there's mussels, and that's to the east, isn't it?
no
Yeah.
visible venting or
I mean, we can also just well, wait. We can also just do it on the marker and check our box and
right.
call it good. I mean, they left the marker for us, so perhaps
I
we
mean,
just follow
we
directions.
could at least do the second one, which seems very easy. About 30 meters east, past marker super 1 area with no visible venting or clams or mussels. Like that seems
That seems like
that
low-hanging
seems
fruit.
like a
Why
more of
not?
a sure thing that is less ambiguous.
Okay, Ben, thanks for your patience. Let's press ahead on our current heading, 35 meters, please.
From here.
From here,
Okay. 35.
35 meters.
Descending.
Yeah, basically 090. Good call.
35 meters. Straight east.
And then we can see if we can hit the muscle edge on our way back in. Watch there be a gigantic mountain of muscles where we end up.
Well, yeah.
I'm joking. Sorry.
You never know.
Exploring the unexplored.
Except we don't really have any. So many dives.
Probably 5 years ago, when we realized that's not the case. Sweet Ocean gives me. I didn't think it was the case. I was like, well, that's pretty good. It was, whatever. That's how that cursive had to do with it. Serialized into license and value.
Great. Looks pretty bare, which is a good sign.
There are no muscles here.
There's a little siphonophore, I think. Yep. A Calicopsidae. Let's see if we can mimic that down here.
Ten years.
10 meters. Would it be helpful to you if I picked up the Logger 1 computer while you're supering? You've got both,
This is
okay.
very easy. Yes, this
All right.
is very, very easy.
Haley's double fisting.
Land,
We are going to land, please, and deploy the super nozzle and the high temp.
Yes, we're facing
The dive plan says we're carrying the pH meter. Are we in fact carrying the pH meter? Take your time. Okay. I can check in a little while. Look at the camera. It's an interesting fish. Don't know who that is. Looks like a gaddig. All right, we've landed. Let's please deploy the high temp probe and the nozzle. Yeah. Well, okay. So the reason I wanted to verify that we have the pH meter. Rachel, if we do have the pH meter, should we measure pH co-registered for half of the pump?
That's not a bad idea because we will be pumping for 22 minutes.
All right. Give us something to do. It's not noted on here that these are 1,400-second pumps.
Nope.
Did you just get news that they are?
Wait, get news
I
that
believe
what?
you. That these are long pumps?
I've known that these are long pumps.
Gotcha. Okay. I'm glad you know because it's not written down anywhere. But I can find it.
No, it was only in the chat for the super team. Your time.
That's why it doesn't want to stand up.
I'm going to super 4.
So this will be the first of 4 super samples we collect today, examining the chemistry leading away from a mussel and clam hotspot. This is the most distant point on the east running transect. It's pretty clear that there are no bivalves here.
Can we pull out, can we pull out the super wand?
There, yeah. Yeah, there it is.
Kaylee, would you mind passing me that pen?
I figure we'll just go as far as we can with this without entangling everything and then just move the height through. Unless we need to get down somewhere that's going to revolve. This is it. So what height would you like that? Not to see that? How high? 10 inches?
Sure.
That's what we've been doing over the animal beds previously. Right.
Yep. Can we just line up the super wand and the temperature wand? Perfect. Gorgeous, hun. Okay.
Show off.
It's time for impressions. Okie dokie. We're gonna start with the bypass of number 4 for 120 seconds.
Yeah.
Purging now.
Now.
That's my Christopher Walk.
I love
Could you tell?
it. I like it.
Is it Super 1?
No, this is Super 4.
Okay, Super 4, thank you.
Oh boy.
Fasten seatbelt sign will probably turn off at the end point. You're free to move about the cabin. We just ask that keep your seatbelts on when you're in your seat. Folks, this is thank you for flying. Where is this? See, there's Sebastian.
Can I play around with the camera a little bit? Do you need to see the probes?
Oh, no.
Rachel?
Can I see? Oh, it's over here.
What do you want to see?
You're ready.
I'm good. Do what you want to do.
Live your life.
It's your thing.
That's
Do what you want to do now.
Which one is this?
one. I'm just trying to. Yeah, we might have to rack it in a little bit to see to double check the pH probe.
It's.
Thanks.
I'm gonna call it background.
I don't
Regular
see
or
a pH probe.
Other.
Okay, I had a feeling Pete might have just packed it up.
30 meters east past marker super 1. Just literally just.
Did they catch?
copy this
Did they
whole
capture them
thing.
intentionally, or did they just kind of volunteer with the muscles?
30
Okay.
meters
So I know it was Stuart had been talking about getting one. Match
Purge is
heads.
complete.
Purge complete. Okay, we have no pH probe, so nix that.
Oh, well, no. Look,
No,
that was
don't
just
nix
the pur-
anything.
That was just the purge.
I know.
We got a long time to go.
And we're not going to spend any of it measuring pH. Just heads up.
That's right.
We're not?
We're not. We
Oh,
don't have.
'cause
We don't have a
it.
sensor.
Yeah.
All right.
Rack it back out, please. Can you get a chance?
That's fine.
Okay. I know it's that. I don't have to bother you about it.
All right. Ready? Pumping now.
We've been seeing a lot of these little crab larvae. These little zoea. Kind of cute.
That's what those are floating around.
Yeah, it's like a crab with a tail, right? Its tail hasn't folded up yet. These are interesting. I guess these are probably the cyanogrea crab larvae, and they seem to metamorphose pretty late. And depending on the species of crab, their tail will fuse and they'll settle. You know, could be
All
just above a
regulars.
microscopic size, or sometimes they get. You know, I figure these are a couple centimeters, 2-3 centimeters wide, and they're still larval. Same thing happens with other crustaceans, like spiny lobsters, for instance. The ones in California, you pretty much never see them in their larval stage. They're still pretty tiny. But if you go to a lot of the tropical species, if you go to Hawaii,
Oh, that's a pretty
you'll
fish.
see lobster larvae floating around in the midwater. They're perfectly flat. The name for them is phyllosome, which means leaf body, and they're like that big. They metamorphose really. I don't know if it's late or if it's just they metamorphose larger, but giant larvae.
What's the name of the kid? The little thing? Alfalfa.
Yeah, I don't know what kind of fish this is. Maybe one of our online friends does. I'm not a fish person.
Not that kind of fish.
I mean, I am a fish in an evolutionary
We
sense,
all have an inner fish.
but not a fish person.
Fluorescent
I need to make a note that we're not actually carrying
Yes.
the pH probe.
Actual fish. Can I get that sea log
You
timestamp?
may get that sea log timestamp.
Thank you, thank you.
That is 10/30/21.
10/30/21.
Indeed.
Okay,
Is your latitude?
latitude.
Yes, 2.104033.
33.
Indeed.
Indubitably.
Okay.
Okay.
Negative 91.926075.
See what else we've got here. Oh, this is one of those flatwormy things. That's cool. I forget. I don't think it's a proper flatworm. Maybe it is.
Do you have the depth?
The depth. No.
1667.24.
Might be a
Do you have the altitude?
nemertine worm. I would have to check our animal guide.
7, 6. Thank you.
I don't know that it's properly a flatworm. I think it might be a ribbon worm.
Weird worm.
Rose worm.
Fake worm. Not a real worm. Either way,
Faux worm.
there's lasers on it.
Are those little, who are the little guys? Circulids?
Yeah, they are. Do we have the animals for geologists thing?
It's in there
Yeah,
somewhere.
I doubt it has this guy on it, but
No, it doesn't.
That's for
oh
sure.
well.
Too rare for us.
Yeah, there's serpulids, there's harp sponges, there is a cute little
Who is that?
mushroom coral.
Get some of that eDNA.
Don't know why they call it that.
Mushroom
Doesn't look like any mushroom I've met.
Looks like a everlasting gobstopper.
It's sort of
It really
retracted.
does.
Yeah, right?
Yeah.
Wait.
An everlasting gobstopper.
Yeah, that's what that guy is. The Latin name will come to me in like half an hour.
Absolutely. If Timothy Chalamet handed me that, I would stop
Thanks for
that
playing
call.
with the lights. I'm gonna probably keep you on your toes.
Jacob's on the lookout for that Benedictino.
Yeah, you read my mind.
Darth Lobate.
Darth Lobate.
I mean, that happened the last time we were in the middle of sampling.
Well, that could have been the larva of Darth Lobate for all I know, but
It was black.
it was. That's it, but we've seen black sidiphids before. We've never seen a black lobate before.
Oh.
That's what, yeah.
Shame on me for not knowing.
No.
No.
Shame.
No.
Let's just make the charitable assumption that if I sound condescending, I'm just
No,
tired.
not that at all.
I'm not
No,
being
I think there's only
self-deprecating, I'm
one.
just being sarcastic.
Yeah, I think there's only. I don't know.
It's fine.
So if anybody out there somehow is wondering what Darth Lobate is, I'll note only that you will know it when you see it.
Yes.
Except Rachel, who does not know him, and she does not see it.
Latina for me.
It could very well be that the black sidipid ctenophore, which is a ctenophore with 2 tentacles, is a larva for Darth Lobate. We were just discussing how, we were talking about crustaceans, but organisms with larval stages, throughout a group, there can be huge variation in how big the larva gets before it metamorphoses. The same actually goes for ctenophores in the tropics. We saw some of, earlier on this dive, on the way down, we saw some of these big lobate ctenophores with gigantic lobes. Remember seeing them like these big, beautiful 4 sort of shiny lobes?
Clover leaf.
Yeah, exactly. And they have sidipid larvae, like all lobates. In California, those sidipid larvae grow to at most half a centimeter, and they metamorphose. They grow their lobes, and they lose their tentacles. In Hawaii, it's a different species, but the most closely related species grows to upwards of 2 centimeters across. Like, it looks like a full-on adult. You can see it with the naked eye before it metamorphoses. And larvaceans never metamorphose at all. Peter Pan of the invertebrates.
For stabbing the Tina
I found
Sudden but inevitable betrayal.
I found SpongeBob.
Just like
It's right there.
I've got something else to say.
Wow.
Fascinating, but
Wow.
This? Yeah.
turning
I
out.
mean-
Why are you turning on me?
Well, I don't know. Maybe I don't want to be.
It's the Donner Party. It's more authentic.
Today the day. Today the day.
Who
you
is
find
that?
your
That
father.
looks like a very large circulate.
Right here. Right here in the Stop and Shop.
Huh. That's a different species.
Look, I'll just
A lot of flow. I wonder where it's coming from. For station keeping. Okay. What is the second thing? Get a load of that.
The thing that's
The
like,
second, the worm on the left,
like
the really
the
weird
worm
one.
is
like
not the
it's like
circulate.
it's like licking it a little bit.
Alright. Observation.
Yeah. The polychaete. Whoop, sorry, I should have held in there for
No,
a while.
I got it.
You
I
got it?
You're good. I got it.
Licking.
There's the crab larva. Interesting spot.
I'm gonna go get coffee. Does anyone want anything?
I would.
We all want coffee.
Well, I can make this.
How many hands do you have, Rachel?
Do you want?
You would? Or how do you like your coffee? Just black.
Why don't I go with you, Jacob?
Why don't you just get one of the buckets from the lab and fill it up?
Yeah, the one that
Thank
had
you.
dead animals in it.
Maybe not that one.
I give it.
Try the sulfide bucket instead. It's got a good lid.
I didn't murder any animals.
Oh man,
I did
my
not
lucky
murder
day.
any animals. No, I'm not on the animal murder team. I I am on the rock.
Is the super just like?
It's just sampling
second?
for 20 minutes, so you guys can have fun.
Is there? Do we have an ETA? Just I don't know in case you.
I won't be up there that long.
All right.
Yeah, we just take that.
No, we're just gonna pump the rest of this dive.
Black coffee?
Yes, please.
Black. Is this Jacob's? It says Jacob. Okay, anybody else? Ben wants black coffee. Josh, Alex. Good. Alright. The world out there, everyone need coffee? Okay.
All these samples are gonna last Roxanne and friends a long time. It amazes me too. I'll be amazed in 3 days if they all get there. Trust. I mean, I've been pretty spoiled, working mostly with domestic samples. Though I have to say, I've had really good luck. A number of people have collected internationally, foreign meat, shipped me samples, and it's always gone pretty smoothly. Like, in what sense? Sure, and that's why. So all of the rigmarole we have to go through, the forms. There's oh yeah, of course, but chances are a person who are actually carrying something dangerous, if they with intentional nefarious purposes, wouldn't probably carry it in a case full of tubes. Hard to say though. Yeah.
Are you describing like the concern about something we don't know, like someone who doesn't have any ill intentions?
Right.
We just don't know.
Carrying something accidentally pathogenic, and then taking it on a public aircraft or whatever.
Or even just relocating it, like just you think about the USDA, the way they regulate.
Absolutely. Yep.
Across state lines.
And depending on what we were bringing back, for instance, if we were working in like the mesophotic kelp forest on the Galapagos and we were bringing back kelp samples, we would probably have to go through USDA screening. Right. As it is, we do not go through USDA screening, but we go through U.S. Fish and Wildlife Service screening. Right. And they want to know every species that's in every tube. Count them.
And that's more of just
and so on.
Those people don't know.
No, they don't know what they're looking at to a granular scale. Right. That's absolutely true. And yeah, I mean, I guess that's one of the persistent fears that comes with globalism. Anybody scientist or otherwise, I mean, you're absolutely right. We could do that we are intentionally transporting live materials sometimes, right? You never know. We can just do our best. Scientists, of course, tend to have their own motives for keeping things contained, because if you have contamination within your lab, all of your experiments go to seed. Like you don't want one thing getting into another tube or chamber or whatever. But it doesn't always work. Pete was just talking about how his house, I think in Acton, which is outside of Boston, and there's a house some quarter-mile distant from his, where a former Harvard faculty member, like a few decades ago, lived, and he worked with gypsy moths, and he brought some home and did some experiments in his basement, and they got out and completely devastated the trees of this town. Right? That's what gypsy moths do, and they really liked, I forget, maples or oaks. Or they like, I mean, the ones in California anyways, eat oaks. So yeah, it happens. I think that we have the deck stacked in our favor safety-wise by working at these vents, because the environment is so different from anything we see on the surface. But you never have a guarantee. No, go ahead.
So it's kind of curious to me to see like a lot of the fundamental microorganisms, you can look at bacteria, which
to that point, we have found that a lot of these bugs are pretty stable in viable form. For instance, we can take these sulfide samples, put them in a sealed container with no headspace, the oxygen's been scavenged out with residual sulfide, keep them at more or less breach to room temperature, bring them back, and they show, like, you wake them back up in a bioreactor basically, and they show most of the same metabolic activities that we associate with vents, like being able to oxidize methane, being able to oxidize sulfide, and so on and so forth. So yeah, they're definitely viable.
That's right.
And some of them are probably anaerobic digesters here or there. Whether they win out against the things that are already dominant there, who really knows?
Right.
But I bet that's a thing. A lot of archaea too, but I mean, while they're distantly related, they fill a lot of similar micro ecological niches.
Still going.
You guys are probably, thank you so much.
Yes.
You're probably aware of this, but the lasers are kind of tweaked.
Yeah.
Would anyone want an Oreo? Found a six pack.
Oh my gosh, kind of sick.
Got a secret stash.
Don't give them the whole thing, they'll take the
Oh,
whole
thank
thing.
you.
Last time you just ran out of the whole thing, didn't share.
They're free.
Sharing is caring.
Sharing is
Go. Anyone else?
no Oreos.
Anyone else? You want an Oreo?
Yes, please. Are there enough?
There's
For everyone?
Oh, look, we have 66.67% left. So.
Don't you worry.
What kind of precision is that?
Math?
Math precision.
66.67.
Yeah, because there's 6 Oreos. I gave away 2.
Math.
1/3. Math is math.
Rounding error is error.
Excuse me. 66.6666666667.
There you go. Much better.
Yeah, like 10,000 times better. Thank you.
Oh. This was not ours. That was sitting up there this morning.
What was? Oh, the water bottle?
Mm-hmm.
What?
Let's stretch it a little
Let
bit.
your Oreos dunk
Yeah.
them whole.
And savor it.
The texture change. There's one of
Double stuff. I haven't bought Oreos in a long time, but yeah, I appreciate the double stuffing.
It's kind of wild, like the marketing gimmick for like the multi-stuffed Oreos, because it's just like way cheaper for them, because it's like the filling to cookie
Yeah.
ratio,
Yeah.
and you get way fewer cookies.
Yeah.
But they charge way more.
Yeah. Well, I check this out.
I can't. I don't think
Cookie
my body's
conspiracy.
capable of handling that sugar.
These are our friends. These not monsters.
There's an amphibot.
Yeah. So you make up for it when you're out here. Nice. You're trying to catch up.
Are spot lobsters on our permit?
Do we have a Do we
It's
have
too
a
late.
too late to
We've already
collect them?
done all of our export
Yeah, yeah.
permits.
They're not on the export permits. Do we have some ballpark of the beam distance, or are they just not parallel at all?
They're just
Oh,
not.
we're talking about
Well,
the lasers.
they're
I was gonna ask
this
about
wide,
super
but
circle.
was always
But you're never going to stop super.
Oh, really? Okay. They're still 10 centimeters apart?
Yeah, that part is about exactly. It's obviously not perfect.
Yeah, yeah.
It's worse vertically than it is.
Right, right. Okay. Thanks. So close range, we can still figure on 10 centimeters horizontally.
Better than
Yeah.
that one.
Still wanna know.
That's pretty far.
What spot?
Yeah, yeah. Or-
It's not weird.
No, you're not weird. But they taste weird.
Why do you think they taste weird?
I don't know. It feels like
When we
it tastes
published a
they-
paper on
they
the
run
record
really
largest
big
swell
heads.
lobster ever observed, and
I bet they taste better than crabs.
No, I said when we
Crabs
do,
don't have much
you know,
flavor.
because we just took a
Oh,
picture of
the
like a 30
crabs.
centimeter
The
small
bent
lobster.
crabs.
I
Also agree.
Yeah.
Lobsters are a
Mm.
superior,
So, morning, Rusty.
superior food item. In
We're
all
at the
respects.
distant end of the east right transect, and we're pumping.
Yeah, cool.
How we got here is, well, we went to, okay, so the clam collections, I gather, were first on the dive plan. So we kind of started scouting them first, then we realized that we probably wanted to do the supers before we started.
Yeah.
Trying to learn lessons from last time. So we went east from Chinlet 1.
Yeah.
We went to the marker that was left. Didn't see a clear edge of a mussel bed there. So we saw a lot of dead clams kind of interspersed in basalt. So we continued out to the 30-meter east site where there are clearly no bivalves.
Yeah, right.
We're pumping. The thought is we can turn around and head back in, and once we see the margin of the mussel bed, maybe
Yeah,
we'll
that
stop
sounds
there.
good. Yeah. I
Um,
don't know if maybe the marker got trapped at the rock spot. But
Yeah, I
it's
mean-
kind of hard to tell because it's like there are going to be dead ones, and then you'll start to see some sparse live ones.
Right. Though I think that what we were seeing mostly at that point
It
were
was
clams
only,
rather than mussels.
it was clamshells, and it was like pulverized clamshells.
Um,
They've been dead for a while.
Now, we did, when we were still kind of in clam hunting mode, we dropped a marker that I don't think is on the display right now. It's off to the southwest. If you don't mind dragging us over there real quick, Alex, please. It is clam 6. So there's the chinlet, and we basically sat on the chinlet and made a 270-degree turn,
Okay.
sweeping for mussel sites that didn't match the bearings of either of the previous,
Yep.
or for clam sites that didn't match the bearings of either of the previous sites. And this one
Okay.
is it's I think about 15 meters south of Chinlet 1, and a little over 10 meters outside the radius of the nearer of the two
Okay.
old sites.
Great.
It definitely has live clams, so we
Perfect.
can
That
use that
sounds
if we
great.
want.
Yep.
Beautiful.
I haven't seen what the vinyl boxes look like, but
Let's have a look.
it's going to be a little tight, I think. But
Yeah, you want to have a look? There's two. Yeah.
It's very active in
We
there.
have 2 crabs.
Actually, this would be a great opportunity to aerate the crabs, probably, while we're sitting here.
Married crabs.
And they have those buddies.
Or mussels on the right-hand one.
Yeah, mussels and
Um,
both.
Yeah.
Yeah.
Yeah. So basically, we need like 15 clams that are big. So we're going to try to get them into both boxes. As long as we don't put any mussels in there, it should be fine.
While we're pumping here, could I trouble you to give a couple flaps on the box doors,
Well, maybe
please?
don't
Because the crab might climb out.
Well,
Oh.
no, because it's going to change the water.
Oh, good point. Yes. Sorry, we're actually looking for mussel DNA and stuff, so maybe not. Good catch. Thanks.
Oh, right.
Probably doesn't matter much in the how much volume you put through.
Well, this would not be the site to do it at. If we're doing it on top of a mussel bed.
Right. Yeah.
Do you know, are these the flat things ribbon worms?
I don't know.
Okay, I should get. You're gonna rack out, please. Thanks. Go ahead. This can't be the same crab larva. What do we know about these? Well, have you done any biogeography on these crabs, on bathygrade crabs, Roxanne?
Yeah. I know we're
Yeah. Oh, come on.
Little gobstopper. He's like.
Oh yeah, it's locked in.
No.
There we are.
They're so cute when they're in
Is it
focus.
gonna settle?
Oh. Well, there he goes. Bye, guys.
Sorry.
Alright, super complete.
Wow.
Complete. God, that's so fast.
92.65 liters.
Can we get ready to pick up?
Yep.
Or we gotta do a super somewhere else.
Oh,
Yes,
we
we
are.
are,
So
yeah,
keep
we
the
are.
super stuff deployed, please. Let's pick up off bottom and head due west 270. Until we see muscles.
Until Roxy says stop.
We're hoping to go back to the approximately super one site, but we'll see what else I can do.
Yeah, so maybe rather than a 270 heading, just
Supers and 271s and 270.
great. Then they're the same. Then we're good. Let's head 270.
Love it when the plan works out. No, we're gonna go back to super.
Oh, that's right.
West.
Right. Super 4, hun.
Sure thing. Sorry, I'm just trying to center up. There we go. That looks like the edge of a muscle bed. What do you think, Roxanne?
Yeah, there's some live muscle right there.
Yep.
Yeah, so this is great.
Okay.
We can set down and basically, if I understand right, we want to sample right above that margin.
It doesn't have to be very
Okay.
close. Yeah, this just generally this is okay.
Okay, yeah, sit down and we'll get started. No. It's we're okay right where we are. How far off of the marker super 1 are we? We're on it.
Yeah.
Okay, then I don't think we need to create a new marker.
Yeah.
Okay.
We were just facing the wrong way.
Yep.
And backed up.
Not far enough. What is that rectangle? Oh, it's a skating, right? Looks like a piece of wood. It might be a piece of wood. Oh, it's a part. No, it has a bolt through it.
Yeah.
One of the weights
Oh, it's one of our
down.
plates.
And it is a weight plate sitting on its side.
It's
and we're looking at
ours.
yeah. Lander's gap.
It's like wombat. Little cubes.
That's the one we forgot to release before we started flying. So there's no rope on it.
Oh.
Are we happy here?
I'm happy here. Are you happy here, Roxanne?
Yeah, looks great.
Okay. I am going to start the purge on number 3. Beginning now.
Third super. What number?
3.
Trace.
Good grief.
Yeah, my dad called me total black tuna.
Oh, fish.
Fish.
Yeah. Which one's more important?
He's just.
He's
swimming.
really struggling against that current.
Yeah, dose fish.
Dose fish.
Trace fish.
He's barely out of the plankton, that little guy.
You got it, guy. Believe in you. Oh, little one.
Say hi to our little boundary layer fish friend.
Aw.
Nice barbels.
Aw. Hi, guy. Oh,
Is
goodbye.
that the snailfish?
I don't know. I don't think so. No, no, no. He's a.
He's got too many whiskeys.
Too many whiskeys.
I love him. He looks like a little version of the guy we saw earlier.
We're gonna run it
True.
up against the.
Frogfish.
Yeah, that's what I think.
Yeah.
It looks like a larval that.
Alright.
Oh gosh, I get a little
Purge
vertigo.
complete. I like the big chimera.
Light.
Oh, I love them
Wait, let's
too.
see.
At least they're in the correct direction.
I just like tennis.
Yeah.
Alright.
Alright, pumping now.
11
Something super 4.
That's a setting.
and 5. Alrighty.
Give me that data.
That data is the following. 11:02:53.
11:02:53.
Latitude
Latitude.
2.104015.
Okey-doke.
Another little one.
Longitude
Longitude.
-91.92636.
926,363.
Yes, ma'am, indeed. Baby squatty.
There's another one of those little
That
fish.
is a wee squatty.
Right there. See it?
Okay. May you also share the depth
Ooh.
and altitude.
1666.
Oh.
0.39. Altitude.
Altitude.
Are we? No, okay.
0.97 meters.
It's lagging. It's
0.97.
not actually in
Chemosynthetic
auto.
animals, we have clams
then
and
there's-
mussels.
Land animals.
Is Roxanne back there still?
Nope, she left.
Yes.
Okay, well, I'm wondering whether she might be interested in this snail.
In the what?
The
This snail.
The snail.
The snail?
Do you see it?
That snail?
Oh, I do see snail, but we have not. Have we collected any snails yet?
Well, we've collected limpets. I mean, there's the export permit issue. But
Yes,
I'm
that's
just curious.
I'm alluding to.
I'm just curious. Stuart might also be interested in it, in which case it won't be exported.
I like our, well, maybe Stuart wants it.
There it is.
Oh, beautiful snail.
There's some. Oh, that's a shrimp under there.
And
I don't know if
Right
the snail's
away.
alive. I just haven't seen very many of them.
Aquaticus.
The eDNA here will be pretty rich. Just gonna sit here and wait for Roxanne to. Well, if we think Roxanne will come back.
We got plenty of time, that's for certain. Look at all these little babies.
Yeah. The nursery is a tiny little picnic owner at the bottom.
Yeah. Well, skate eggs and
Those look like they've hatched.
all this.
Yeah, that one does look pretty raggedy. Probably hatched.
Spider, I say no.
What is? Oh, it's the edge of a clam. It's looking weird.
More snaily.
Oh,
Look at that very living snail.
Let's zoom in on this.
Where? Oh yeah, there it is.
Living snail alert.
Cruising.
Observation. Brochure live. Snail.
Or is that the same snail?
No, it's a different one.
Different snail.
Same species, different individual. And that's full telephoto. Yep.
That's okay. I've got an observation.
I'm so excited to see a snail. That's where we're at.
Hey, snails are inherently exciting.
Apparently.
Snails.
I said inherently, not apparently.
I thought you said inherently.
You know, I'm not gonna be as psyched about it as Ian would be. Ian would be like, you know, that
We
that
have
snail is
either a
so
pooper or a
dope.
spawner.
It
See
would
that?
be real clutch if we could collect it.
I think it's spawning. Mussels are notorious for sort of leaky spawning
We got all
when
these
they feel like it.
Ew, gross.
It's the circle of life.
It's not safe for work.
That's right. All these babies.
You should write that observation. The snail spawning.
Mussel spawning, not snail.
Also
Not
spawning.
snail.
How do you know it's not the clams?
Because it was coming out of a mussel. It's still doing it. Cut in the act.
It's like this,
Ew.
Right there.
this nastiness.
I'm pretty sure. It could actually.
Okay, spawning.
Could be coming from that clam.
spawning, Potentially coming from.
Oh, get a closer look. We'll invade this bivalve's privacy.
I mean.
Oh, I
it's
can't.
Like,
I can't get any closer.
Is it
you know, public
from mussels?
indecency.
I'm gonna get in trouble, guys.
It is a clam. You're absolutely right.
Clam.
You're absolutely right. It's coming in right there.
Spawning, potentially from clam.
Ew.
Yeah.
Cut it out, guys.
I think about other snails alive.
Oh, this is gonna be a fun super sample.
It's gonna be interesting.
Comments. We saw a clam spawning.
Yeah, it is. This is very zoomed all the way in. It's pretty far away. You got your observation?
Yes.
Yeah, do clams, are they hermaphrodites or do they?
Generally, I believe they are separate sexed.
Okay.
There are often exceptions to these rules, but yeah, I think.
I want to see some skates.
What's that?
Yeah, we've not been able to get any skates on our watch.
I think they only had the one skate that they
I
saw.
think there were two.
There were two.
And it's probably the same one skate. Because it was this area. We saw some clams spawning.
More snailage.
But yeah.
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 Yeah. Do you want to look at it again?
Yes.
Yeah, let's see if it's still going. We also have these little snails. I was
Yeah.
curious whether you're interested
That
in
it
them.
is not spawning.
Interested in the snails?
Slurp some
Okay.
But after we're out here,
Yes.
Yeah. Okay, where was our...
it was off to the right.
Off to the right?
No,
Left.
never mind.
Left.
I'm full of garbage.
It was
Don't
for
be
sure left.
hard on yourself.
Literally, figuratively.
2 hours of sleep will do that.
I did just eat a pack of Oreos.
No, it's good.
Yeah.
He's got stuff to do.
Oh gosh. Oh, it was near the. That's our reference point. Is the hatched.
Yeah.
That's right.
That's perfect.
Okay,
That's
and
right.
then.
And those to the right of the hatched eggs.
There
And it was. That's right.
Yep, yep.
It was up on end.
Underneath that squat lobster. That's the one.
Which squat lobster?
The one on the right.
The one all the way over there?
Yeah.
It was no, it wasn't. It was more standing up more straight than that.
What?
I'm
Right?
sure it was to the right, not the left.
It was that one. It was that. It was the one with the shrimp on it.
It's the one that's
Right there.
You're the nefarious
But it's
one.
done now.
Oh, it's
Oh no,
done?
it's still going. It's still going, and the shrimp seems.
Enjoying it.
It's spawning.
I don't. Or it was spawning. I don't really see it anymore.
We're
Oh no! There it is. You see the wispy stuff coming out of that clam?
Mm-hmm. I see it.
It's a little sparser now. It used to be blowing to the left.
Oh, yeah.
There's some.
Get yourself
Last dribbles.
I did.
We have been watching.
I'll do it again.
It is uncomfortable.
Spawning again. We're the fans.
It's just.
Can you note that in the notes?
I think
Yeah,
it
I
is.
did. Yeah.
Yeah. Is the shrimp feeding on it? The shrimp wasn't here before. The shrimp.
I think he was. I thought I saw him, but he was.
Well, it was
like,
around, but it
tucked.
wasn't hanging out right there.
You're just thirsty shrimp. Thirsty shrimp.
I mean, yeah.
I got so many notes.
So many notes. So much.
These two are right next to each other. It's like a half chair apart.
The red and the orange.
The red and the orange. And then the green is like 2 meters away. But it's clearly near another event. And then it's exhibiting like the opposite kind of temporal pattern.
They're no baby fish.
Wow, I love it. It's so cute.
I'm so glad that we are done with these.
Yeah,
Beefs are out.
Beefs have always been out.
Little
The
doofuses.
blue one is a little.
Except it is fun to watch them like run into things.
Yeah, they're like,
Yeah, they
Like-
just like run into the earth.
they're like, stop. Oh,
We're gonna finish this pump, and then
Headbutt.
afterwards
There
we're
are
gonna
just a few nodes
slurp some
along the
snails.
fiber
There's
from
one that's close to
Just
us
give up.
time series.
that I want to say is like behind the
I
left.
want
We
to see
obviously
your
got
ones
some
do
Hopefully it'll be easy.
This is like the full length of the fiber. See, that's probably where the similar muscles mentioned lower there. Yeah, and there's a few other sites. The coolest thing I've found so far is that this is kind of like one chimney. This is a chimney that's like 2 to 3 meters away. And they both have this kind of tidal periodicity here.
Very cool.
But they're inverse from one another.
Hmm.
Oh
So
yeah.
close, which is
Huh.
kind of cool. Yeah.
Does that imply different aperture diameters because the pressure differential has a differential effect on each chimney? Or I should just say different flow resistances.
Yeah, it's possible.
It's like Ohm's law, right? Like, well, I mean, you all are hydraulics people and probably know this better than I do. But like,
Sorry, go ahead.
no, go ahead. If one of them has a higher resistance, then the pressure differential, which is affected by the tide, should have a different effect on the two chimneys. So I would expect that the whichever one has less resistance. This is just one model. There are probably other reasons it could be happening. Whichever one has less resistance should not be, its flow should not be suppressed by the tide. And basically the one that's more choked off, when you lower the pressure differential, it chokes off more proportionally, and presumably that flow gets diverted to the other chimney, which would result in
Yeah,
the inverse effect.
that's what I was thinking.
Okay.
Yeah, like if it's the same like
Source.
source water
Yeah.
Yeah.
That's cool.
Yeah.
pressure gradients and all that.
You could think of it as like two resistors in parallel, right? And then the tide is varying the voltage on the lower leg. And the current, you should be able to come up with a system where the current for each will follow like an inverse pattern.
Hopefully from the video we'll be able to see apertures as well.
That's really cool.
Yeah, that's cool data. Congrats. the squiggly line that makes sense.
Oh well, I was saying
Much better than squiggly lines that
it's
don't make
analogous.
sense.
Yeah.
Those are the
Right.
worst.
I also don't.
Especially squiggly lines when you don't want squiggly lines.
I don't think that what I'm talking about is exactly Boyle's law either.
Smooth
Boyle's law is
like
about compressing gases. But I mean, you're still named after. It's
Yeah.
I can never remember. It's
Disrespectful to appear in my data.
well, I guess
How dare you?
I think it might like the Hagen-Poiseuille law might be the one. That's where like how you calculate resistances based
The
on
other
cross
one
sections.
is
But
a little less interesting. The Northwood one.
I just think electronics better than I do
Not as
hydraulics.
many points of interest, yeah. But there's still a few like useful spots.
Cool.
Still going? He's still going a little bit.
Yeah.
Little weirdo.
All right, we came, we observed.
We observed.
That's really borderline, Rachel.
Any last words?
I think that's the snail we're going to target. Afterwards, you see it? Unless you spot another one, it looks easier.
He's meeting with ONR. Yeah. So we've been chatting this morning and afternoon.
Because I'm a doctor doesn't mean I have to be boring. Trying to liven up our super sampling. I love it. It's great.
Okay, manual. Good.
But this is we are now going to be funded for this project.
How's our
Starting this.
Looking great.
pump status?
It pumped. We started actual pumping at 11. So you got it.
Oh, okay. Yeah.
Yeah, we're like 4
Yeah.
minutes.
Sign up, but it's
About 4.
this guy who
And then we
works
slurp
for
in the
ONR
snail?
with those.
We're slurping the snail after we're done
So we're
slurping
gonna
the super.
slurp the snail,
Well, yes, not currently, but yeah.
and then we're gonna do this again
Wow.
On the northern transect,
on the
right?
northern. Yes.
Wow, what an exciting site.
Little Chim does not disappoint.
No. We love them.
If we have time for the end of the cruise, I'd still love to get. We can work with J-Rod to get the little
Yeah, I'm
3D
gonna
printed map.
probably do some the Happy Mappy
Happy
hour.
Mappy, we get high res
Yes.
3D printed.
3D printed map of the entire site or of Little Chim.
Of Little Chim.
Just
What resolution can you get that?
Well, on the M3, it can go up to like 10 to 5 centimeters.
Do we have the data?
Yeah,
So we
we're
could
gonna get
see
some data.
individual
And then,
clams.
yeah.
We could get that kind of
No, I
topographic
mean,
relief.
I need a higher frequency sonar for that. This isn't individual clams level, but we can get see what we get. I can't promise anything.
Okay.
I'm concerned about promising things. I can't do it.
No, that's fair.
Yeah. This M3, they can go a lot higher res, and we just have like the basic one,
Gotcha.
500.
Yeah. I thought J-Rod was printing something last night.
Yeah, he's been doing, he did the one for Jill, like Kaylee's on the list, the park is on the list. And he's been sort of just churning through the new site.
Yeah. Yeah, they take a long time to run.
He said he'd make the file available, so if we all don't get one, we can take it home.
Oh, that's really cool.
Nice.
That's so
That
nice.
would be very awesome.
That's so sweet. But yeah, we'll work on it.
You can order these things on the internet too. I'm a big fan of I'm not a real 3D printer guru, and I find that 8 times out of 10 it's worth it.
But there must be some, at least like at URI, there's a couple places, I don't know if they have the right printers, but we have a couple of facilities that have them where I think we can just bring a file and be like, we want this, and then they print it.
We do as well.
Yeah.
It's just so great when you have someone who knows all the care and feeding of a given printer, and the thing comes out.
Must have that too, probably.
We do. Most of the online services are cheaper.
Oh, interesting.
I mean, Harvard's kind of notorious for its job shops being really expensive, and they do good work. But if you're just getting something
Close
3D
themselves
printed, it's
out.
not like you need a skilled machinist, you know.
Like, literally, they can't keep the doors open because they're so expensive.
They send out. It's pretty hilarious. They send out emails periodically,
It's sad.
That little bee.
saying that the job shop is out of work. Please send requests. But they do quality stuff. And the head of the main engineering shop just retired, so that we'll see if his replacement changes anything.
Also, public libraries, like around Cambridge, have 3D.
Yeah.
Actually, the whole
Yep.
Minuteman network, they have 3D printers.
That's the new thing in public libraries is little maker spaces. I think it's pretty cool.
I
It's really
love
cute.
public libraries.
They're amazing.
I love working with them.
Underutilized resource.
It's a nice mat.
Big fan of libraries.
The Nyack one's really nice.
Oh yeah,
Really, really lovely. So,
nice view of the water. Solid.
yeah, it's really comfortable,
Ooh, who is that?
accessible, like approachable kind of space.
Who is that? Look at this little bug.
What's bug?
The one in center field. I can't zoom
Oh.
anymore.
Wait, the spongy thing?
The thing with antennae.
No, the scale worm?
I think it's a polychaete.
This thing?
Maybe like a weird kind of sea mouse or something. Any thoughts, Roxanne?
Weird animal.
White. Looks like a scale worm to me.
Yeah.
I think it's a scale worm. Yeah, it's a scale worm.
Scale worm.
I know albino scale worm.
Scale worm.
Got it?
Got it.
It
Thank
is
you.
observed.
It's a nice patch of sponge.
Nice sponge.
Yeah.
So far, the only comment we've had on our watch
Bob
has
Esponja.
asked where, ¿Dónde está Bob Esponja?
Oh,
And we found him twice.
Esponja de Bob.
And now I have the Spanish version of the SpongeBob theme
What
song.
is that little thingy on the? Oh, that's a fish.
Yeah.
I was like, it looks like a little ghost, little Pac-Man ghost. It's a fluffy patch.
Yeah, are those? I can't tell from this distance exactly what those are.
I can't either.
I don't think they're worms.
It doesn't.
Distance
Wait, is
issue.
that the? No. Is the fish grazing on it? Can we zoom back in? Sorry.
No. I got nothing better to do. The fish doesn't appear to be
Just
grazing
mowing
on
the
it.
lawn.
Just
It's just hanging
Oh,
out.
he disappeared. Oh, there he is.
Jacob, did you see more than one of those snails?
yeah, there's at least 3. They're not all. They're alive and crawling around. They're varyingly accessible. How many does she want?
I don't know.
Oh, okay.
I think we have. It's three sounds good.
Yeah, it might take us a little while to collect all 3, but they're around at this site. I'm sure there are more that I've missed. Do you know who they are?
I don't think I did.
I've seen them before. Like I've seen their shells in the collection.
Yeah, so right. I don't remember what they're called.
Where's our textbook?
Maybe they're in there.
Oh, that textbook.
Oh, the real thing.
Real
The
textbook.
literal textbook is over in the main lab.
No, yeah, I don't think they're in that. They're
Really?
No, in the main type of thing. They're
Yeah,
not in this.
Yeah, in the digest.
Yeah.
Oh, there it is.
What is?
Yeah. Phimorhynchus.
Oh yeah, fine.
Yeah.
Yeah. Okay.
The guide has been updated. Snail. the one fit?
It's not. I'm just making a bad joke.
I was looking at Ian's lipids. It's very impressive that Nicole can tell those
Tell them
species
apart. Yeah, the microscope helps.
I guess that's true.
Still,
But still.
it's Some of the ones like the Pelta, Notopelta, they're
Yeah.
kind of cute. They got the little yellow, and when they're alive, they have like little yellow bumps in their back.
That's
Notopelta means back fur.
Like Notosaurus.
I guess so. What do they have?
Well, they're in the Ankylosaur family, but they did not have club tails.
Huh.
I'm loving
Remember when I was talking the other day about whale falls, but there was a dinosaur
Yeah.
that was found in Canada, and it was a dinosaur fall situation
Yeah.
in the ocean, and that was a Notosaurus.
Oh.
They were. It was
So,
pretty dope.
how far was this dinosaur from the land?
It was
At the
like,
contemporary.
at the time.
Yeah.
It was thought to be like 300-meter deep water.
Huh, that's continental shelf, but like.
Yeah.
Yeah.
Like
that
it was
fair.
washed out to sea.
Right. Right.
All right. Super complete.
Super done.
Super complete.
Amazing.
All
There's
right.
snails there.
Please stow the super equipment. We'll have to pull that out later, but we're gonna sample snails first. Here, I'll come. Yeah, I'll come center for you. So, all right.
Here's the snail that never fails. It makes me wanna wag my tail when I wanna feel snail.
So the super, and we're gonna switch to the suction.
I appreciate that you
I
started
suppose
that.
so. Yeah. Well, I mean,
Blue's
if you
Clues
can
was my
suction
jam.
with the super nozzle still like
Of course, absolutely.
tucked, in the
Yeah.
go for it. But
Yeah, I mean, I'll
Yeah.
use this.
Yeah.
we're gonna use the super again, but first we're gonna suction.
Okay. You know what?
Yeah.
Every time
Great,
I find
thanks.
that I get mail,
Yeah.
you gotta sing it.
Just like the Loop and Pull song.
We have a fantastic mail lady. Her name is Leanne. She's awesome. She used to bring our dog cookies,
Yeah.
and she with all the dogs in the neighborhood, when they saw her, they just were so happy to see her. They come right up to her and beg for cookies.
I think the postal service supplies their carriers with dog cookies.
That's wonderful.
Pretty sure.
It's good. That keeps dogs from biting postal carriers. But
Make friends.
yeah, there was one time where I was outside with my dog and working in our garden, and she my dog was always never had to worry about her off leash or anything.
Yeah.
And then so I was watering those plants around the outside of the house, and then come back and I was wait, where's Freya? And then I look down the street 100 yards away. It's oh, there's Leanne. There's Freya. Freya ran straight up to her and just sat down.
I love that. Freya's a great dog name too.
She was a great dog. She was my girl.
Oh.
She
Sweetheart.
was
Yeah.
baby.
Yeah. What a sweet face.
She knew it. She was such a diva. Loved attention.
I know, I'm precious.
Got upset when she was not the center of attention.
That's always. I love when big dogs are like that.
Yeah.
Like,
No, you pay attention to me.
yeah. How dare you? Just moose their way into things.
Yeah.
But
Yes, good job.
Always have to be part of the action.
2 emails back to back. Also, why did he send an email on whatever? Like he sent me 2 emails back to back, one at 1:50 and one at 1:51. The subject line is, for the first one, what is the name of the person that Kinde mentioned at NASA? And then at 1:51, he's like, what is the name of the person that Lindsay mentioned at NASA?
Nice.
Yeah.
Just 1 a.m. thoughts, it's fine.
It's
2 a.m. basically.
Oh, Pete. Just like, I like default, like never email Pete because I know it'll just get buried.
Yeah.
Like I will text him if I email him to be like, hey, I just emailed you.
Yeah, that's a good idea. Solid technique.
Yeah. Because otherwise he won't see it.
Yeah. Just gets drowned.
I can see it there is camera. I just need to bring it to the
Are both of these T4s equipped with wrist cameras?
just the server.
Just the run.
Or you could just put those on. Yeah, I'm pretty sure it's just.
Should I reply to both of them? The first one, I deliberately misspell the name, and then the second one, I actually spell it right.
Yeah, we do it.
I think he'll know this. I think he'll know.
Yeah, I think you'll get it.
Cool. So we're going into bucket 2. That's correct. Looks like there's some sediment in there. Is that what that is?
Did we slurp the snail?
We have not yet slurped the snail.
We have just grabbed it.
Gotta be here for
Yes.
I mean, I guess we don't care that much about the sediment. But it's kind of sparkly. And then I think this first snail we're going to target, pretty sure is over on the left, but I don't want to,
It's on top.
oh, it's up above. Okay. Yeah, I guess it is. Somewhere pointed.
Is that a wee baby squat lobster?
I love the wee baby swat lobster.
It's a wee one.
Just the weeest.
Babe.
Yeah, we're probably going to go after a few more
Yeah.
snails in the mussel bed after we get this first one.
Those. Woo.
You still know where it is?
Searching for snails. We gotta zoom in.
Over, that's a crab. Or a
Enhance.
squat. Oh, yep. It's crawling down the side of the rock. I think it is right here. That's a good spot.
Dive bomb that snail.
So really, would you like me to zoom?
Little bitty squat lobster.
Okay.
They're so
Kind of a-
tiny.
They're so little.
Mhm.
Those things.
I want to call this marker nursery one.
Nursery swat nurse.
Squat nurse, crab nurse. Clearly, there will be clam nurse.
There's baby, yeah, baby fish, so.
Oysters. It's a little tide. It's like an entry tidal pool. Those are like, younger mussels. On that little rock.
Okay, call out if you want to zoom
Zoom.
to you. Well, what?
You're so patient.
Now you're
We're at Tina 4. We've already been zoomed in.
Zoomed so fast.
Alright, zooming. There's this guy sailed with Ola. Is that too tight?
That's a beautiful shell. That is some craftsmanship. Some nice biomineralization.
If you like snails, you should try the Western Pacific. Tons of snails.
Tons of snails. Are those like fire volcano snails out there?
No, that's an Indian Ocean.
Oh, okay.
We have the punk rock snails.
Oh, that sounds pretty dope.
There they are.
I have snails at home in my aquarium. Yeah, the mystery snails.
Don't they have like?
And then I have one assassin snail, because the mystery snails had babies.
Yeah. Naturally.
As one does.
Yes. Here we go.
Is there something compositionally cool about their sclerites? The albanella or the albanacocca? Yeah.
The periostracum.
Yeah.
I think that it's just periostracum. I don't know if anybody's
I remember seeing something about like metals, but I might misremember.
What is it? The volcano snails?
Yeah, the volcano snails have the iron sulfide plate.
Oh, okay,
Yeah.
that's what I'm thinking of. That's what I'm thinking of.
It's holding on for dear life.
What? There you are.
There you go.
Beautiful animal.
Okay, and let's try to find some others.
Yeah, it's
Yeah.
reflect this one.
Yep. Watch the bucket. There it is. Okay.
You flew through?
No. I think we can stack them, unless you think otherwise, Roxanne.
No, they go in the same one. Yeah.
Stacks on stacks.
Okay. I am going to try to locate another. I definitely saw a few others in this neighborhood.
There was one near the spawning clam.
Which is over on the left. Pretty sure. It's next to there's so many skate eggs in here. They're just all kind of dark, blend in. Did you spot one? On the far side there? Maybe. I don't think so, but maybe. I think it's a clam hiding behind that rock. Yeah. Yep.
That one?
There's one right there, though. It just moved. Right above center now, straight above the squat.
That's a good spot.
Good eye.
It's so pretty.
You think you can get down in there?
Is that a different species? Or
Doesn't-
the shell looks different?
We're just looking at these.
It doesn't look different to me, but I am often wrong. Yeah. Okay, great.
We did get a team?
Yeah, we got a Kiyohimea on the way down. I have no idea what kind of condition it's in, but the DNA will be interesting.
Which color are those?
They are almost perfectly transparent. They're an interesting species that's appears to be highly dimorphic. They're actually so much so that they're described as two different species, and yet the DNA overlaps. And if you looked at photos of them, you'd find it hard to believe that they, are in some way admixed. Would you like me to zoom on it? That's okay.
Probably good we didn't get those ctenophores that we lost early in 571 because that was a very long dive. It would have been mush. We would have had no ctenophore.
For
left.
population genetics purposes, it's fine.
Mush is fine.
Pretty much.
Cool.
DNA in the
Fish and
mush.
wildlife? Not so much.
Most of at the very least, these ctenophores are going through to Shannon,
Okay, yeah.
who is sequencing them.
Nice.
So will you get to describe them as new species?
I mean, if one of us wanted to put in the time and effort and had good enough images and sketches, maybe.
You. If you want to.
By one of us, I'm referring collectively to like the Midwater Lab in Abari, or whomever everyone on this cruise was comfortable with describing them. But it's a lot of work, basically. Where?
What
So I sort of lost track here.
does it entail?
But it
It's in the middle of that crack of the rock. the pillow basalt
Okay.
underneath the skate egg.
Coming up.
Nope, not too far up.
Oh, underneath the skate egg.
No, not a skate egg. The dark shell. Sorry, I thought that was. Wait a minute. No,
No.
I goofed.
Oh,
Mine eyes have deceived me.
oh,
Oh, it's up. It's up. I know it. I recognize this because it looks like a it looks like a cigarette butt. It's a deep sea cigarette butt.
You see one on the right?
I see one of the muscles.
Yeah. Oh,
There
it
is one on the
There's one right
muscles.
there. Yep, that's a good target. Can you reach that?
And I think there's another one further up.
Can't tell what.
Yeah, there's the muscle one.
There's further up.
Yeah, I'm pretty sure you pulled right up on it.
If you
and I
pan
should've
straight
paid closer
up,
attention.
there should be another one.
I think there should be one down though, and that's the one that we pulled up to look at, or to get. It was in a little fissure. We may be on top of it.
It is a
now.
crushed snail. Pick no go
Yeah.
in.
So many
You can use one for boss.
Right. It might have finished the job. It might be hiding now.
It's not further up?
The other one is right there. The muscles. Do you see it? It's right at center field now. Zoom out. Yes. Yeah, there we go.
Well,
Can't reach that. Okay, so we have to jump over another rock, or we can jump back, or there's another one over here. Bring it to center.
That's
I think that's the
one.
one we were after.
That is the snail we are looking for.
It's all still pretty far away. It's alive.
Sea spider.
What is that yellow thing?
That is a funky looking yellow thing.
Oh, a little yellow thing.
Yellow thing.
Yes. You should slurp it.
Alright.
Slurp that up on the floor. Slurp that.
If we can get these both from the same position, that would be extra cool.
Oh,
Hello.
goodbye.
I'll actually keep track this time.
Oh, city sit. I think I see another one.
Yeah, I think there's there's another one in these muscles, so we'll be fine.
Yeah, I see 3 now.
Roxanne, would you be okay doing the outer sites? Doing the outer super site on the northern transect first?
Yes.
So that we can do the same thing and the muscle margin is in front of us on the way back.
Yes.
Okay.
Good alliteration.
Yeah, I think the skate eggs hatched. The upper one definitely.
Yeah, they look
Yeah.
pretty hatched to me.
Where are the baby skates?
Let's hatch now.
Inquiring minds need to know. Bloop. That's a different species.
That one doesn't to me, but it's different angle. We'll find out.
Nicole will tell us. Bloop.
There you go.
Here you go.
Careful.
Uh-huh.
Alright, and we're gonna go for another one that's just on the left.
No shame. No shame. To bring
Boom.
fun to watch.
Alright.
You gotta. Is this going?
Yeah, it's all
Right
gone.
there.
All the snails go in suction 2.
Okay, just
Might be a bit of a stretch.
Super slurp.
snail. Slurp, slurp.
It's gonna break the bungee cord.
Keep it snail.
Oh, that crab! Look, listen. You got a shell inside a shell inside a shell,
Oh, and a crab.
and a crab inside a shell.
Wow.
Shells on shells. On shells. So yo dog, I heard you like some shells, so I put some shells in your shells. Shell while you shell. Shell page.
I love it. I love it.
You know, back when memes were made with, you know, the impact font. Not Twitter screenshots.
No.
Excuse me. X, formerly known as Twitter.
Yes, I know, right? The artist formerly known as Twitter, yes.
What is the verb for X now? Like, so you know,
I don't
to
know.
tweet.
It's like his children. I don't remember how to say their names.
Kyle.
Is one of those named Kyle?
Well,
Hydra.
it's like K-A-13.
Frame grab, snail collection, also spaghetti worms.
Okay. got it.
Also shrimp.
You see the spaghetti worm living on the with its tube attached to the egg.
Oh, yeah.
Shrimp, spaghetti worms,
Shrimp.
snail collection. I've already got one snail collection.
Oop, 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 oop.
Ludicrous speed. Whoa!
Slurp.
Bonus worm.
Well, we're getting a lot of volunteers here.
Does not want to go past. There it goes.
Can we go for one more and then we'll split? There's one there and there's one there. Take your pick. There's a couple more right up here. Upper field. There's one there and one there. Take your pick or steady up in the middle and go for both of them.
Shrimp. Spaghettis. Is the singular of spaghetti worm spaghettis worm? I think that's what the
I
Latin.
think spaghettis is singular.
Spaghetti. That's what I'm saying.
Oh, I'm sorry.
Spaghettis.
Plural.
Singular. Oos, oos, ooey, ooh. That's first clenching.
Got it.
There's another snail.
Oh my god, so many snails.
Above, I believe there's one above that other skate egg. Yep.
Yeah, there's two right here. Do you want to go for 5? Or do you want to get on the move? Okay. I think, we'll just see. I think they'll both be in range.
There's 2 of them.
Yep.
Dose. Yeah, they're cute.
Interesting what's happened to some of these clams.
Oh yeah, that is a very little baby one. There's one that's inside. What is that? Is that a fish?
It's not a zoar.
I want it to be a babby octopus. Lots of limpets on these here.
Okay.
Muscles. See, that's it. See, that's the same as the first one.
Yeah. Got it.
But there's another one. There's
Oh my
3.
God,
There's
little
3. There's another one.
Oh my god.
There's like 4 snail. There's another one.
Snail City.
There's like 5 snails right here.
We're living a great
We'll
time.
settle for 2. That's great. There's a 2-fer. There's actually, you could probably suck 2 at once right there. See them? They're right on top of each other. Just under the electrical tape. Yeah.
I'm not gonna say anything.
What did you see Tina for?
What? No.
That's really suspicious.
I like that that's how you're responding.
No.
Or am I just missing all the snails?
We'll tell you at
Silly.
breakfast.
Is there another clam spawning?
No, well, close.
I can interpolate. So, either Roxanne or Rachel, you might know, but are there, on these seeps, are there areas that are subcompensating for calcite? Like, do the shells just dissolve spontaneously after the animals die? I'm seeing these holes in shells and wondering what they're about.
Yeah, I don't know.
I kind of-
Specific water.
That's what I mean, yeah.
That's what I thought.
Okay,
I was
yeah,
thinking
because it
that
really just
too.
looks like chemical.
Yeah, I wonder if there gets to be a point where like the diffuse flow is too powerful and they can't keep up, like making their shells.
Mm-hmm.
Or it's like there's like this happy medium.
Right.
Oh no.
Oh, there's 3 of them.
No. Oh.
You can't. They're all attached to the same muscle.
All right. Slurp the muscle.
No, we do. Okay.
Get that out.
She may not go to.
Oh.
It's, yeah, you're afraid it'll clog the system. Yeah. So you can try to like shake them off, or we can give up. It's kind of up to.
There's another baby. Look at that. Oh, yeah. No, there's another one down here.
Okay, we're going for another one. I missed you guys.
Well, it was an even smaller one, also on a.
Oh, there we go.
Get in there.
Get in
Tumbling.
there.
Get in there.
Get in there.
Okay,
Let's go. Oh, books. Shells on shells. Shells for days. Yay!
Okay.
There's another one. And another one. And there's another one down here somewhere.
Gastropods galore.
There. No. There.
Babiest.
That
You
is
think
the
it's
babiest.
in the there it goes. Just fell in. Okay.
Ever watched the YouTube video Wizard People? Dear
All right.
Reader?
Are we done?
It is so funny.
I think we can be done. And let's be done. with
It's
snails.
basically all of
Um,
Harry Potter and the Sorcerer's
At this
Stone,
point, we can
but parodied.
I think
we can stow the suction and redeploy the temp probe.
The baby is the most powerful
Yes, please,
baby
thanks.
in the world.
Got 5.
Was it 5 or 4?
4 or 5.
I believe it
I
was
believe.
4. We can.
Yeah, it might be 4 in the end. Yeah.
Or the one in between. 4.
Yeah. What did the name of that end up being? I forgot.
It was.
The,
Snail?
got some wise.
Hierarchus.
Something rinkets.
Thyroid.
Spelled weird.
Thyroid.
Biorinkets. Anyways.
Thyroid?
P.
5.
Where did I put it? I have it right here.
That's right.
It's PHY.
Yeah.
OR.
Biorinkes.
HYN. Chess.
Alright.
Snail. 4.
Super time. Super time. Well, we gotta fly. First we fly.
Awesome.
Okay.
Goodbye.
Adios.
Find more Rincus.
Find more. Not
Phimo
find
rinkets.
that. Find less.
MOR. PHYMOR HYNCHUS. Where is my biosync leaving? Rock. Fluid.
Good morning to those who have recently joined us online. You're here with either the ultimate or penultimate dawn watch. We're back at the Iguanas vent field, our first sampling site on this cruise. We've recovered the distributed temperature sensor that we deployed here weeks ago, and now we've gone on to some other science objectives. We just collected a bunch of snails, and we're going to do a couple of more supers along a transect leading back towards a mussel hotspot and a chimlet. Stay tuned. Yeah, but,
20 minutes super sample.
22.
22 minutes super sample. Drilling. Conclusion.
no, we're going to do it elsewhere. I just figured if you want to arrange the stuff while we travel, we can do that. we are going to hit, so
We are going to,
you want to be 30 meters north of Chinlet 1.
past supermarker 2.
Oh, actually, yeah, is it 30 meters past supermarker 2 or 30 meters north of the...
Yeah, super 2.
Okay, so yeah, so we have a marker super 2. You see it up there, Alex? And you're trying to do two things at once. Yeah. Do you see a marker super 2? Should be north of the Chinlet. Yeah. You see it?
Yes.
Okay. Yeah. And well, wait. And, we want to head to a point 30 meters due north of super 2. 30 meters. Thanks.
Just the man for the job.
My passive aggressive question for the green sea developers is, can you make the markers any color that isn't green?
Are you
Yeah.
red-green colorblind?
No, it's just the same color as everything else.
Well, that's
And there's so many
That's
markers,
just
yeah.
the bathymetry here.
Excuse me.
You've got a problem with the markers. You got a problem with Haley because
Well, I also have thoughts on green sea, but they're probably not the same.
change the
We are ready to head to that point.
Well, there's just I don't like importing things in lab long. I like importing things in X, Y. That's how we know it.
that is how you roll.
Is the superpower still on?
It still is on.
Okay.
She is chugging away.
No, she is not chugging away.
Not literally. Not yet.
Not yet.
pumping.
Not this day.
Not until we get to site. Yes.
It is not this day.
Just out of curiosity, why do we secure the super between samplings? Does the serial trip up if we leave it on, or?
For the power, you mean?
Yeah.
The IGTs can't be used when the super's on.
Oh, interesting.
IGT free zone.
IGT zone.
Did you see that Science Craft made it into the week 2 video?
Oh my god.
Yeah.
No, I did not.
It was pretty awesome.
Yeah.
He was directing this, like the IGT
Yeah.
right in there.
Nefarious crap.
I feel like when we went back the other day, when was that? Oh, this was yesterday when this guy started. I probably
Where
saw
were we?
that South Science Craft again.
I came out like,
Where? Number 2.
We should tag Science Craft
He's a real guy.
and write proposals to come back and train and learn, basically train Science Craft to be our remote sampling craft.
Absolutely.
I mean, like the military trains dolphins, and we can train
Crap.
crabs. We got it.
Absolutely.
We go back to those crab people. Tell them
Teach them. Science.
science.
oh, right, well, I did that earlier. It's harder on the super side, it's easier on the front row. It's pretty close.
have any kind of read on when Dan may show up? Usually like
Yeah, usually around 6.
6:30, yeah. And then the question just becomes whether, I mean, I think it makes sense to finish the super objectives before we transition to that.
Yeah, I'm not sure actually how many objectives he has on this, because I think, I don't know if he's found a spot that he's interested in. I don't know.
I remember him saying something about Albanelids.
Yeah, he wants
Yeah, I
to.
know, but I don't know if we have a spot here. I asked him to find the location, and I haven't heard
Okay.
anything on going back through other dives.
Hmm.
I think that it might be at the other side of the vent field that he might be thinking of. We're not at right now, so I don't know if he was
I saw some Albanelids last night, but it wasn't spectacular.
I think he's wanting to see some turf wars.
Whoa.
I see.
Like sock and boppers out Alpenells.
Because then the question becomes if he does come down here, and wants to is interested in retrieving the lander and stuff, whether we do our clam collection.
The lander will not be part of any of that, because the batteries are all out.
The camera's out. The batteries are dead too?
He said that he doesn't want to do anything with the.
Oh.
Yeah, I think he thinks
Great.
that so the lander's only for recovery of the IUT.
Understood. Okay. Thanks. Just saved us a couple 100 meters transit at the wrong time. Alright, looks pretty barren. If you want to come around 180 degrees to your left, then we'll be pointed in the right direction before you set down. So just come around, just point 180.
I can still work on this thing.
Is the idea to try to correlate these data with Johann's DTS data?
No, it was more just like I'm interested in having a lot of spatial points around the semi-contained mussel bed, and we have like a bunch in the mussel bed, and we have the background, and so I'm just trying to get different.
I see.
We see differences in the abundance and structure of the
Yeah.
science. I mean, we might be able to use this data too. I don't know, but a lot of this is just background temperature.
Mm-hmm.
Is the ICL looped around the super hose?
Yeah.
Alright, so you can set down. Pardon? It shouldn't, no.
120.
Something I noticed. Shouldn't affect anything else. We pull on it.
Did we ever clarify the Latin long being the vehicle coordinates or the USBL coordinates? Like which one is more accurate?
Well, let me ask you all the question then, because we were told a long time ago, we thought that when we were writing down the best coordinates used, it should be the USBL one from Ceylon, but now that they've changed, which is the best coordinates to use?
That's a question for
That is. So, the better one to use would be the vehicle real-time nav data. That would be a sprint data, that would be the data that is used by the IMS system, whereas the USBL is the raw USBL position. Okay. So that might jump like 7 to 10 meters.
Yeah, right. Okay.
So,
So
yeah.
that's right, we're in the wrong one.
Yeah.
Alright, okay.
But it should roughly still be within
Yeah.
decent range, but yeah.
Well, we'll go back here and just, we have both from Ceylon.
Yeah, yeah. Yeah.
Are we ready, folks? We super ready?
Super ready.
Super duper.
Alright.
We are going to purge now.
Which filter?
5.
5. Okay.
Wheels turning. Good.
There's more of that yellow stuff
Where?
on the left.
Oh, that I think that's that's crust that's under the crust that we just knocked off of that rock with the ram.
It's like iron oxidizing bacteria.
Yeah.
Let's zoom in.
Stand by. Timestamp. I'm just trying to do my job.
Hold on. Should have lit one.
Zoom.
Hold on.
May I zoom?
Yes.
Ooh, wait. Does that recall? What's the thing?
Enhance.
Oh, what was you? I know, but we're interested in it.
Ooh.
Wow.
That's well, the stuff on the right is definitely iron oxidizing bacteria. I have no idea what.
It's glassy too, right? The crust of the rock is glassy. Yeah.
It's weird. That's a
Maybe
weird
make
rock.
some notes.
Hold on.
Weird rock.
Should we collect said rock?
Huh. There's an idea. It must be pretty. The crust must be pretty fragile though.
Yeah, 'cause you touched it and
We
it
just
like,
bumped it with the rubber.
Yeah.
Well, actually, that was probably the metal plate scraping.
Mm-hmm.
That's really weird though that it's like on the like rusted out from the inside.
Right.
It's a really strange rock.
Actually, it's iron oxidizing bacteria. They kind of prefer it to be like slightly hypoxic.
Mm-hmm.
So that kind of makes sense.
Mm-hmm. But kind of strange there would be no surface presentation at all.
Yeah,
But yeah.
that's a
I'm gonna message rock people.
rock.
Well, but first I will pump actual sample. Okay, 1400 now.
It's the head's okay. Well, thanks for breaking that rock then.
Hey Jacob, can you zoom back in so I can take a picture? For rock people.
Touchy.
Hold on.
Oh my god.
Is that alright?
Yep.
The actual jaws on these manips, are they electric, hydraulic? Curious. Are there any manipulators that offer adjustable grip force stock? Or has anybody played with that? That's not what I mean. I mean what I'm envisioning is basically like a back pressure valve on the on what's up? What's up?
What is this sliding rail called?
The on the outside there? What you pointing through the bumper? That's a ram.
Ram? Thank you.
Yeah.
RAM.
Yep.
Sorry for interrupting.
No, like the truck or a mail sheet. Yep. Or you call it a slide arm. Yeah, the idea of basically just having a force limit. The feedback is helpful too. That's the thing, I think, right? But it probably starts really high. Sorry. I was just curious. Seems like it would be a thing that would be useful for science if it can go low enough. And you know, if I think of my simplistic approach of, say, engineering a relief valve into the hydraulic circuit, the problem might just be that the friction of the linkages is so high that you know you won't be able to achieve the minimum force you want.
We have like synthetic kitchen sponges.
Yeah, I've seen that. That works
And
nicely.
you just kind of like visually look for start to compress.
Yeah.
Okay.
And Steve's cruises, we always fly with kitchen sponges or soft grips of some kind. But I guess for here, for rocks, they don't work so well.
There was I think there were some students in Harvard that were actually working on like a pneumatically actuated kind of soft grip thing.
Is it the one? Is it the beanbag one?
I don't recall.
Hmm.
I think that's what I'm thinking of.
Pneumatics for deep sea application?
Well, that's the thing. I mean, you could still probably might be able to do it hydraulically to some degree.
We found SpongeBob again. I think we're just a closer range. Yeah.
To be fair, I don't think they were designing for deep sea.
Right.
More just
Yeah. There was an interesting paper I read years ago that I thought kind of had a lot of potential. It was hard to control. It was basically a water balloon full of beads, and you'd you know deform it around something, and then pull a vacuum, and the beads lock together. But I think it would have pretty niche applications.
Yeah.
It's got like little striations.
We're gonna be here for like 20 minutes.
It almost looks like a salmon fillet.
Yeah.
I know. I was thinking it looked like a fruit or something.
Dried mango.
There was the
That's
soft.
breakfast mango.
There was the soft minute thing that they put on. There have been some really funny, I think, sort of capture devices. There was the like dodecahedron or icosahedron. Remember that one?
That was the one that like.
It doesn't hold water though. It looks beautiful. Yeah.
But does it need to hold water? I mean, what's the idea to just like?
If you're just observing in situ, like you want to put a camera on the inside and capture an animal and carry it around. I thought was kind of what they discussed it for. It's not really for sample retrieval. Of course, in that case, you may as well just have a D with it. I think it doesn't, in my opinion, really improve on a detritus sampler, but it sure looks cool. Like what would the price?
Is a detritus sampler? I mean, does it need to be a cylinder, or could it just be like could it be square?
It could be square. The only issue is seal loading, right? But if you can put an O-ring in a square groove, then fine.
Good.
And the seal's not, well, it's spring loaded, right? And you're just relying on the cantilever of the lid.
Yeah.
Do we have a marker here?
We do not. We should drop one.
We should
Yes.
drop one, and we should include the orange rock. Maybe call it
What do you want to call the orange rock?
orange rock.
Orange rock. Okay. Orange rock / Super 4.
Super 5. Super 5.
Oh, okay. You want to do it by filter. That makes sense. Yes. Okay. / Super 5.
What do you want to call this marker?
Orange rock / Super 5.
Number 5 or the
Yeah,
third?
number 5. You could do a pounce on without pounce.
Oh,
If C-Log doesn't hate you for doing that.
Actually, it should be Super 5
Super duper.
right now.
Yeah.
Yeah, I was just thinking like with the detritus sampler, if you had like a flat wall, you'd
Yeah.
probably be a little more amenable to putting a camera.
Yeah.
Timestamp.
Sure.
Oh yeah, I guess I need that.
Though honestly, I don't. I mean,
Instead of
Oh yeah.
I don't see.
Jesus, Joseph and Mary. All right.
I think we can do at least 12 lines.
12, but wait, 57.
Because you can't photograph it in a container where you have no control over it.
57.
Yes. Alright, so now we got.
I was just thinking of that particular like
2.10.
buckyball type device. I was curious if you'd seen it too.
That's
That's really something.
I didn't realize it had cameras associated with it. I thought it was just something that like you could like grab something and then like
1.16.
I mean, it was kind of a hammer looking for a nail.
0.01. Altitude.
Yeah, in my opinion.
0.84. Llama.
Orange rock.
Orange rock. Strange. Is it less bright than it was before?
I don't know.
Maybe it's the lighting.
Ooh, we could do some mood lighting on it though.
On the orange rock?
On the orange rock.
Well, we just got to get this cable out of the way first, but absolutely.
Natural frame.
I mean, we don't have to do it for like the gram. We can just do it.
Unfortunately, the video light here is in front of, in the foreground of the red rock. Because we rammed it, so it's, yeah. Very natty mat down here.
Yeah, I was looking at that.
Mat.
That's some veggie-toa.
Mat. Observed.
You wanna go back through C-log and give me the actual coordinates for the last couple of
The last one we did.
samples?
Yeah.
Yeah, thank you.
Of course. Alright, so
It's a tiny squat.
tiny. Oh,
It's a really tiny squat.
very
It's like less than 2 centimeters, like the leg probably.
baby. Very. He doesn't look like the other spots.
No,
Too baby.
too baby, too curious.
Small baby.
He looks like he's smiling at us though. His little mouth parts.
I got the
Observed.
focus lag working against me.
Is
There
it-
we go. There's that looks like a tiny mussel. Or an egg.
Oh yeah, those are little baby mussels.
Baby muscles.
There's 2 of them here.
Babby mussels. And then what's that little baby stocky thing?
That's a worm tube. This here.
That's moving?
That there.
No.
Where?
Along the same little crack as the baby mussels on the left-hand side. It'll pop its little head out again. Maybe it's a baby worm. See?
Oh, I see it.
You see down, left, down,
Warmer.
left, warmer. The little string sticking out.
Yeah, that. Rate it.
Interesting.
It's
You know what it looks
moving.
like to me? Oh, it looks wormy. But what it reminds me of, which is probably not. Let me zoom in before I start saying things that aren't true. I can't zoom in. Well,
It's an Alaskan bullworm.
Weird. Strange.
it looks like a worm.
Mystery.
Yeah, some kind of worm,
Baby circulid.
maybe. It reminded me of a caprellid amphipod, which I don't think they have down here, but they don't extend like that. They just wave back and forth.
Okay, I guess I'm ready for it.
Look at the colors on the little squat.
I know, he's so pretty. It's like ghosty, kind of.
Mm-hmm.
Alright, so for, this one's for 4.
Okay.
So we have, let's do latitude. 2.104017.
4017.
Longitude is -91.926085.
Okay.
Depth is 1674.
Come again?
1674.
And altitude is the same, 0.83.
Along these little blebs, the brown things. Well, that one up there, almost directly 12, like maybe 11 o'clock from the squat lobster, looks like a juvenile mussel to me. Which I think is interesting because, and there's another one. I think a lot of these might be.
I agree with you about
The lower ones I'm not so sure.
Yeah, these other ones.
Though to me just the upper ones are notable because I always think of mussels as settling gregariously. Like I expect to find H classes in a mix. I don't know that much about dead mussels though.
I can't, like, I could try to go.
Like
No,
when I
that's-
at least
that's the
with
super team's
tide pool
problem, don't worry
middlets,
about that.
I tend to find juveniles in among big adults, right? Like they settle gregariously, they settle on each other. And I mean, I've never really looked, but I've never seen a mussel bed that's all like a young size class. It must happen at some point, right? They have to start somewhere.
At the last site, we saw little baby mussels.
Yeah.
Lots of babies.
But let me
Where we just
pan
were
down and try to
when they're
focus in the
small.
foreground and see what those things are. They actually most of them look kind of muscly. Like the one at the bottom
Are
does.
definitely mussels. Some of them, others look like, I don't know if they're like egg pouches or something.
Yeah.
Snail eggs or something. Even though, I don't know if snails lay singular eggs like that or.
Muscly and juvenile.
That's both. Not the words for me. Maybe the second one.
I don't know. Like maybe they're eggs. What do you think?
The little nubbin?
Well, so some of these nubbins are little mussels, and some of the nubbins we're not so sure. Do you think they're all little mussels? The aspect ratio looks a little bit off. Like they look fatter than
No, they
should
look
be.
like baby snails. Cause some of them they have like little like.
They have a little
dimples on them.
proboscis or something
Yeah.
crawling around. Yep.
They look like little snails.
Well, maximum effort on Zoom. Maybe you know some other tricks.
They look I feel like maybe my eyes are deceiving me, but it looks like little circular
Can we deploy the
spirally
microscope?
shells.
That's what I was trying to figure out. They're trying to tell that if they're on what was starting
We totally do it. We have this great thing. I forget what it's called, but I can pull it up on my computer and show it to you. We bought it for lab use. It's almost like an endoscope. It's like it's a fisheye lens that's tiny. It's about that long, and you hook it up to it like it has a regular F mount bayonet on it, and so you can like put it on a SLR and stick it. Like we have made a little submersible cap, so you can like stick it into a tank of water. You know, the photo you get makes it seem like you're a tiny amphipod next to the subject, right? You could I would love to see one of those compensated and put in a little dome. Probably break the tip off with a couple times, but you're saying
Ooh, there
They
might
have
be
these
those.
little handheld microscope things with a little LCD screen.
Yeah.
And like you put it on like really zoom in
Yeah.
on like insects and all that. It's great.
I mean, imagine just having one of those on, you know, that you could put with like a little light ring on it. And you take it out and just stick it in between these rocks.
It's like
It'd be a
only
total
up to
first. I
like
think it
3
would be a first.
millimeters across. So they're like little guys.
Because if you pan down, can you get close on a closer?
Let me just see if I can see any others, but
Are we sitting on the front porch?
we're
So I'm gonna try to
that's
slowly bring in the porch and then
okay.
that'll allow you to put it down.
You all right with that? He's gonna bring the porch in.
Oh yeah.
Okay.
It shouldn't move. If
Hopefully it won't
starts
raise
to move
anything.
Anything else, then I'll.
Okay. But yeah, we're zoomed all the way in, so all I can really do is tilt. And am I racked all the way out?
You're racked all the way out, but there's a front porch.
Yeah. Something interesting in that crack. All right. Little thing.
There's some straight in front of the camera.
Yeah. Coming.
As far down as it'll.
I think those are snails.
I'm at the bottom of the hills.
Oh.
Almost.
It'll drift back up to where.
Oh, interesting. not really. I see. I see what you're saying. There's like a soft stop in it. Is it mechanical? Anyways.
No mechanical stuff.
I just can't. Maybe, I mean,
They look round, they don't look
yeah. Annotate egg-like.
Snail question mark.
Snail-like.
Cause this one like up here in the upper left hand corner.
Unknown benthic
look-
object.
ooh,
Where?
it-
Upper left?
upper left, now it's moving towards the center. The darker one, that looks
That looks,
Oh, that looks motile. Yeah.
Motile.
Yeah. I mean, it looks like it's crawling around.
I think these
Sorry.
are
No, I like it.
Yeah. As opposed to sessile. But it's gratuitous jargon.
I feel like I see
Yeah.
like it looks spirally. Like
It-
all
that
of
does kind
all
of.
of them do. Just not it's not as pronounced in the
Yeah,
other ones.
yeah, they do. They kind of, now that I
They have like
squint,
they have like little dimples.
They kind
in
of look
the
like
middle.
periwinkles.
Yeah.
Yeah.
Who are you?
Do me a favor, right click on the photo to the left.
This one?
No, left.
Oh, this
Well,
one. Yeah.
if we find a vent,
Zoom in there.
Oh, okay. I see what you're saying.
Oh.
Yeah. We.
if we find a vent literine snail, I know some people who would be very excited.
It's about as far in as I can get you.
Okay. Okay. We'll look at other
Of course,
photos.
we did that on a bigger.
Can we just slurp some when we're done here?
Can we just
Go to
look
scrape
at-
some up and slurp them. What do you?
I guess
Zoom. Zoom.
they're
Enhance.
gastropods, I think. Ian
Okay.
would want them,
Let's
right?
try this again.
If they're gastropods, almost
Let's take
certain
this
somebody
guy
will
again.
want them. We would have a little bit of a hard time getting them on our export permit at this point, but Nicole might want
Yeah, I
them,
see,
Stuart
like-
might want them.
Yeah.
Yeah,
I
that's
see
a
like a
good point.
spiral.
I see
Can at least
spirals.
sequence them.
We have
So I made some mistakes here. We have 1 and 2 are used. We should be on 3, which is correct. It's empty. No need to index.
Like the little dots
Yeah, I
in the middle.
Yep.
I
Yep.
screwed up.
Yep.
We've
I'll
decided
mark them
these
off
are
definitively once.
probably
So we still have all next positions open.
All Niskins are not fired.
Oh no, Nicole fired.
Oh, she did? Well,
Yeah,
then I
think
just
so.
didn't get. It's not up to date. Cool. Thank you.
I think she did all of them or most of them, I'm not sure.
Yeah, all and except 6 is noted as maybe.
That's okay. Maybe
Oh,
failure.
I can see it. Yeah, it looks like half flag.
Yeah, unsure if fired. Fired at 3:35:32.
So 5 not successful.
At muscles 2 at
I forgot about the
Different
permit,
rocker.
Jacob, I don't think we should, I think we should just leave it, cause otherwise I'm not sure that we can add stuff.
Well, we can put. My understanding, which could be wrong, is that we could always like put them in ethanol and give them to Stuart for the
Yes, that's
collection,
true.
and then they don't leave the country.
Yes, that is true.
If it's not something that's on your permit.
Well,
Yes.
well,
Yeah.
yeah. The issue is that we would have been able to sample these in principle in the past, but we've enumerated all of our samples, sent them to the Ecuadorian government. It's finalized, like they've reviewed it, they've okayed it. So revising that would be difficult.
We can change the numbers, but I'm not sure if we can change the, like, species that we put down.
Zoom in. Enhanced as enhanced as I can get. Zoom.
So Pete mentioned offhand last night that you're thinking of going to school for deep sea biology.
Me?
Yeah. That's awesome.
I think it could help me do my job better.
Take his class.
I think it'll.
I think sometimes Pete teaches deep sea bio through the extension school.
Yeah, he's teaching it this spring.
I can
Yeah,
take-
extension.
He said I can sign up for the
Yeah.
Yeah.
Cool.
That'd be pretty cool.
That'd be awesome.
I might be your TA.
really nice.
Will you be nice?
I've been nice to you this whole time.
So have I.
Do you know what that tentacly thing is?
Okay.
Yeah.
Okay, we're good. Solid.
Don't complain about grades. I'm a harsh
I was
grader.
told-
I'm a harsh grader.
I was told I start off with an A and then it goes backwards.
That's what, yeah.
Pretty much.
Well, yeah.
It's all downhill from there.
When I was waiting tables, one of the best lessons I ever learned was this old gentleman who sat down at the table and put $5, 5 singles on the table. I was like, what is that? He's like, that's your tip. If you mess up, I take one away. And I was like, I like this game.
Does he add to? Does he
He
add
actually
dollars?
did.
He does
actually
add,
did. He
okay.
ultimately
Okay.
actually did because I found it as such an entertaining game.
Yeah, who is that?
What is this strange thing?
The both things. Is the thing on the right just a long filamentous bacterium?
On the right?
The hairy thing.
Oh, that.
This.
This.
There's
Oh.
this and there's that.
I'm very focused on that. not
I
this.
feel like the thing that's moving right now, it looks like one of those circular worms that's trying to make its little mud case.
You. That's a polychaete. It's the weirdest polychaete I've ever seen.
Which one?
The white thing. That.
Pom-pom one.
Like I don't see the segments. It looks like it has a little papillae.
Yeah.
I
It doesn't look.
don't think it's a polychaete.
Doesn't look segment.
Strange one.
You might have some good images on this one.
Yeah,
All the little.
I hope so.
It's pretty wide field of view though.
Yeah, but I mean, it's focused.
Can we get some observations on this?
Yeah, I'm
On
doing it.
benthic object number 2.
I'm doing it. I mean, I don't know what to observe.
Alright,
but we're
super
doing it.
status is complete.
Super status
Okay,
is
we're gonna
complete.
switch. Can we switch to slurp some things if it's valuable to you to know what the brown
It's
things
not
are?
for me.
Oh, okay.
Yeah, so
Well, what do you say?
Yeah, it might be hard to get them to anyone who actually really wants them now.
Except sewer.
Alright, let's just do it.
Okay. It's more of a time question. All right, let's stow the temp probe, please.
Okay.
We'll keep the super nozzle deployed, but we can tuck it.
Okay, so I can just go off to the side.
Yeah.
In the porch a little bit more.
And why not? Why don't we just target that weird worm thing we were just looking at? Unless it's too close in, we'll target it with a slurp, and we'll get some of those brown things in the process.
How did your class like the ship?
I don't know, but I'm sure they like it. I haven't heard anything from them, but seems like if I were a student, I would like it. So hopefully they would like it, but they liked it. So that was pretty awesome.
Are you done with super? I know
We have one more
super.
super sample.
Sorry, I already forgot. Brain processed as soon as the words came out. I was like, never mind. Stop. Be silent.
Wasn't aware of that.
Seen is not heard.
Not even seen sometimes. Just do not perceive me.
You grew up in the South too, I'm sure you like, did your parents also like say when you had like guests over, it's like well-behaved children, like are
Seen.
better seen and not heard?
Yeah, we were better not seen or heard. Just go play outside. Yeah. Don't play with your sister. Okay.
We could actually, while we're slurping, if you're interested, the easiest way to get the orange rock might just be to slurp it in another slurp.
Okay, we
Just
can
the
try
index
that.
one and suck up some of the crumbs.
We can try that. Have we already collected talus? I'm not sure. Because I'm kind of confused at the annotation on my sheet. There are RO3 and RO4.
RO4 is talus.
Yeah.
RO5 is talus.
She mentioned it.
RO3
says it's in.
All right, so the there it is. Wow, bingo.
talus. So much talus.
All of the talus.
All of the talus.
All the above.
All of the above.
If you can somehow get that little piece of talus up the nozzle, we'll be very happy. But
I don't think it's talus.
or you don't think it's a broken off piece of it's basalt?
Wait,
It's
oh,
basalt.
I thought you were talking about the orange thing.
No.
Yeah.
Yeah. Okay, there it goes. Yeah. So if the worm comes off, it's great. If the rock comes up, that's also great.
That is not the rock we are looking
It is the
for. It is some
worm.
That's the worm we're looking for.
Weird worm.
weird worm.
Definitely not a worm, but
Weird creature.
oh, we're
Well,
still.
it does
Okay.
look like a sour worm.
If we can't get it, then we will give up because it's not worth like
Sorry, dude.
grabbing. But
We could try to slurp the little baby snail things.
yeah, we will try to slurp those for sure.
Oh.
You're gonna scrape it off.
It definitely looks like there's some ironite oxidizing bacteria. Like before, yeah, they're just munching on that basalt.
What section? Are we on a different?
We're on bucket 3, which is not the snail bucket.
Right.
Not the snail bucket.
Thanks for checking. It's almost Nidarian. No, it's not Nidarian. It's okay.
Definitely one of those cards.
Yeah. Yep.
Without them.
Yep.
Except
Credit bad, credit no problem.
Get it.
Oh, that's even better position though.
Yeah. No.
Oh,
No.
No. Ah, river.
Oh. Get retracted. Anyways.
Oh.
Oh,
is there another one?
flick of the wrist.
There's two of them.
No, there isn't.
Excuse us, dude. Get in there.
Whoa.
Oh.
Welcome to anyone who's just joined us. You're here with the Dawn Watch on Iguanas Vent Field, Western Galapagos Spreading Center. We're attempting to suction sample an unknown benthic organism. If anybody has ideas as to what it might be, we welcome them. Otherwise, we'll hit the books later and find out. Okay, you're in good scraping position.
Oh, you almost got it. Almost.
You're not on the touchdown on the rock. It's almost about to break off. Wow.
He's really trying his hardest.
Sometimes the game's gotta respect game, you know?
Oh, there it
I think
goes.
you got it.
Okay, watching the bucket.
No. Well, there were two of them.
There were?
Yes.
Whirling around.
Yep, there's one. It just came in the bucket.
There it goes. Okay, now.
Nice.
Wow. Okay. Will that go down the tube? This is like when you suck up a quarter with the vacuum cleaner.
Sometimes it will like rotate the nozzle towards the segment.
It's still in the tube.
Just went down to.
Alright.
Peek in there.
Down the barrel.
Shake it. Like just much forward. Yeah.
Like a pulp, like.
Shake it like a Polaroid picture. Yeah.
Shake it.
Shake it. 15 seconds. Shake, shake, shake.
I just don't want to clog the section for our future.
Yeah, just the rotate that you're doing, that's typically all we have to do to get the
Oh, will this bounce down the tube? It's got a long way to go.
It's gotta go up to the. I mean, the other thing I guess we could do, since it's kind of good and well in the tube, I mean, we could try to put it in a coral cover.
Can you reverse? Does this thing go backwards? No.
No, I'm just assuming it just kind of.
Spill it back down the tube.
If we stop slurping, it will just gravity will run its course.
Yeah, I just don't know how. Because then we have to get the low spot of the tube up above the coral there.
Did you think it's, is that it there? No.
if it's as heavy as you think it is,
It kind of looks like something is right in there. Anyone else see that? Maybe it's an illusion?
I thought there looked like there was something higher up as well.
Oh, well.
Oh, I
That,
see what you're saying.
yep. That's definitely something that is stuck.
Yeah, that's what I was talking about. Well,
But that would be, it would be moving a little bit.
We did just observe this worm
yeah,
Like,
it'd just be like a
suck
scuff.
really hard onto a rock, so maybe it's sucked really hard onto the tube.
Well, sorry to have possibly disabled the suction sampler on a whim. Just thought one of the invertebrate people would be curious.
They just come pre-macerated. We just won't have
On the other, we
a
could
laser
keep
show.
slurping and just get those brown things and then move on. Yeah, they should pass through.
Yeah.
It's
This
probably
tube
not
shall
fully
pass.
occluding the tube.
We might be able to get the rock from the tube at some point.
Yeah. So what do we think about trying to scrape some of these little brown? Yeah.
I was just looking to see if I could.
Yeah. No, you can select that much of it.
I hope we don't need that slurp anymore.
Right. Yeah.
Yeah, we're like little worms.
See the
and
black
things that are
uniform.
probably
Yeah, this is time.
sending tinophores down
So
into
the-
Yeah. Well, that's secondary. And we can always just pump. We can pump ad infinitum in the water column. See if we can get it through. All right. We'll proceed. We're gonna try to get some of those little brown things.
And it just comes
Scrape
like
them off the
this.
bottom.
You want me to turn the section off and see if it falls out?
Yes.
Sure.
Well,
Yeah.
Oh, there's our friend.
Friend fish.
I think we will like not. You can just keep the section going for now.
Strange fish.
You wanna look at that fish?
No, we've
Find
seen
out the fish.
we know that fish well.
We can do that.
yeah.
They're all over the rock in the foreground.
So, where were you working at? You were right there. Yeah. So, see how the little brown
one large sample.
Yeah.
Got it. Understood.
We just got an error on the super.
Oh.
Device reports readiness to read but return no data.
Call out if you want it power cycled. We can pause what we're doing.
I can do it while.
Hold on 1 second.
There's a nice little patch. There was another rock that we might end up sucking up.
Oh no.
If you just scrape a few patches of rock, the one over to your left has a few on it too. Yeah, here we're just sampling a few unknown invertebrates, so far unknown to us. It'll be interesting to find out what they are, even if it requires sequencing them. We've seen a few juvenile mussels at this site. Kind of makes me wonder if it's an incipient, sort of diffuse flow animal patch. We've also seen some really interesting evidence of iron oxidation, biological iron oxidation, just under the surface of these rocks. When we zoom out, it'll be visible again. A bright orange layer under the black crust. They might be. Oh, that looked like one that you got.
Exposed rocks rather than cracks or.
Yeah. Just take a couple more swipes and then call it good.
You turn off superpower and then turn it back on.
Hey, there's another tentacle thing.
Super off.
Oh yeah, look at him.
And super coming back on.
Thank you.
You can annotate it, but I don't think we should collect it.
Okay.
Oh, nice flake of.
Wow. It looks like sweet potato.
It does look like a sweet
Yeah.
potato.
What?
Beautiful iron oxide.
This is the iron oxidation that I was mentioning.
Gosh, no, super. Why are you doing this to
Sound
me?
super?
Sad super.
Sounds super, right?
Has it been cycled?
Yes, I did it.
So,
You shut it down there?
yeah, I shut it down a few times internally to try to restart it. Nothing, and then it power cycled it once up here.
So I would try unplugging and replugging it. Sometimes it's just so loose.
Okay.
Like it doesn't look like it's blinking on the
Yeah.
thing, so I think it's
Alright. That's probably well. Yeah, I think so. Alright. We're gonna stow it. And if the rock falls out, probably a good thing in the long run. stow it, and then we're gonna redeploy the temp probe.
We got a few stuff.
Two.
Alright, index.
Yeah, please.
Yeah.
Thank you.
New node registered with same names. Okay, that's fine. Hold on. Let me just. That's.
I shut the terminal down.
Alright, you want to do a full temp, please?
Yeah. Kill this.
Try computer restart too.
First I'll try the terminal again.
Looks like it's the computer's being slow right now.
It's because the battery's almost dead.
Ah, good catch.
Like, which one is this?
It's a US.
Here, just use this one. Doesn't matter. I'll find the right one. Actually.
Let's go to your
That's the mouse.
Okay, good.
your comms, Mike.
Yeah, keep moving.
Okay, and Sam.
Yeah, try to put the battery on.
Let's jump down.
Okay, now it's behaving.
Okay, so we're gonna do the same thing. Put the super and the temp probe together. We're also going to pick up and make our way on a 180 heading.
So is this just kind of like a background midwater sample?
No, we're not gonna pump while we're flying. We're gonna fly south until we see the start of the mussel bed, or until Roxanne tells us to stop.
Okay. But I guess, should the probes be a
The probe-
specific height? or
The
put up
we're
front?
when we land, the probes should be about a foot off the seabed. That makes sense.
So we might wait until we land just because the
Yeah,
terrain-
sure. The terrain, we don't know how it's gonna be.
As soon as I say that you're okay, stall on Rule mistress.
Just bring some back.
You said 180, right?
180, correct?
Okay. Pull status. Alright, now you are behaving. Good.
All right, we're heading in towards Chimlet 1 for what should be our last super sample of the day. That's not necessarily the case.
No, it is.
It is? Okay.
We only have this one filter left.
All right. Confirmed the last super sample of the day.
We'll find from there.
Alright. Yeah. Okay, we're behaving now.
So we get. Oh, did we get the little scrapey?
No, we didn't get the scrapey rock bits.
Okay, cool.
Dude, we stowed the slurper already.
I think we got a piece of it.
We probably. Oh yeah, we
Yeah.
must have because we broke a piece off while we were slurping.
One sweet potato piece.
Oh, but
That-
it wasn't, but not like the big salmon fillet looking.
No, but it was
plant.
pretty darn orange.
Those are definitely iron oxidizing bacteria that we like picked up, but the other thing looks different.
Well, there's not. So there isn't a single piece that we broke off that we could like grab. There was nothing that big.
Okay.
I'll be mapping here, so if they needed to stop and pick up,
Okay.
We
can
do have a
always stop.
marker on it. We have the
Yeah.
orange rock
How
marker.
high do you want me on pilot?
While flying? Close enough to see mussels, please. That was orange rock / super 5, right? We're going. Sorry, go ahead.
Right south.
Space out.
Until we see mussels, or Roxanne tells us to stop.
Will you open up my iris or so I can get a little bit higher or
I think oh, that's a skate egg.
Okay, I was gonna use this camera right here to do that.
Yeah, okay, yeah, I guess it's fine.
Yeah, I can come down
It's
low.
just a little hard to see what's live and what's dead, because there are shells too that are sort of looking for the edge.
Let's see if I can get that. See if I can not do some pattern in time.
Yeah, it looks like we're getting there.
Starting to see him off to the left there.
Mm-hmm. Yeah, if we are able to dip somewhere under the 5 meters altitude. Thanks. That should do it.
Lovely. Oh, is that an extinct chimney?
Inactive.
Inactive, excuse me.
Good catch.
Is there a marker here?
Can we drop a... There is a marker here.
Okay.
Can't tell what it says. Oh, well, there is some of that.
There is inactive here, and
I see the
oh, the mussels.
Yep.
Yeah, so I think we want to set down somewhere here, obviously not right.
Maybe like right. Can we set down here?
Where the dead ones are or the live ones?
What do you think of this spot here in the lower right as a set down point?
It should be fine, although I don't really want to sit down on a bunch of sediment.
Sure, okay.
So if we could maybe go to a spot that has less sediment laterally. Basically we're trying to be at the very edge of the mussel bed.
Very good.
Just
Left might be good.
to support the rams against the chimney pieces. Would that be
I could set my ram on this inactive chimney.
Yeah, that's fine.
Nice.
So you want the one that's in front of me or the one that I'm right above?
It doesn't matter. Either of that, whatever's easier.
This one looks a little bit more stable.
Good morning, folks. You're in the control room with the Dawn Watch, aka the Donner Party, DAWNER Party. It is probably about dawn, 7:02 local time on the Falcor 2.
What's that?
We are about to take our last super sample of this dive,
Those look like eggs.
possibly or possibly not the last super sample of the expedition. It's a larval crab. It's just settled. Good morning, Corey. 5 minutes early.
Once you're settled, we can pull the wand and temperature probe back out.
Yeah, it's behaving now.
Okay, great, so, yeah, it's just the, I got it. Okay, I'm gonna, Sarah's taking, is that the
Mhm.
7:30 meeting? You guys have it under control?
I think so.
No, we've got it.
It's the home stretch, victory lap.
That crab has just a little arm that's regrowing.
I've been doing super samples, basically, that's it, because the really like off chimney mussel patches or, clam, the clam beds that are way off chimney were all dead. So we didn't want to sample right at Chimlet 1 without approval of clam person, for you. So, we just did supers.
But we did find a clam site that Roxy seems to think is okay based on nav data. That definitely has live clams. It's called Clam 6. It's about 15 meters southish of the Chimlet and like just beyond 10 meters from any of the other clam sites. So you might be able to walk out a little bit further. It's like, I don't know, where
Thank you.
is, if this is, yeah, if Chimlet 1's down here, the clam site is like over there, southeast of it, southwest of it. You'll see.
Ready, I'm ready to purge when
She's ready to purge.
ready to
Anyways, if you
purge.
wanted
Ready
to,
to cook.
your next step would
Starting now.
probably be
Starting
clam
now.
sampling.
Six.
Not much, honestly. Well, there's biotransects and there's filming, though Roxanne seems unsure whether Tebnia. Yeah. Some Tebnia hiding under those muscles.
Oh, look at that.
Little ones. So these supers I need to check off. Hang on a second.
Limpets, nope, like we always forget the limpets.
Crabs.
Supers are all done, basically.
Supers are all done.
Well, we're doing the last super now,
Last super.
in the next 24 minutes.
Caitlin mentioned that Amy may not be that interested in inactives,
Okay.
so you might want to message her before you sample any inactives, which you probably do anyways. Clams we got to do, bio we got to do. There's mapping, and there are potentially some BBC objectives, but again, we're not really sure what their scope is. What Roxanne has relayed to me, though I haven't talked to Dan in
Dan's
hours.
awake.
He's in the
Okay.
corner.
Oh, hi Dan. See, he's awake.
Awake and alert.
That
Yeah,
makes
what
one
are
of
your
us.
goals for today?
Well, let's find some
it's regrowing
yeah.
its
Oh, yeah, it's little claw.
regrowing its little claw.
Wait, where's the
That
Okay.
one. That one.
Oh
This one
yeah,
looking at.
yeah.
There was a little baby one somewhere.
Kiloped challenge. Yeah, I saw one that like looked like it had just settled. Maybe it took back off again.
What was transparent one?
Oh no. This was one of the little orange ones that I think is a larva for, technically a larva though, it's quite large. For the Cyanogrea. You've seen them, they're like little crabs, but they still have their tails sticking out. I thought I saw one settled here, but maybe it's moved.
Yeah, I think it was back and to the right a little bit.
That's alright.
Yeah.
Don't worry about it. I'd rather look at all this stuff than delay. No. I'm not looking anywhere special.
Just confirmed the IGT's not plugged on, right?
It's, yes.
All
Two of them
said-
are
We're done
fired.
with the AGT segment.
Yeah, that's what I had to start to some of them up.
Yeah.
Throw them on the thing. Should come back. Oh, wow.
Aren't you spotting? It's spaghetti worm, yep.
Spaghetti worms.
Spaghettis.
There's a shrimp on the left and spaghetti worm at the bottom.
Okay.
We saw one of those in the water column earlier. I'm not sure what lifted it up. It was, I think it was diffuse flow or maybe disturbance of the vehicle. This is kind of interesting geomorphology here. What do we think is going on here, Rachel?
Going on where?
Porousness, like this.
It's just pieces of talus.
Okay. But like, why does it have all these vermiform sort of channels in it?
Because
Or
it used to
those
be
are
a
worm.
chimney.
Those are actually worm tubes that fell.
Here,
Okay.
on top of a chimney.
It used to be a chimney.
Geomorphology is the fancy word for the shape of rocks, as I understand it. Vermiform means worm shaped. I'm really not good at this outreach thing. Some of these words are kind of fun if you're a word person, but they get in the way.
Learning to define as you go is a skill for sure.
One of the questions in my qualifying exam for my PhD is they had me give me a bunch of species like, you know, the ctenophore sponge, worm, lizard, human, mouse, or whatever, and they said, okay, draw the phylogeny of these, and you draw the phylogeny, link them together, kind of like, you know, which of these things are most like each other. And they said, okay, now explain it to a fifth grader.
I would love that.
That's a great idea.
That is a great
I
question.
wish my qualifying exam was like that. It was much more chaotic.
But it was actually my comprehensive exam. It was a pre-qual, more general, but same drill. Yeah. Fire. Nice colorful spot. Yeah. Yeah. It's a nice little, I like that little inactive with,
Yeah.
with the squatty on top.
I think it's kind of diffuse. I wouldn't say
Yeah?
it's inactive.
Okay.
She's
Oh,
so
because of all
sparkles.
that, because of all
the
that.
fluff.
Yeah.
I think what we're sitting on is inactive,
That.
but that is fluffy.
It's fluffy.
It screams of,
Suspiciously fluffy.
it screams of diffuse flows.
Alright, got it. Observation. Suspiciously fluffy. Probably diffuse.
Run away.
Area where the
Gorgeous.
Yeah, that's not.
Yeah. We also haven't
No, it's
gone
all
up
over, it's all the way
to
open.
like the Chimlet
You don't
lately,
think it's all the way
so I
open, do
think
you?
that's probably the first place
Yeah.
to look.
Yeah, that's what
Just right at the orifice.
Yeah, it is all the way open. That's it.
I can do this now.
I think I tend to overuse the iris variation of sensitivity.
One click up on.
Yeah.
I can get you more.
No, it's fine. That's fine.
Yeah. How many claims do you need?
Yeah. clam up there?
Yeah. I just know like the clams can be tricky sometimes.
Yeah, I know.
Yeah.
10 would be optimal.
Focus. Let it try to focus.
Cause you,
Oh.
yeah, cause also those bioboxes are very full already.
Oh, it's a crab.
So it might be, and there's 2 crabs in one of them.
Those little
so
tednias in the
might-
crack are pretty cool.
It might be tough.
Curious if we can find more of those.
No, it's alright.
Yeah, of course. I was thinking
$17 for
that's
the site.
yeah, kind of the same thing I was thinking, but you'll do it quicker.
Mm-hmm.
Yeah. Yeah.
Yeah, they're just very large.
Mm-hmm.
That it looks like that squat lobster's gravid.
I think there would be some depths sitting.
You see the little orange eggs in there?
Yeah,
I
the
think
one
you might
Make
get
sure those are eggs.
3 per biobox.
3 per biobox. Okay.
Yeah.
This parasite
That's
that
fine.
you get in swamp lobster.
Oh well, maybe that.
And it takes them over
Don't
and
expect that much
makes
variation.
their eggs go where
Yep.
the swamp lobster eggs were, and they take care of them.
Mm-hmm.
I don't.
It's a horror story.
Yeah.
The
I don't
write up is but
Horror story if you're a squat lobster. It's dreaming for the parasite.
It's what?
It would be surprising.
Oh yeah, I agree.
Yeah, I vaguely remember hearing about something like that in a completely different species, different station.
Try it.
Yeah.
But they do, they aerate their eggs and
data
stuff.
were very interesting.
Yeah.
The raw data was showing, so it was showing that like there's like tidal cyclicity with the temperature from 2 vents, but like when one is kind of like at high tide, the other one's at low tide.
Oh, interesting.
Yeah, and vice versa. So we think like, well, it's just like maybe the pressure differential for one, like it just preferential, like it pulls more water out, and then it just goes back and forth. It's pretty cool.
Yeah, that's pretty cool. That looks good now.
Yeah, no, it looks pretty good, especially for raw data.
Good morning for those folks waking up and joining us, or coming home from work and joining us, or joining us at any time of day. You're at just the tail end, the home stretch of the Dawn Watch, diving on Iguana Spent Field, the first site we visited on this expedition. We have revisited it, recovered some equipment. Right now we're at the edge of the mussel bed near Chimlet 1. Nice familiar site, one of our favorites. And we're taking our last super sample of the dive.
No, I have not. We put a marker down. So if they want it, they can pick it up. Yeah, and I assume Caitlin went to sleep because she's like the 12 before a.m.
Barnacles. Yeah. Yeah, crustaceans. There's some that grow through, that bore through the shell entire. Yep, that's this one. Yep, Rhizocephala. Scary stuff. And they're also crustaceans. Wow. But that's not what's going on here. Those are not those eggs. I think these are the actual eggs. But if I ever see that, yeah.
I also took invertebrate biology.
Yeah, it taps them around the legs. Wow. Cool. Thanks.
Having this rock just reminded me of it.
So J-Rod was just sharing with me a little story about parasitic barnacles that infest these squat lobsters. And there's a feature on this, I think, on one of the old Falcor blogs. Does that gather where you picked that up?
Fun barnacle facts.
But we got on to talking about the barnacles because one of these squat lobsters, the one that's facing us, the one in the middle that J-Rod's focusing on, appears to have its own eggs, not in this case barnacle eggs. They're not super visible right now, but there's right below its face, there's kind of this little orange stripe. And those are probably squat lobster eggs.
This is where you can zoom.
I don't know, but I also the lighting's just not very good right now. For to see them, if the animal turns, they might light up again.
How many eggs are there?
It's like a whole clutch. It's probably hundreds or thousands. They're bright orange, and they're right there. They're tucked under the tail of the animal.
Yeah.
Yeah.
I see it. I saw it earlier too.
That's pretty dope.
Come on, show off.
Very fuzzy.
Sexual dimorphism.
What's that? A snail, I think.
Oh, it's a shrimp.
Oh, the shrimp over there. Yeah.
I didn't see that eyeball.
No.
Oh yeah.
A worm. Yeah.
Is that a barnacle?
Where?
No, it's a shrimp head.
Yeah. So you got a worm down here, a serpulid worm
Not that.
with a cool little feeding bit coming out. Which part?
Between the worm,
That's a shrimp peeking out.
All right.
Yeah, the eyeball is right there. I think there's a snail down there.
Yeah, we got Come on, you were showing off earlier.
So, crustacean eggs rely on oxygen basically directly from the water. It's not like the umbilical cord in a human where they get oxygenated blood through the cord. So when a crab or a squat lobster or some other crustacean has eggs, we call that, we say they're gravid, G-R-A-V-I-D. Oh, there we get kind of a peak. Yep, they get a little bulge in the belly under the tail there, and periodically the mama crustacean will unfold the tail and kind of rustle the eggs around, which aerates them.
Are the lasers off?
They are.
Or at least out of the frame.
This might be a good video highlight.
Yeah.
Okay, gotcha.
Yeah, I have to tweak the white balance and post a little bit, but that's just how water works. Does DSPL do RGB lights yet? Would there be, would it work?
Showing was trying to.
Well, we have blue, we have all kinds of weird colors, you know. Steve has a red one, a blue one, and what have you.
Are those red and blue emitters, or are they kind of filter from?
Oh, as far as I know, they're yeah,
Yeah, I don't think
pretty sure. I mean, we used to have filtered ones.
yeah.
I don't know that I've heard of any that are
Like the, well,
adjustable.
are there already two on the
That's when I asked RGB, RGB, and then yeah, like those, you know, Philips smart light bulb, 3-element LEDs.
Showing was.
Seems totally within
Working
reach,
on it.
and you need pretty high intensity.
Okay.
Showing,
Oh,
yeah.
So basically
You have
all
to wear,
4 IGTs
when
are
you turn the
on
camera as well,
ROD? Yeah.
because
From what I
everything's
understand.
combined on the set,
Mm-hmm.
it would automatically adjust the brightness and color spectrum of the lights to get the best photo.
So does that have a market in offshore, or is it just for like filming? And
That was actually just for offshore.
Huh.
Mainly.
Interesting. Didn't know it made that much of a difference when you're like turning valve handles and stuff.
Just to get the clearest, that's the thing, because when the video that they want to record, they want to record it the best possible.
Right.
There's a good shot.
Yeah. Mama squat. I have no idea.
Or the boat
Her
mamas.
buddy.
Is that Mama Rock?
Or maybe it's daycare. Maybe it's like 2 moms.
Very little is known about squat lobster social norms and gender roles.
Thinking, well, no, I know they're not shrimp, but the shrimp in my tank, they alternate back and forth between being male and
Mm-hmm.
it
Yeah.
turns out in the clutch.
Yeah.
I don't know.
I don't know about these. They're in a whole different. Well, yeah, they're in a whole different subclass. no, suborder. Anyways, they're pretty distant, but yeah, they do all kinds of invertebrates have all different ways.
Lobsters are actually crabs.
Yeah, these are crabs.
Well, they're anamurans.
More crab than lobster.
I think that's fair.
Well, lobsters are definitely in a whole totally different.
Oh yeah.
Like they're more crab than lobster. According to
Well,
the tree.
yeah.
When we were in Australia.
Yeah.
Constant conversation with the difference between a prawn and a shrimp.
I know. Yeah. And they're based the taxonomic definition is basically the opposite of the fish market definition, right?
Mm-hmm.
Yeah. So depending on where you are in the world, but they were talking presumably about the second segment, the saddle class.
Yeah.
They were talking about the kilopeds, the claws.
I think it was a couple small the number of them, the size of them, or something like that. If I remember correctly.
Yeah, I mean, I'm not a crustacean scholar. The character I learned was different, but there were probably more than one, and they change. Are you familiar with skeleton shrimp, which is another misnomer, another crustacean misnomer?
No, but
They're well, they don't look like. I mean, they look kind of skeletony, I guess. They're generally like shallow subtidal, where I know them from. But I keep thinking I see them here, like that there. For the life of me, looks like one, but it's not moving like one. They're really weird amphipods. They are, but they live on the bottom. If you look them up, they're pretty cool. But I don't know whether they're deep sea. I keep meaning to look it up and see. Yeah, like suspension feeders. Yeah, look it up. Yeah. They have a great display of them at the Monterey Aquarium. They're really pretty small. Or I mean, you'll definitely get there if you look up caprellid amphipod. CAPRELLID. There might be other things that people call skeleton shrimp. There you go. Yeah. Funky dudes. They've got like 6 legs and back they grab onto something with, and then they just whip around in the water and they grab things. They're very small. I mean, the biggest one I've ever seen certainly wasn't in the deep sea. Was maybe 4 centimeters, 4.5 centimeters. You've probably seen them diving, plenty. Yeah.
Once we're done,
Yeah, they're pretty cool.
to aerate the biobox.
Agreed. I think they're kind of cute when you see them.
That's what a mantis shrimp should look like.
Yeah, they really do. They really remind me more of mantises. And actually, we used to.
Oh, drama.
We used to catch these at the little aquarium I worked at in Santa Barbara. We had a moon pool on the wharf, and we'd like drop a sediment, scoop, and we'd pull these guys off of pieces of hydroid and stuff like that. And a bunch of people, visitors, told me that they should be called mantis shrimp. Yeah.
Domestic disturbance.
But that's a different thing already.
You're in Santa Barbara?
I'm from Santa Barbara.
Nice. I was stationed in Ventura.
Oh, working for Veneco or?
No, in the Navy.
Oh, right, station, yeah. Wainini?
Magoo.
Yes, it's the last one. Yeah.
I was at one of the test squadrons over there. Started off in Oxnard and decided that I wanted to move over to Ventura.
How long did you live there?
2 years. Got there at the end of 2005 and left in 2007 when I went to Iraq. I went back.
It's a really nice area. Go back there if I figure out a way to afford it or if I got job UCC or something like that.
I remember if you wanted to fly somewhere, you had to ask your friend first to take to the airport at LAX. And you always just thought like, man, what did I do to you? Like, are you mad at me? Like, why do you want me to take you to the airport?
Yeah.
Such a horrible guy.
There's no way around it, really. Neptunes? Wait, that's on Ventura County line.
Yeah, Neptune's net.
Yeah.
Yeah, we used to go there all the time. We'd ride our motorcycles down there. I bought a motorcycle when I was in California just to get
Just
gas.
for that.
No,
Oh.
just for gas.
Mhm.
Like the amount of money I was spending driving from Ventura to the base, I was able to pay for the payments on the motorcycle and the gas, like, and still have a lot left over.
And that section of the PCH between Magoo and the Neptune's county line is glorious.
Amazing.
There's a buddy of mine come down there, and we would dive right across from Neptune's and then just cross the street. Eat afterwards. Neptune's near like Deer Creek, Malibu, Ventura County line. Nice area.
Should be wrapping up on the last samples.
Alright, HD1 died.
Yeah, like these 20-minute super samples a lot. Yeah.
Just randomly.
We're done?
Not yet.
No.
Should be
Okay.
Very soon.
soon.
Very, very soon.
You go to
Got a nice little highlight there.
diagnostics and go to the port scan.
Alright.
6. Alright, I'm gonna come on with it. Let's see if it comes on with the green at all or any kind of jump. So no ground fault. It's probably a riser. I'll check it a little bit later.
Alright, this is the Donner Party shortly signing off. We're leaving you in the capable hands of Corey, Esme, Jesse, Drew, and J-Rod. Thanks for joining, and please stay with us.
Almost any of them are going to do that. Almost any of them are going to do that. Eventually, I would just have certain ones to recognize that you absolutely need. And then
That's okay.
Wait.
My family calls
It's a
me
different
Jessie.
system.
Alright, who am I calling Jess?
Jesse, in our lab.
Oh,
You just said Jesse.
that's true.
Jess.
That's true.
Alright, super complete.
At least I'm not setting the standard for mixing up names.
No, not a good. Okay, let me just back down.
Rachel and Andrea switching names.
Oh yeah, Pete called me Andrea. And then Pete called Andrea Rachel. It's a whole thing.
What's next?
Next, well,
Not yet.
Not yet.
do we start stowing
You can start stowing.
both of them?
Yes.
We are super done. But
Not
power off.
super duper done, yeah.
this should be up to date. The first two supers were not written in. These I messed up. 4 is actually free.
Was it?
There is a rock somewhere in the suction hose.
Is it Biobox 1 that I need to bid?
Both of them.
Both of them?
It's in there, and
Oh man, those
maybe the pilots will work on getting it out.
Okay.
What are we trying to
It's
get
not
out?
super important. The rock in the suction hose, I was just explaining there that there's a rock in the suction hose. If we end up dumping it back out of the hose backwards, that's not the end of the world. It has an interesting animal on it, but maybe not worth disabling the sampler.
Yeah. Okay.
Thank you.
Okay. This is for you. Thank you.
Do y'all want the crab in Biobox 1?
Yeah, those are,
Okay, so it's supposed to be in there thing? Okay, 'cause
well, sort of. I think that Stuart wants that.
I think he wants to come out.
Bonus crab?
I'll
As long as
make
he's not trying to eat the muscle.
Alright. Came with the muscles.
I'll make sure it's
Oh, you're fine. I can
superpower off.
Yes, please.
Well, I bet there's some cracked muscles in there, so I bet they'll just go for the head ones.
Yeah.
Alright, Biobox 2, does it have crabs?
Biobox 2 I do not believe has crabs.
Alright.
Corey obviously knows this for the instructions that were passed to me, where we'll probably need to put clamps in both boxes.
Yeah. That too. I mean, I don't think I will get the one, definitely not 15, definitely not, probably not 10. It's fine. They are not, I don't expect that much variation, so I'm not.
And I mentioned, so the site we found was
struggling there for a minute.
clams. It's
Clam
written-
6, right?
Clam 6, yeah. But you have options in that area. Look around.
Yeah, we can go there.
Alright.
You might just have to shoulder it left a lot to get that IGT cable to pop over.
So
So once the stuff is stored, can we please go to marker clam 6 and try to collect some clams?
Okay.
I guess I'm only maybe for Jen. Let's see if we can pull that IGT cable back a little bit. But I'm only seeing it wrap around one side, so it's almost like it's threaded through.
Okay. So it looks like here. It's definitely, it goes around that middle. Yeah, your weave through it. Can we bring the torch back out now?
Maybe not just yet. Pass it through. I can hand it off to starboard arm. We're still gonna have to get a good view of it in the sit action actually. So yeah, I need to go through that bite.
Yeah, if you go through that bite, you might be able to just stow it after that and it'll pop out of there. But if you want, you can give me the silver next time. Yeah. I'm trying to understand why if you go to stow it now like that, then you can brush the IGT cable off.
I kind of want to hand it over. Let's switch gears on stove though.
Probably gonna need to porch out a little bit to get you the stow you need. Gonna go real slow. Once you following me.
Yep.
Tell me when you're live. Are you live?
I am.
Okay, start coming out now.
That's probably good.
Cool. said mussel bed 6?
Clam 6.
Clams. You grab it and you can, don't you want to get your jaws like through that?
What?
I thought you wanted to get your jaws through the loop and pull it out.
I just,
Okay. Yeah, I think you need to go through the IGT loop, right, with your jaws and grab it? And then pull it through. I think you have the same grab as what you have.
Yeah, but I think what you're asking.
You get that with the stuff though?
yeah. So yeah, now I'm on the correct side. We just need to get to the. Oh, you know what?
What?
I was just looking at it down here, and it's just distributed differently. I can't remember. And if you can, as you clear that, if you can get away from the holsters.
Like, and let's see what it does.
Yeah.
Or Andy, have your hearing.
What?
Let's set
Oh, I
this
was
down.
yawning. Sorry.
Right over here, possibly. Or like up here. Going right back over there. Quick, we're on to clam bed 6. Somebody needs to pick up some clams.
Welcome.
I don't care. Whatever you want to do. That's fine. You can just leave Drew here for another show.
Nope.
Okay.
This time.
Yep. Thank you.
So clam 6 is the next location. Trying to get some clearance from there.
Let me get you up there.
The-
Maybe on there.
Give me a heavy one, man.
I'll get you there. Clam 6. Is that the one we did today?
Can't see anything.
So we come down, well, yeah, 180, 36 meters at 180 feet.
Roger that. Are we ready? Falling forward. Are we ready?
Yeah, you're ready to go. We have already collected the muscles, so we have the bio boxes are very full, so I think we just get about 5.
Or this might be our last chance to get plans though, so I think we should try to get as many as we can.
The bio boxes are too full. The ones that they have already confirmed that it's maximum like 6 clams or something that will fit in there.
Yeah, I looked at it, I don't know, we'll have to see, but I think we should try to get as many as we can if anything, so.
Yeah, but they're just really large. Even without anything in the bio boxes, it's difficult to fit things in there. So I don't know, from the other team I've got the information that based on the bio box capacity it's maximum of 6 clams in both like across the bio boxes. So
Yeah, we'll see, I mean, yeah, there is stuff in there, I agree, but we might be able to get more, but.
well, I don't think we really need more because I don't think there's a much variation in those.
And we're going
Did we get,
here.
so I don't think we have 30 yet, we don't want 30?
No, I don't think we need that many.
I have to move the boat 10 meters. 2 meters shy.
Excuse me?
And I have to move the ship 2 meters shy.
That will be, that will be 110. So yeah, around this little bit. So I think this whole area was clam 6.
Or next year, where you have a specific target, they're like 10 meters old.
Oh,
Yes,
this
we're
is
good.
it.
Yeah.
Okay. Yeah, whatever is, I mean, with the one in front of us, whatever spot is good to sample to get a few clams into both bio boxes. Yeah, this is the one in front.
Okay.
Be great.
I think I need a rock to probably sit under the coach. There's some live clams right underneath me.
Mm-hmm. Great. Get a couple of those and be good.
that was in it? Is in the thing, small thing?
If you find some that are
Anything bigger?
difficult to.
Alex? Small.
Smaller would be better because then they fit more in the bio box.
Like these?
Well, not the white ones, so not the white
the
ones,
white ones.
not the other ones. You can get some of those from this patch now.
Okay, I was looking below on one. Bring your head. Okay, I'm gonna torch out. You can reach those, yeah. You can reach those. Okay, you wanna pick some climbs? You want Thea?
Hmm?
Do you want? Is it just gonna grab any climb?
Pretty much, yeah. I think the ones, I don't know, it's difficult to see. The ones here like look a little smaller, but I'm not 100% sure. It's difficult to see when they're stuck in that.
That's it. Got it. Sticky. Firebox too?
We will distribute them about across both fireboxes, so whatever is easier to reach. Let's see if we can.
Kan man få tag i den?
Let's see how many. Well, firebox 2 looks actually pretty good.
Yeah.
A little bit better. Let me try firebox 2 first.
Let me open it first before you swing. Sure. I got 40% in. Hold on, I'm on. Okay, try that.
Yes.
crank it, probably will not.
No, it's probably where the like there were some cracks going into it.
Yeah.
Yeah, it's
Oh no, I don't. I think we don't want a vessel in there because then it will mix with the other.
You need to scrape it. Oh, there you go.
well, if it's attached to the clam, we can see it.
But if it falls off, I'd really rather not have it go in if we can get it off.
You get it.
Knock it off with the other one. There we go.
Okay, put you on. Yes. But you want to get that one? Let me port you in a little bit more.
who's 4 here right now?
Sorry.
Is that 4 or 3? What you mean? 4, I guess. Still be some. That's a big one. See the size of the jaws.
Yeah.
I think the flip, I might make it easier to do that.
Oh yeah. Got it.
Sort of shrink up as well. Why not?
Watching out. A bit more. Okay. Opening my box. Six, I think.
Are those? I thought we had like 5 or
there's-
something.
there's- No, yes, there's 5. There's 5.
I think if we could get 3 more, that would be ideal.
Okay.
Like maybe in the other bio box.
I think.
Unless the crab is trying to escape.
I think Alex will manage 3 more.
Sure.
Maybe pick the victim.
Is that all the way out at the same time?
Yeah, when we port in, we'll do that.
It seems to, it's a little easier when they're closer because it can get
Yeah.
right.
Roger.
Try flipping this one through.
channel. Find a spot. There's six. I think we could fit 8 in there. Easy. That's great. Easy peasy. Okay. And victim. Pull that one right sticking out.
Yeah, but I think it might not be as
Big.
well as free as I thought it was. Find out.
Is this just the number 7?
This one's a 7.
Number 7. Ah, this. Yeah. No, I know.
Just put one of these in, so.
It's kind of
Okay, good. I was just making sure.
Let me zoom you in a little bit more. Channel.
Get my copies in a minute. Any chance you have electronic
Perfect. No, I'll
Thank you
close.
so much, Alex. It's great.
Might have to. I don't know.
No, once we close it, it's gonna.
You think so?
Yeah.
They're not professionally portable at once. Oh no, you're right.
See, not even touching it.
Oh. Can you get in there and flick it, maybe? Can you come up more with the porch?
We're out with the porch. Let me open this my box for this one. I'm afraid it might hit that black muzzle.
Been holding out on me, man. Jeez.
What?
You're making me reach all the way back there now.
Is developing your skill.
Thanks, man. I just try to real lightly tap it one way or the other and see if it falls flat.
Yeah. You want to close short. There we go. I still think I would have found the spot.
Yeah, it is true. I just dropped it in wrong place.
Easy peasy. One more?
If you can make it work, one more is
Oh,
would
yeah.
make me very happy.
Come on. You gotta have faith in Alex, you know?
I have faith in Alex.
Yeah.
Much faith in Alex.
I personally like
See.
even numbers,
Oh,
so I
okay,
feel
it's
like
not
we
on
should
the
go
map.
for 8.
Well, if I get 8, then I have 25 in total, which is a good number for me.
But I like 25 because
I like
4
25.
times 25, you know?
It's very good
Okay,
math.
okay. We're
It's
in
very good
agreement.
math.
You get one more.
You like my math.
Yes.
Thank
I
you.
do.
I'm glad it makes sense to someone.
Tell how big it is. So the one, this one right here is probably easy to get just flipped around.
I'm
It's not a giant.
Are we only going for giants?
No. I said that's perfect size. I said it's not a giant.
Oh, I was gonna say I can get a bigger one. Which one was it?
That one? Yeah. That's what we're doing now. Yeah, it will make shoreline court very happy. Oh yeah. Shoreline court will be very, you know, almost watching out. Some big scratches on the cover. Look. Perfect. One more?
We have 8 now, right?
Yeah. Yes, it's 8. One more.
That's perfect.
You sure?
Yes.
Over here.
It is.
8
It
is
is
where?
good.
8 is good. As you wish, madam.
Thank you.
Very great work, everyone.
Thank you.
Perfect. Next thing would be to really put the IGTs, 2 IGTs on the lander and send the lander up. The mini lander.
Oh man, that means it's almost IGT time.
Jesus God.
Well, we have done all the IGTs already. These have to be sent up.
No, she means. Oh, I meant like.
Is this actually
Oh,
the
yeah,
lander right here?
Okay. Yeah.
Is it?
IGT time for you.
I mean, it's called lander.
Is that the lander, Corey? Very close.
Yes.
Oh yeah, I think that's a recent one. This one? Oh, see, this is the problem. There's a bunch of different ones named lander.
Yeah. We are looking for the mini lander, the BBC mini.
That's the name of it. Is because there's one recent lander here.
Yeah, I might have to get someone from the previous.
We can get the pointer.
Which one would you like more, the acai berry or wild berry blend?
Do you know what beacon we have on lander right now?
I believe. Oh, on the BBC.
It's my flux. I don't know. We've got a bunch of markers named lander. So I think I know which one it is. Actually, see it.
There's also marker BBC lander, but that's kind of pretty much off, like in the.
Exactly. It's too many landers. This time we can figure it out. Because there's maybe it's this like. That's not that one. I'm looking for the most recent one named lander, assuming that was ours. The one we're interested in. I don't know that. Sure.
Problem is we had 2 landers on this.
Yeah, well, the other one was labeled SOI.
Okay. Consistently?
Well, no, I don't know. I don't know the answer for that. I think the lander I was clicking on is like pretty, I don't know, not over here.
Yeah, I can just turn the beacon.
Yeah.
Right.
Let's just do one marker. Because we need more markers.
Yep. There it is.
Okay, just a second. Let me check this. Okay. Let's see.
I can read it out for you.
Okay.
107.35.2. 12-91 decimal 92.45.856.
Oh, it is the one labeled BBC marker. Okay,
Okay, good.
so we're going off from that then.
North.
Okay, good.
North.
Yes.
Yeah. Okay. Shall we, madam? We are gonna go.
That's the one I just added.
Oh, okay.
For line.
Well, because I didn't, there's
We're
so
gonna
many
go.
landers.
Yeah.
And that's not the most recent one.
I'm good.
What's
Sorry.
going on?
Oh, we've got a ways to go.
It was
I gotta
my
move.
chair was blocked, and I
Oh, okay.
went
Oh.
real far back.
Okay.
So just for rough estimate then,
Really
we're going
scared.
about 400 meters at 14 degrees.
Roger that.
I'm just
10 degrees.
start getting the boat moving that way.
Yeah.
Are we stopping anywhere in between or going straight there?
Straight there.
Okay.
We do everything else on the, like once that is done. I think we have some biology transects left, mapping and filming. But I think it's best to send the IGTs up as soon as possible.
We are on the moon.
Bridge, are we?
Already bridge.
Hey Ollie, we're about to run up about 400 meters north-ish.
400 meters north, copy that. What speed would you like?
0.3.
0.3 is good, and it's north-ish, but it's close.
0.3, fantastic, thank you.
Thank you.
What's the matter? Nothing's the matter. Why?
You're smiling.
I'm smiling? No.
Not everyone is mad when they're smiling like you, though.
Thanks. Thank you.
What's the matter with you, huh?
Wunderbar.
Wunderbar?
Yeah.
Huh.
Are we in INS depth?
That's a great question. No.
Okay.
We're now in INS depth and INS position. And INS altitude.
Okay, great. Now I can put them all at depth. If it's on CT depth, it won't hold depth. It goes down. Cruise control.
You know how they can, in the bridge, they can watch us, and so they can kind of see what we're doing
Mm-hmm.
and like see who's flying and whatnot. We should put a camera on the bridge so we can see who's there, so we know what their name is before they
Yeah.
respond to us.
I know, it's kind of, they can see the RV hangar and this room usually.
Oh, they can see everything.
And we don't see anything.
Yeah.
It's
Well,
kind of
no,
like,
I mean we could. I don't actually.
we could?
see the
Can we,
bridge?
Oh, yeah.
can we?
We could, but they've put a little sticky note onto their camera.
Oh, can we put a sticky note
Can
on
we put on
this?
a sticky note on that camera? We can. We've 3D printed really nice covers for most of them on the main deck. Just J-Rod uses this one to.
Make sure we're not doing bad things.
No.
Yeah.
No. What is that?
I don't know.
Sea
Sea
cucumber.
cucumber.
You know, I thought I knew what a sea cucumber looked like, and I'm realizing that I had no idea. Or at least like the benthic.
But
I know
usually-
what benthic ones look like.
And usually they're like benthic, they're like few
Yeah.
pelagic.
Like, what the heck is
It's beautiful, that's for sure. Whatever it is, it's mesmerizing.
I think usually Ollie is up on the bridge during this time.
Yeah, I had to wait to hear his voice. I want to know who it is before I.
Yeah.
I can't
Okay.
tell if I love it or I like really hate it. Not the sea cucumber, I'm sorry. Just for context.
I love it.
You love it?
Yeah.
Okay. Maybe that's why I'm not a biologist.
They just see us or also hear us?
Oh, they can hear us, definitely, for sure.
We
Right
don't
now?
typically have the volume on, but anybody on the boat can hear us. I mean, the whole internet can hear us.
Yeah.
Yeah, that's true.
Right.
Not just part of it. The entire thing. All the tubes.
You said the entire internet, but feeling like it's a total of 50 people.
I mean,
I feel like it's pretty generous.
what's quality over quantity?
What?
It's quality.
I was gonna say
Oh yeah, I mean in terms of metrics and whatnot, it's hard to measure quality.
really is.
I'm sorry that I just keep doing
Oh, that was yours? Was it still locked out by the time we got to it?
Yeah.
Was it really? I was trying to guess after Lamont.
That's hilarious. Yeah, maybe I should leave
What is-
things in.
Is that your personal one? Okay, good. It says it belongs to your organization. I was like, well, that means I can try to open it. I guess all the obvious ones.
I guess it belongs to the organization, so you just don't be alone.
I figured if they didn't want me open, that they would have made the password 1, 2, 3, 4, 51.
I was about to.
Looks like you know, it's right now.
J-Rob was right, you said shut up, like, please.
Oh, that's amazing. Yeah.
Yeah, I'm a lot more careful now, I've learned.
We used to do show dollar mails to those computers.
Oh, what?
Show all, like every contact around the world.
Okay.
When we used to like publication. And that guy showed out at some point, they tried. It was super fun. But yeah, exactly.
No fun.
Everyone was up there with a lot of fun. Conversations with myself. That was a Teams on my phone. And then it's a computer Teams conversation. And then I would speak like that. And then I wanted to maybe get things up like just to remind you how awesome. But then also, I don't think it's still other people from
Aw.
Teams. Not like you're program on this was really affectionate for what's going on.
It works.
I was asked to stop that one. It was really fun. Yeah. It was. And I'll do it.
Sounds like you're really pushing for the password.
That's an organization's password. Where you can go on the screen. Is that pretty much is that an MLT specific?
Yeah, it's fine.
Is that how you run on ship shorts?
Yeah, I mean, I can just run it through the computer, but it doesn't just take it anywhere.
Yeah. Do you have any more?
Today's almost done.
Was it up there?
It's gonna be with the Lucas again,
Oh, really?
Yeah. So we might just pop in and we won't ask questions.
Is everything gonna do it?
I think I texted it.
think you're gonna find a lot.
It's actually quite a big one.
It is. That was a 400-meter move from where we started.
We're on top. Almost. Yep. It's almost 20 meters.
How's Ben doing today?
Wunderbar. Lightening up again.
20.
Show up.
Sorry?
Living said to show up.
What did you say? You were wondering.
We decided to show up.
Gee.
What's next after this quarry? Is it the dance show?
Well, that's our dream.
The what? Oh, well, you have to send the lander up first, so.
Yeah. Then
Well,
what's
once
after
that's done and we follow the lander and stuff, I think then we do the filming or the transects.
Gotcha.
It's
The
cold, they
dance
want to
show.
train a bit.
The camera's small, the battery was.
The camera's finished, yeah. So yeah, we'll send that.
It's a dance show after this. After we send the lander off.
Yeah, I think so. Either the filming or the transects for
Transects the quarry show.
Philip.
Philip?
One of our students. Yeah, after this we just have some biology transects, mapping and filming. So I'm assuming the filming is maybe what we do
Already.
first.
Hopefully it's filming. Gerard's gonna come walking.
You're excited about that. I'll break down.
Figure out from then though where he wants to do the filming. We can check some of the areas where there may be vanilla.
I just realized you can turn my phone. Sorry. I didn't know myself too. I just realized
It's done.
the battery will probably last this. We're gonna turn around that night.
Bridge, can we increase it to 0.4, please?
0.4.
Hey, can we down it or
Yeah. Pulling a dead horse.
Archie showed us yesterday how to make knots.
Knots? You want to
Sailing
learn
knots.
knots?
Yeah.
Who showed you yesterday?
Archie.
Ooh.
Yeah, Dan, Katrina and I were learning.
Nice.
I like the splicing.
Anybody on this ship can teach you knots. Maybe. I was gonna say maybe not Alex. But
What?
by his own admission.
I'm surprised whenever I get a bowling that turns out it's a bowling.
Yeah, I saw it the other day. That's not a bowling.
Yeah, but what it is, it's pretty exciting.
Yeah. Okay, start moving that way. They must have used the cut already. It was the other lander they need to cut.
That would have been the other one. I don't know that did they use it?
I think so, because Edwin mentioned something about Gerald told him the knife really worked good.
Would be what?
The knife really worked good.
Okay.
To Gerald told him.
What?
I'm looking for something to munch on.
You what?
Where people
I'm looking for a cat.
the most common way is like when someone's really hungry on the bed.
You want to write it down?
But normally you don't come out this place. So what you do is you just lower it down until it's stationary. That's it. The job floats. The people just come and sit and have this just crush. So, but then there was other excuses with all this stuff.
It's so weird. I don't do cordial today.
You don't have to what?
Cordial today?
Ah, yeah.
Usually a cordial.
Not only just like not right at the train, but it's like
This is Corey again.
Yeah.
I guess again. You know why I'm calling?
There's so much like elasticity bumpers or at least you kind of let something go to before the SOI for recovery and erosion, then no one has to be there. We won't need to be we don't operate vessels, so it doesn't matter. But other investigators when it happens. It's all relative. There's a lot of stuff.
Oh, I'm
Any moment now, Corey.
Any moment?
I know.
Yeah. We need to put the first two IGTs on the
I
lander.
don't know what my problem is.
IGT 1 and 2,
I think
probably.
it's if I
It's
get
easiest
too
to reach.
much sleep. If I get too much sleep. I don't know what's going on.
40
My
more
sleep schedule is so messed up. I was sleeping for like 12 hours the past 2 days because I had time to sleep and so I utilized it. And last night I got like 4 hours maybe. Tonight I won't get that much sleep.
You gonna go fish washing then?
Yes.
I'll get your lights.
That's good. Tomorrow.
But usually when the ROV comes up and the only 2 of the IGTs still have to be processed,
Yeah,
it takes 4 hours or something?
about 4, yeah. It's about 2 hours per IGT. It depends on whether or not Jill does gases. When, like, sometimes for the last IGT, she'll take the gases, but she won't run them. And so that takes less time. But if she runs gases, it's typically about 2 hours per IGT.
What is the most time-consuming part of the sample? That takes a lot work.
I would say gases. Yeah. Yeah, sometimes gases take about an hour. And then sometimes titrations, but for the most part you guys get done with your titrations and everything. And we have an order in which we do things, so like you can't finish the IGT until all of the gases are done.
Yeah. And that's just because that's easiest to maybe contaminate other?
Well, like the very last thing we do is gas residue and drag. So gas residue is just like you take a little bit of sample and then you sparge it with nitrogen until it doesn't smell.
That's some sort of parasite.
And that stuff comes with like, hello sir, what is that?
He's about to do his business.
He's what?
He's about to do his business. So I'll leave him be.
Oh yeah. Nice.
Okay, I'm gonna go away, leave him be in private.
Yeah.
Oh wait, he's turning.
Look at his big eyes.
I should have left the lighting. Beautiful plan.
Probably blinded if he's not already blind. Hello. Hi, friend.
Oh,
I'd like to name him Frank.
Oh. Okay. So I'm gonna head out.
Thank you.
I drew Earl for the book.
Yeah?
The
You
squad.
drew?
Yes.
What is that? Yes. I'm
Thanks for
so
doing that.
excited.
This is very exciting. I was about to draw Earl on my little cup, but then I drew an evil crab instead.
So, I think it's Earl, definitely.
R.I.P. Earl.
I see.
I hope you ended up somewhere in Small Lobster Heaven.
Yeah.
Or if not, maybe he ended up in the bottom of the ocean again. Oh, there it is.
He looks confused.
I don't know. I don't know.
Earl was the squat lobster that was attached to, I think it was this thing actually.
Yeah, it was this
Taken
lander.
up.
He was there for so long. I was really hoping I could see him on the other side. He didn't make it. It was really depressing. I was very sad.
Well, I don't know. We just maybe let it-
Crustaceans are pretty hardy. I feel like he probably made it, and then maybe he made it back down.
If it's this side.
That crab is gonna come up fighting.
Is he still there? I mean, yeah.
In the bioblocks? Oh yeah.
Yeah, I think
He's gonna
Stu.
put up a fight.
Is there, how many are in there?
I think just one. I think Stu
I
had
don't
one
think
or two.
both are
There's
coming.
two.
The one get out.
Oh.
No, there's two in there.
Oh, there are two in there? I just saw one.
All of the tiny crabs have made it like hours after it comes out. They're really cute.
The basket's on the other side.
I know the crustaceans are very hardy.
baskets are there.
Oh, is it? Yeah, yeah. I see it now.
Oh yeah, that crustacean's gonna put up a fight. Nicole's gonna be throwing hands. Sure.
I think the crab is for Stuart though.
Huh?
It's for the foundation.
Mm.
The crab.
Oh, it's for
Charles
CDF.
Darwin. Yeah.
Ah.
Wonder if I should land this on the porch. What do you think?
I think they I think they preserved one too, like a big one.
You think?
Wasn't it the dead one though?
You
We're
think?
dropping off 2 RGTs.
I don't know.
I thought they got a dead one. I like.
So, I think what we'll do is, you want me to park it on top of the yellow block and we could drop it off? That's the
Let's
best way.
forge out, yeah, maybe.
I said good morning to the lander.
Oh, okay. I think
Say
they
good morning
appreciated
to
it.
the lander. So cute.
Oh wait, this is like
How
a
did he make this?
yeah.
Like genuinely? No, I don't think it is.
Yellow plastic he made to hold as a mount for his GoPro.
Oh. It's so impressive.
The way it's cut different, it gives it that thing.
We're getting a porch out.
That hat really fits him.
Oh, yeah.
It's okay. No judgment here, man. I'm also misfired.
Okay. You think if you land, you could bring it up? No, probably not.
We could try it.
We could land in the corner maybe, or on the yellow part.
I mean, try landing on the yellow part. I'll
Okay.
give it a shot. If I can't, then
Yeah.
You have a
Just-
friend.
I do have a friend. He's been here for like 3 days.
Aww.
Such a dive streamer.
What's his name?
Freddie?
Freddie?
Yeah.
Did you just make that up?
No.
You kind of looked like you did.
I had to think about it for a second because
Did you
Frank
make
was
that
still
up?
stuck in my
Oh,
head.
yeah.
So I was like, not that.
Yeah, not Frank.
Not Frank.
Sophie just don't make up stuff.
Freddie. He's so cute. I'm pretty sure my dog had that exact toy.
Yeah, it is a Walmart dog toy.
Yeah. It was her favorite. She loved the dog. She had that.
I should get one for my dog.
Yeah.
You have a dog?
Yes, I do.
What kind of dog?
Sorry. You can't mention that you have a dog and me not ask for dog pics.
Yeah, no, I totally understand.
I love dogs, but mine passed away.
Mine passed away in December too.
Mine in March, but he was old.
Just because he's old? Oh, he's old.
Sorry, I didn't mean it this way. Just like he was just old and
What kind of dog?
an Australian cattle dog.
Too low.
Aww. Sweet baby.
Yeah, he was really sweet. He was from a shelter.
It's from a what?
He was from a shelter.
He's so cute. Yeah.
He had some problems.
Come on. No. No.
This is a
Hello. What's their name?
Go.
Coco?
Coco.
Coco. Sweet baby. Have I shown you pictures of my dogs?
Oh dear.
I have multiple. What a good girl. What a good cute big dog. She's a good
It's gonna be easy.
Pardon?
Be easy.
Easy peasy. That's what I want to hear.
Have we been handing it up or just picking it straight up?
I think handing
This is
it off would be better. I think.
and bear.
Because it's hard to put that up.
Bella's the one who
If I
passed
want
in
to get it right
December.
quick.
And that's Mr. Bear.
Let's do the starboard.
Okay.
Actually, I
Look
could
at
have
him.
grabbed
He's
it.
such a little rat.
I'm gonna pick up.
Oh.
I'll grab this one.
Okay, so which one do we want to grab first?
And
Lauren?
then
Next
baby.
one. So you
new
want to
addition.
grab.
I guess here. Yeah, let me get that one.
She
I'll
is
give
a
it to you when I'm ready to.
little cutie.
Okay.
I'll get you like you because this is loose
Yeah.
enough perch. It kind of
Yeah.
should let you be a little far if you need to.
Look at her. Oh, super fuzzy. She went for her first haircut and they gave her rose. It was very cute.
yeah, Bruno did that too.
See what happens if you do it.
Yeah, and then this is Bellaboo. And Bear. We're besties. Best friends. Best boat dogs.
Whoop.
That's right.
The tubes are hooked up to each other.
Oh, it's cute. You're so cute.
Come on. There you go.
Yeah, those hose clamps. There they go.
They had Christmas pajamas.
And the next one.
Oh,
That's awesome. I love them so much.
Oops.
I assume we're kind of coming down on it with the hammer. Grab 90 degrees a little bit.
Yeah. The nozzle goes down first.
Oh, that's right. That's
Yeah.
okay. So you can just spin it.
Okay.
The other one. Okay. It's locked, but the jaws are not locked, just the
Right. Right. Sorry.
That's alright. Let's see it. HT2. Yes.
Do you want me to tilt it?
So they're just gonna come from the top of the disc.
Are you sure you could do that? Because
Do we have to go from
the nozzle has to point down.
yeah, but if I was thinking if they come in at 90 degrees, typically,
Try.
I don't know.
Give it a shot.
That'd be great. So,
Wanna go?
yeah.
Yeah, I can give you a good picture. Let's see. Let me give you a little more zoom.
Almost like the fear of the porch.
bass. I think so. Joe Corey says you're the best. Right, Corey?
Hmm?
Said,
So what were you?
yes, it's all the way. Alex told Alex.
Yeah, that looks great.
Cable. You want to just deal with the cables after we get both of them?
I mean?
You want to just deal with the cables after we do both
Yeah,
since we're so
yeah,
close?
sure.
Yeah.
And then give you some sit. That's it.
Trade.
Yeah.
Good. Yeah.
Right. That's a bad angle.
That's the same one as I did last time.
90? Okay.
Yeah.
Ready?
Only difference is on this side I have the pipe to deal with, so we'll see how that goes.
No, that's actually, I thought, looks like earlier you were angled more towards the nozzle.
Yeah, I did.
I think
First I kind of fell into the notch.
Stand by and give you a good zoom to get the zoom in.
Yep. Just get ready.
Thank you there. Wonderful.
Great. Very nice.
Very nice.
Is it easiest to put the bungee on there over the handles?
Oh, I think he has to put the rings first.
Oh.
Oops, wrong. Which one is it? That's that one. That is. Is that it?
Yeah.
Is 4 on top of it?
Yeah, I think that's right.
Did I drag 4 out when I took 2 out?
That's okay.
No, I think it's
Yeah, I'm not sure if
it's
it's
ready
if this
for-
one is the right one. Is this?
They would have. They would only have put 2 on top.
Unless it was put backwards. But no, I think it's the right one. I think it just might be threaded through,
Yeah.
of course. Yeah. Threaded through or looped?
That looks like it was threaded through. I could pick him up. You want to pick him up and see? Hold on, let me zoom in.
Yeah. That loop.
You pull it. Oh gosh, I think you're right. Oh.
Just a
That's
lot of
no
time
good.
loss. That's fine. I'll just lift it up. See if we can't make it a little easier to untie.
Why? Oh, sorry.
That's when you tilt it a bit and move it and go this way.
What a bummer. I think you need to go underneath it. Oh, you know what you can do is
I'm gonna try to set this to the outside of it and just put the pedal back on. So you're gonna try to reach. It's gonna look really weird. So lock the drives. Just park this out here for a second. Does that zoom? Can we zoom out on the text?
There you go.
Yeah.
Who do we have to thank for this?
I think they do this on purpose. Just to mess with us.
Can you throw it?
Throw it?
No.
Probably should have stuck out HT a little further. Maybe that would.
Yeah, that one.
Here's the port.
Okay, let me move this puppy.
Right a little bit. Yeah. Cool. I'll just stick this back on this for now and get the arm out of your way. I needed to send this in. It just took my view away.
Sorry.
No worries. I got it. Let me get out of your way. Actually, I can probably turn the lowers on, not off, but all the debris is gone. I have no idea how to say you're welcome in German. Hey, Corey? How do you say you're welcome in German?
You're welcome.
Yeah.
Bitteschön. Bitteschön.
Oh, that's right. I knew that. Bitteschön.
Wow.
He said,
German?
German.
I'm practicing my French
Bitteschön.
right now.
What?
I'm practicing my French right now.
Are you going to France?
No.
Oh.
I started Duolingo and I can't stop.
I'm jealous. I have the opposite problem. I can start Duolingo and it's super easy to stop.
Which language did you try?
Greek.
Oh, okay.
And he's Greek.
No, I'm half. If you ask my cousins, I'm not a real Greek because
You
I
can
don't speak
practice your
Greek.
Greek with this mirror.
I know. I practice all 5 words I know.
Which of those? Which of those?
Like, good morning, how are you? That sort of stuff. Please, thank you. I know like 7 words. Shoot, something like that.
What's good morning in Greek?
Kalimera.
What is it?
Kamimara.
There you go.
Ah.
2 out of 2.
Come on. I was trying to
You
get
could go to Greece. You could get by.
Oui.
Oh yeah. Ben could. Come on. Alright, I'll just leave it to the outside and then fix it.
Speak English. There's a lot of multilingual people here on the ship.
I learned I speak as much Greek as Joey does.
Hey, what is that? Come on, give me a break.
Did you know Joey lived in Greece?
Who?
Joey.
Oh yeah?
Yeah.
Is he underneath it? Oh
Yes.
my gosh.
I think they did it on purpose, man.
Hold on.
I can just set this over here and grab it from this one.
Is it better? I think it's just. Yeah.
No, it has to come up from over here.
Yes, yes. You wanna just. Okay, I should be able to recheck. That is not fine.
No mistakes. Thank you all. Doing so.
Who do we have to thank for this?
Who has the.
This is like
Oh my gosh, that's tangled in that too.
7 years of French gets you. Beginner level.
This is like on the file list too, because this is also tangled on this one.
This is not
I
fun.
spent 7 years of my life taking
I mean,
French.
someone probably is laughing at us right now.
Yep.
Ooh, wait. Do a post-op on like the Mediterranean.
Really?
This is stuck
yeah.
right
Oh
between
yeah.
here.
This one's stuck.
I lived
Oh, yeah.
French at my major level, so intense. Like I didn't have time to focus on it. My math classes and science classes were. Also, maybe if I hadn't placed a
I
different
think Corey gave him the idea.
Yeah, that's probably what it was.
Yeah, I think so. Corey, this was your idea, wasn't it? You told the boys to do this to us.
I think that's what happened.
I promise I'm innocent.
That's what they would say too.
Mm-hmm.
I'll be back.
You're gonna blame Jill.
FYI, we might come in here with the kiddos.
Jill did
Who are the
this.
kiddos?
We're connecting with a local school in the office, so we're just
Oh, nice.
there are people here.
Is that all we had to do? I thought I had to go underneath.
That's enough.
Okay.
That's clear.
We do in fact exist. We're doing work in theory.
I thought it was crossed again.
No, I think it's just something. Well, we're about to find out. I thought it wasn't, so I could be sadly
Happy
mistaken.
birthday.
No, I think you might be right. Wait, wait.
I gotta get around to this.
Ship to shore.
Oh yeah, there you go. Perfect.
Still like outreach.
Still the best.
I'll be back.
Hear what Corey says.
That's right.
See? Corey did that swan smooth action. Wasn't there a band?
Wasn't there what?
Wasn't there like a band?
There's a bungee on it.
There is?
On the
Yeah.
box. That's why she asked if we wanted to do it before this.
Yes, that's what I was asking in the
And then
beginning.
you-
The,
oh,
the
yeah.
bungee that needs to go over the handles.
That's alright. This will get the cables as well.
Now,
you know what? That's gonna be where it's gonna be. That's not. That ain't going nowhere.
That's not going there.
You gotta hit it first.
Well, that ain't going nowhere.
Yeah.
Did you see the knots we found?
The what?
Did you see the knots?
Knots.
There was the paddle for 2 was threaded through a cable for 4,
Oh.
and then the paddle the cable for 1 was underneath 3.
Nice.
Yeah.
It's a course thing for them.
Wondering who put it there like that.
And then we found out that Corey convinced the other ship
Yeah.
to do that to us.
Oh. Yeah. Definitely.
Where is the hook?
It's on the side of the basket.
Can I see it?
Oh, the yellow one.
It's the
Yeah,
yellow
yeah.
bolt sticking out.
Hold on.
Oh, yeah. Okay.
Oh, sorry. Then you rack out and you got a view.
Have we been stretching it over the handles like this before we go?
Yeah. That's what you need to do.
I don't know if we'll be able to get it with this arm at this particular location. I can hand it off though.
Yeah.
I think you can.
And then she let it go.
possibly-
Have we been doing it on the rope or the bungee?
I've done it from the cord arm, but you would hook the bungee.
Yeah, so
Hold on.
The
Let me
ropes
zoom
that
yeah,
the
there's
rope.
no way that I'm gonna reset without crushing the box.
Okay. So I will grab it from you. Give me a second. No.
And the rope exists so you can let go of it.
The rope's a great thing. It's just we're in a kind of a
Yeah.
perch, just real close.
It's definitely easier on the port side.
Which is why we have that.
That's the only reason we have that.
That's the only reason
It's
we're
always
on
good
that.
for. Just for this one maneuver.
Fortunately, it's still good for something.
Yeah, Corey was trying to tell Ben that it doesn't. When things get old, they're not.
Yeah,
you know.
just let him die.
Just put Ben out the pasture.
Twisting my words.
That's what she was talking about Ben specifically.
I didn't talk about
Yeah, just it was old, so it died.
Sorry,
What?
hang on, to freeze it here.
Okay.
Sorry, Ben.
Yeah,
Just the way it goes.
I know.
It's life on the farm.
You know, that's true.
Well, Alex, you took the picture of Ben sleeping.
I deleted that picture after I
Yeah.
showed him.
You did?
He hit it.
When was he sleeping?
I mean,
I was not as resting my eyes.
well, you get it from that pocket right here, because I can't give you a good spot otherwise.
You say you snore when you rest your eyes? Is that
Yeah. Corey wasn't entertaining us.
What?
There you go, right there. One more. Oh. Think so?
I got it.
Okay. Open it.
Just swing back and hit you anyway. Nope. Stay close.
How do you all feel about the master cables being right over the desk?
I prefer it, but it's a little bit tight for pilot
Course.
if you don't care.
Of course, this cable.
I like it.
There are certain slips, in fact, you can let out more cable. It's,
I think it's nice because I like how gravity pulls it back.
yeah.
You do that?
Who?
Drew, did you do that?
I did.
Yeah, thank you.
I tried to leave the surface loops hanging a little bit so that gravity would pull back now.
Am I in?
Are you just gonna do? Are you gonna do the
rope, or are you gonna hook the bungee around?
The bungee goes on the rope or on the pole.
Oh.
Otherwise, it would just.
Now you say that.
Actually, so
We just had this conversation. That's why you're putting the board on.
welcome to the conversation.
How am I gonna put that on the bungee?
You can
just
do
stretch it out.
Where there's a will, there's a way.
If I look up, you were not in the conversation.
Apparently, you weren't either.
Okay, all right. Enough jokes at my expense. Okay, wait a minute, I can't see that.
You're a little bit lower than it, but you need to come towards us still. A little more. A little more, and then
There
come
you
up.
go.
Come up and towards the basket.
Thanks, Dad.
Yeah, no problem.
Oops. Oops.
Nice.
Oops.
That counts.
Wunderbar.
Or-
Wunderbar. Yes,
Oops.
I would agree.
I'm gonna make that cable.
Yep, that's fine. We'll go with it.
What is wrong?
Well, the cable's still in our porch too. We probably should figure that out.
Probably.
Yeah, I got it. I can do that at the start.
Stretching my neck here.
I'll just do it.
Okay, what are we doing now? Oh, that's it. Yep.
We're still behind the soup there, it looks like.
Okay.
Oop. That's a
Oops.
piece. Still there.
Okay.
You gotta do that with the
What I
board.
can do is, okay. Okay, never mind what I can do. You can do it.
Just gonna fly away really fast.
Yeah, that's what I was thinking of doing.
No, let me get that with the quarter. I'll take it off the super.
It is hooked under the super one.
Yeah.
Is it?
Yes.
Yes, it is. I knew that. Just Alright. Okay, let me give you a better view. Nope, that's all the view I can give you.
Can you not just brush it?
I'm tired of trying to brush something. It always fights me.
Could have, yeah, I was gonna say you could have just flipped it over.
Except now
What's on top of it?
you're in the bite.
Yeah, now I can just put it back. Because the problem I had before was that it was behind, like underneath the hose. So I just lifted it.
Okay.
He wanted to show his manipulation skills.
No.
No. See, expertly done.
So at least now it's not hooked on. It's just not on. I think. It just made it worse too.
Yeah.
It's better than it was.
Oh, and use that arm. Pick it up. Flip it.
Yeah, I would pick up the cable and only damage it.
Hmm?
Oh, there's the cable. I guess the transmission would rather pull it from this side.
No, we're gonna do it.
I'm just gonna take the paddle off and pull it away, so that way I don't push it back under where it should be.
As you wish. Can you,
I think it looked great.
I think you need to move the port manip. I think
Outboard a little bit?
Yeah, inboard a little bit.
Inboard.
Then it's gonna hit the light.
Because of the ramp?
Yeah.
You go on like this?
Yeah, there you go. That should be enough, I think. Right, what's the next? We release this thing?
Mm-hmm. Yep.
Have we been releasing it from seabed? From like 20 meters out?
I think just from seabed.
We'll watch it.
It looked like the descent weight's still underneath it. Is it still connected?
Yeah, I think so too. Okay, so I'm gonna take off from here for line quarry, and then I will port in, and then I will land in the seabed, and Master Alex will release the weight. Yeah.
Sounds great.
Yeah. Okay.
Thank you.
Oh.
Slide around.
Why is it? Oh, I'm buoyant. Okay. Are we on the right side, Alex? Yeah.
I think we're on the side for the
Release?
ascent weights, but I think
Isn't that the green tube?
yeah, there's another one on the other side for the descent weights,
Okay.
and I don't know. I think it's on the other side,
It's on the other side?
and I don't know that those have been released. I know that they're sitting underneath it, but I can't see the other end of the rope.
So we should go on the other side?
Yeah.
Yeah. Okay. Actually, we shall go on the other side. Thank you. Dankeschön.
Also, do people just say bitte as well?
Bitte.
Mm-hmm.
Yeah. Come and say hello, bitte.
Hmm?
No. Oh, are you sure? Yeah. I guess. I think it's the other side. Maybe.
Yeah, there is a weight on this thing, and it looks like the pin might still be. Yeah, the pin's still in, so you have to do this one also first.
This is deep. Of course, I got big boulders underneath me. Let's see how far we can land here. Yeah, I think you could reach that. Nope.
Oh, it's pretty
Yeah.
far, but we might have to do like come in at a weird angle.
Yeah. It's in a hole.
Yeah. I mean, I'm happy to try whatever. It's just I don't
Here,
think we'll ever.
I'll get you your starboard arm closest.
That's too much.
Let me lateral it. Am I hitting anything on
No,
the
you'll
left?
be able to.
There you go. Now I'm gonna sit down here.
Lean medium. Oh, that's pretty awesome. Roll over. I like that. Okay.
Hold on. Let it stabilize. I'm gonna get you a little closer.
Well, let me see. I can see how close I can get.
Okay.
Is that you?
Yeah, it's me. Stand by. I'm trying to stabilize.
also see the pin. It's flying just up the middle.
Yeah. Oh, here we can see the pin. Pin's off. Wunderbar. Yeah, it's off. I saw it come off. It's a wonderful job. Wunderbar.
Beautiful. Ist auch so wunderschön.
What's that?
Wunderbar und wunderschön.
Wunderschön.
What's the difference?
It's the way the.
It's not really a, I think they're interchangeable words.
So now we need just the other side, right? Yeah. Other side?
Yeah.
Okay.
Would that be Esme? Esme? Would that be Hidea?
What?
It's 8 words. But it's like beautiful, right? Like, yeah.
Having a German English translation going on. What is that? Oh, we moved them from wunderbar, wunderschön to Orea. I don't know. Or we moved them from saying amazing.
Yeah, or just, yeah, beautiful.
Mr. International Plus.
When you send the lander up,
The what?
This thing?
yeah,
We're just going to leave it.
I'm just gonna leave it.
fair enough. Because you've got your IGs on now,
Yeah, they're both loaded on.
is it going up soon?
Like
Yeah,
now.
like
Can
now.
you just test the light reading up?
Yeah, well, that
So
might
turn it
be-
on and turn it off. I'm just interested to know whether the battery
Is that on the other side?
Yeah, it is on the other side, but I'm wondering if you came in at 90 degrees, if I could push it. Push it over there.
No. This side?
No, the other side, like on the kind of the downhill side. It's that side ended up kind of in a hole. Not hole.
If it's tricky, don't worry about it. I was just intrigued to see. I mean, we can always just test it with the camera back up or coming to be fair. So
Shall be
it'll
intrigued.
be the same outcome.
Anyways, that's what's
Yep.
going on there.
Yeah, let's just do it on deck then.
Yeah.
Abort that operation.
Yeah.
Abort the operation.
Yeah, 'cause we're
Not
gonna
yet,
spend like another
completely.
15 minutes.
So you have to still,
It's never quick, is it?
Well, and then it's
still
in a
have to
weird
do that
spot.
on the, you see that there.
Did you, is this just for how it landed? Because it still had the weights on it.
Yeah. Yeah. Have the descent weights been taken off?
It is, but what we can do before we release it is kind of wiggle it and make sure it's free before we lift it.
Yeah, that sounds good.
Have you removed the deceleration weights?
Yes. Or yes, we did. Well, we pulled the pin and the ropes.
Yeah.
Not released yet.
You ready for action? I'll put it close to you.
I'll do that one.
Do we have to bring the plates down?
Huh?
Do we have to bring the plates off?
No, so
Okay.
I don't. Wait, are we taking off those 2 weights?
Yeah, you got to rip those
Yes.
off.
Yeah, we're
Yes.
taking them.
That's compensation
come
for the
off.
IGTs.
Yeah.
Right,
Okay. I guess I'll land it in the middle. Just to make everybody happy.
Next time.
Especially Dan's here now. Hello, Dan.
Hello.
Hey.
Hello.
Yeah.
It's a lighter manila rope this month. We ran out of the thick stuff. I'm interested to see.
Like lighter density or lighter weight?
Lighter weight.
Oh, it's fine. Like thinner.
Thinner.
Oh, in terms of being able to break the zip line?
Mm.
That would be fine. You can always scratch, even if you break the rope, we'll just scratch the zip line until it breaks.
Alright, so drag them on.
Yeah.
Stand by.
Standing by.
It's uneven, that's why it's
So here, the M3 makes a great kickstand.
Yeah. That's what I thought. I was trying that.
It works.
Let's see what happens.
I think it's good.
Okay. No pressure.
It's just a weight. I don't care about the weight. I'm more worried about like things like AGTs.
Just come straight out.
Oh, that knot. I see.
Oh no.
We shall go to
Well,
plan B.
I hope Corey didn't tie that knot. You know, she probably did. Corey. Archie
So one other challenge.
Come on, Corey, you tied better knots in the
Yeah.
AGT cables.
I know you just had a lesson, but obviously you didn't listen.
Well, Dan didn't listen.
Oh, Dan was there too? Oh, here's another
Yeah,
one.
Dan was there too during the knotting lesson.
Alright, this time I'll tie a serious knot. What was it?
I should
what,
have.
what did we learn? Ship shank.
Ship shank. Bolan
Bolan.
on a bite.
Bolan on
The
a bite.
monkey's fish.
Monkey's fist.
Fish.
I think
Fist.
just fist.
Fist.
Fist?
Yes.
Is it? Oh.
Yeah,
It's a shape.
not the monkey fish.
Stupid monkey's fish.
I think that's just Archer's pronunciation.
Okay.
Oh,
And we
who
learned
tried
the
that?
splicing too. That was
That
fun.
me.
That's fun.
Yeah.
Let's make sure I'm on the other end of the rope. Grab it.
Grab it? You think it can?
Yeah, I have faith.
It's a little low.
No.
Well, the fist makes more
Yeah.
sense based on how it looks.
How about a tip? Oh, I'm disappointed you're not grabbing it.
Well, it's hard to. Well, I just didn't think that would actually
Work?
fit.
Of course it
in the-
will.
You think so?
Yeah. No pressure. The Brit's watching you.
The Brit's the one who.
Tied it. Oh, see that? Oh, I
yeah.
have faith. See that? Faith restored.
Take the other knot holds.
Now you're shaking me too much.
Except for to the wrong side, so now I can slide it off to the outside. And it wants to slide off that way, and I'll come back.
Faith restored. Cool. So if they were listening to their knot tying lessons, you could have just easily grabbed.
That's why I needed a knot tying lesson, clearly.
That's true. That's true. You have a.
We did with Katrina.
Like every time I touch
So now
the-
they accuse us that we didn't listen and didn't time
Okay.
us correctly.
Well, let's see. So if I could always back off now.
Yeah, I'm afraid I'm trying to make a big enough gap because
Okay.
I'm afraid the subs is like not unstable right now, so I'm trying to get it big enough for I'm comfortable with.
I'm kind of using a little thrust backwards.
Yeah, I can tell.
Yeah, it's stabilizing.
Is it?
Yeah. See? Faith well, almost.
There's a sliding back down.
Oh, that's into
Almost.
this right there.
Boom.
There it goes.
Faith restored. See that? Katrina has faith. I mean, Corey has faith. Not much, but still there. Sorry. Okay.
Complete faith here.
Complete faith. Now let's. Who tied this second knot?
That'd be me as well.
That's probably you.
This will be better than the first one, I'm sure.
Let's hope.
Oh, you got too much faith.
I'll
No.
try to pull
Oh, I
it
think I
sideways.
turned that one. I think I tied that one around it.
Yeah, see, that has a loop ring. I like that. I'm pretty sure this one will be perfect.
Are you sure you did that?
I tied that one.
That Archie did this one.
Corey.
Looks more professional, doesn't it?
Try to pull on it. Yeah, flying away.
Yeah, no, you're pulling me in. Stand by. Stand by. Oh, never mind. Disregard my instruction.
Just this rock is uphill, right? So we just
Yeah.
need to get some clearance. Okay.
Where are we
I
going?
gotta turn. It's turning me.
Following it down.
Oop, oop, oop.
I think that just might be whatever the back of your sub's on.
Yeah.
With these sub. Sorry.
Okay, let's release it.
Yep.
Hey. Oh.
When we settle out, I
Yeah.
can just bump it.
Okay, I wonder. I hope that sonar works.
Why would you say things like that, Ben?
I don't know. Comes out naturally.
I can't reach it there.
I don't know how to lie. It's already off, isn't it?
No.
No? Okay, let me back up. Okay,
There we go.
yeah, no, I told you it was released. Okay.
Did it fall onto the leg now though?
It's okay.
Oh,
That'll get
don't look at everything, Corey.
That'll slide off when I
That'll
lift
slide
it up.
off.
Okay.
Okay, so I'm gonna grab onto it with the starboard.
Okay, you think you got enough stroke in there
What do you mean?
to pull it out?
Yeah, I can pull it out, but I need to grab onto it first.
Yeah. There's enough space to pull out. We don't have to back out.
Yeah, I'm just.
Okay.
It'll be a little bit awkward.
Yeah.
Grab it'll probably bump into our headache crack when it floats. That first. Because this is a little close on this side. See what I mean?
Yeah, sometimes. So we should. I'll just back out. Now I'm just going on one leg. Okay.
Do you want me to knock that off too? Well, do you think it'll ascend with it on there?
Oh, you're gonna grab it?
Yeah, before we release it. Are we gonna? Because I want to lift it up off the seabed. I would imagine. Because if it's hooked underneath the plate in the rock.
That plate's gonna.
Yeah, you
lift up.
can.
Or do you want to just do it as it is?
Well, we have done from the bottom,
Right.
so it doesn't have to be lifted up a lot.
Perfect. Let's do it. I'll
Let's
grab
do
the
it.
board off that.
I was trying to get you on the right arm. Okay.
No,
good.
this is fine. I was just doing this one. I thought we were gonna get it out of the under the rock first.
Well, we can. I mean, if you think
No,
it's
this
better.
is totally fine. I misunderstood, so that's why I had them set up with the wrong arm. There she goes.
There she goes.
Look,
There you go. There you go.
Oh, right in the headache right man. Oh, nice.
Oof.
So
What is? Wait.
It should come out.
bridge that's
Tight.
lander on the way.
Ah, okay. This is just.
That Corey must have tied that knot too.
Yeah, that's right.
I was thinking the same thing. Just been hanging that up to the. Keep it from going.
We're gonna watch it all the way up, right?
Yeah. Okay, that's it for me. I'm done.
Oh,
Bye
that's
bye. I'm going down.
flying down watching this for the whole way.
Oh, sorry, no plastic. Wait a minute. I'm gonna maybe some. Okay.
Hello.
Hey, mate.
Thank you.
You had fun. Yeah. We're going back. Ouch.
Going backward?
Yeah, that's the
Down.
on the way up. It'll be about 11 o'clock.
Dankeschön. Thank you for the
Danke.
Hey, Josh. Have any trash in there? Nope. No cups? No nothing?
You know what we left here?
No?
Hello.
Where are we going to?
Do we have to follow the ship? With the,
He
land?
said, what time did he say approximate on deck?
11. So an hour.
Eleven. Probably have like
Could be
45
more,
minutes
could be faster, yeah.
to play around.
Play around.
Yeah, if
Okay.
that's not enough time to do anything, then we can just.
Well, we could try some filming. Dan wants to come back. I want to do some Avenella filming. Probably the best area. I don't know. Everything is a little bit further away. I don't know if that's the best spot for that. Would probably be Schmidt 1. I don't know where that is on the.
One is down here.
How far away is that?
I just decided to do a feature today.
No, the nanos are both up on the. Oh, the one.
Chimney one or Chimlet one?
Chimlet one, and this one is spelled out already.
Trying to, because we have to see how the ship will move with the patch the camera. But we can try. If it's too far away, we can also see if we can find something else. I just need to confirm where they saw the Alvinellets before.
He's searching right now. How far away it is. I thought Chimlet one was where the old lander was. What's down there? Down south?
Down here.
Oh no, that's Claims Court.
Thought it was over here. Marking it's
Trying to figure out where the Schmidt 1 marker is.
That's really far.
But I will also
No matter
send
what,
a
that
message to the chat and see if we have any other.
No matter what, it's like 300 meters away. I guess we can just investigate this area out here. That's like 300 meters away. Which way do you think it's going to go, Mike?
I would say it mainly stays in space up to 250 off the bottom. It goes port side and then comes back to the last 250 towards aft. But I would say more or less will stay port side.
It'll move over to the port side, you think?
Yeah.
Okay. Yeah, it's a pretty big vessel move to go over there and then just come back. I can ask them if they're okay with it, but by the time we get over there, it'll probably just be moved right back. Unfortunately.
Yeah, okay.
I can have a look around here and see if I can find anything for you.
Yeah, we can do that.
That one's 360 meters away here. Okay, so it's
The opposite way of where the vessel's going to be going. See what I can find. Let's play out in the unmapped area.
there be dragons.
Hmm?
There be dragons. coming up with 25 approximately. 25 per minute. So 250 and 60 minutes an hour. So and about 11 shortly after that.
Pretty flat up there. Seamount. Let's see, how far away is that? I can make it there. Go check that out.
I already find here. And that's coming up with about 24 meters of minute. So with 24, so it will be just a short, round about 11 shortly after. Okay, welcome. Bye.
Will you periodically look at that beacon over there, just make sure it's not getting super close to the surface and I need to come up fast or get back into position?
Super close in terms of depth.
Yeah, so like when it gets to like, I don't know, like 300 or 400, if I'm not paying attention, let me know. So quiet in here. Mm-hmm. Can you check and see what depth sensor we're on?
Sorry.
Will you check and see what depth sensor we're on?
Minus.
Try CTD real quick. No, that's more like it.
And then we're going to stay on starboard.
Okay.
It looks like it's rather opening horizontal distance.
Pay me out a little bit at the end of the leash now. I'll let, yeah, just pay out nice and easy. I'll let you know.
That's good. Thank you. The weird shadow. I'm sorry. Looking at the sonar, I had a weird shadow. There's a little bit of diffuse glow right here. No, just the bacteria. I'm looking see around this area if there's anything more aggressive. Aggressive. Aggressive.
Cheerleader J-Rod.
When is he not?
Good point.
About what?
Cheerleader. Bueno, acabamos de estar al que fue con todo el tsunami. Podemos ver todos los científicos en acción.
Looks like I got still a little bit more right there. Looks like I'm going the right direction. Does it look a little smoky to you?
Hey guys.
Hello. Hola.
I asked you guys to show us just a little bit of, how you control stuff. Bueno, aquí nos van a dar una demostración de cómo utilizan los brazos hidráulicos del submarino. Todo es controlado remotamente. Entonces, así como los juguetes, los carritos de control remotos, así utilizamos los controles remotos para mover el brazo hidráulico. Wow, okay. Impresionante.
There's also these two sides, they're about
Thank you so much. What are you guys up to right now? Just nothing?
Waiting for the lander to come up.
Oh, yeah.
Well, where are we? Yeah, if we're close to Tunis, then we'd like to do that now.
Yeah.
We'll do it.
We have to see a week.
Yeah.
What's that?
The lander's at 1200.
Actually, I guess this is Smoker 7, so,
Smoker 7 or Chimney 7 now.
it should be the same.
Smoker 7, oh no, sorry, Smoker 7 is different from Chimney 7. I've got two different coordinates.
Are they very different?
Well, I've got 2.104510
Uh-huh.
and 2.104497. So 510 versus 497.
Chimney ones.
Smoker 7 or Chimney 7? Chimney 7.
Yeah, Chimney 7.
2.10.
From the pad. Okay.
2.104497. Minus 91.925. 529.
I know where it's looking.
That's Smoker 7. You're fine. I just don't have Chimney 7 on here yet. You're good.
That's true, Chimney 7. Let me give you a Smoker 7.
Yeah,
Just to confirm.
Yeah.
Diffuse flow everywhere around here.
Smoker 7's there, but so Chimney 7's here, so we should be fine. But I'll mark Chimney 7 so that we have that here as well.
got this bacterial mat everywhere, but no big structure.
We were probing around one of the bases of the chimney
Uh-huh.
earlier, and I mean, there wasn't any like
so
shimmery water.
tell me your other point.
Go down into the sediment, just a centimeter or so, and it's like 10 some degrees.
2.1
Just like in the
Mhm.
like sediment around.
5.0 and negative 94.925.
925, 537.
Yeah, now that is on top of 6%.
There we go.
Yeah, good. So we're good. We've confirmed. Oops. Great. Chimney's up.
Skate maybe. I would
Hmm?
skate eggs.
Mm-hmm.
I'm hoping that is my trail to
Skate?
another vent.
Oh, vent.
What is this your trail to?
Hopefully another vent.
Oh, nice.
I mean, I think
Wait,
that-
one that we haven't seen before?
I don't see anything up here, but my map doesn't have that. I'd have to look on his map. But there's bacterial mat everywhere.
Yeah,
Yeah.
there is.
Mm-hmm. Yep.
I'm just
There are a
trying
bunch
to.
of skate eggs, so
Can you
it's a trail.
bring up your map?
There was this one
Yeah, there's no waypoints up there or markers.
kind of like on a flat area,
Okay.
which was like maybe a couple of meters high, maybe 3 meters high. It was
Okay.
like, I don't know, I don't know,
Which
it's
one
hard to
that one is? Let's see. Let's see.
go through to the original.
Right. Let's see.
It was 506, sorry, SO 565, where the two previous dives ran into the
Yes, for this site. 6-4. 6-3.
6-3.
6-3, 6-4.
We had one dive on the coast.
That looks smoky.
Does anyone know how the egg sacs skates can lay?
How they can lay?
What?
What was the question again?
Yeah.
Does anyone know how many
How
egg
many they
sacs they can lay in one go?
No, that's a great question.
Like, are these? I guess my question is, are these all from one singular state or like 20?
Very good question.
Got it.
And that means that I'm right at this ridge here. Will you measure how far away I am from where the map says this little ridge is at? Okay, you said
What
ridge
ridge?
just south of me? Like that little ridge, just like, what's that? One, yeah, how far north am I of that? No, just straight.
35 meters.
Okay, so that would mean that that hill that I was going to is actually at the edge of the map up there.
Oh, you're saying this is about 60
Okay,
meters.
so
It really would be somewhere about
around the base of it up the north?
Yeah.
Alright, let's try that one first. Go this a little bit further. How much time we got? It's only at 1,000 right now?
Yep. 1073. Another
Right.
40 meters, 40 minutes. But it's on the way back.
For the moment?
I guess, for the time.
I think he's just holding position till.
Yeah. And then I said it's south towards the east so far. But it's going to get an east and it's going to hold it pretty soon.
A little bit more.
The circle here is about 35 meters. So
Okay.
certainly east, if not southeast.
About that heavy.
Well, it seems like we're starting to trend up.
3 is,
At 35 degrees, or 35 meters is 30, 35. That's pretty consistent.
Say that again.
The offset's pretty consistent.
And this is going uphill right here. So yep.
right, yeah.
Yep. Seems like pretty much this is the top of that hill, right? And that's consistent with that 30 meters off, right? 25, 30 meters
Just
off?
about, yeah.
Yeah, this is me almost at the tippy top of this hill.
Yeah.
But no venting here. Or super active.
Just essentially diffuse matting sort of things.
You're smoker free, so I guess I'll check.
Alright, well that's been explored. And what's that?
Yeah, we're
What
just.
else? What else do we have?
Chimney front, chimney center, then smoke three.
Expect three, chimney three, same thing.
I think so, but I have markers
it just for giggles so that we don't lose it.
Quite difficult to swing it through.
Nope, you're fine. I'm just gonna make sure. Okay, it's ready. Alright, so it's probably the same.
Why is that so different? 104, 928. How much of a difference is there between 104, 928 and 104, 84? That's like 2 meters.
5 meters.
Okay, so the last digit
Not very.
is tens of centimeters.
Not super super. Cool. Sweet. Good to know.
Reassuring.
Yeah, yeah. No, it is very much so. Love to see it.
Anything else? No.
The landers?
Landers, yeah.
Yeah, I'm gonna go get back on the starboard side of the vessel.
Seven 50 to get a push to the east. Quite strongly. About 500 meters.
No.
No?
Rat jet.
Yeah, we don't really see beefs here. As much. We see lots of rat tails. These are our seeds.
I can't lie, they look the same to me.
When you see a beef and a rat tail, you'll be like,
The beef looks like it has like an armored head.
yeah, beef looks like a beef. Like when you look at it, you're like, yeah, that's beef.
That's very descriptive.
That's what cleared it all up then.
Yeah. Well, you say that
Thanks
as soon
for
as you
the
saw it. You're like,
description.
there's no other word for this creature.
That was about the thing about Jacob's PhD defense. No, make this,
Yeah.
no, explain this to a five.
Yeah, it looks like a beef. Looks like
You can pay me in. Whole in.
the distance measure of the USBL per sec?
Yeah, it doesn't bother me at all.
And then that's going east now.
Lander's going east?
Hmm?
You said Lander's going east?
Lander's going towards east now. Yeah, closing in. Distance to the ship only 85 now.
An enemy on the block, huh? Is that what you said?
You know, the camera staying on the basket for the next cruise.
I don't know. I imagine so, but we'd have to ask Dan. He would know.
Is he on the next one?
No, but Dan Bernari is. So. Have you ever seen that guy travel without them?
Next cruise. Der Länder hat noch ungefähr 450 Meter östliche Komponente.
It's on the sub right now, yes. Hey Dan, you know, is the miso staying on for the next cruise? Both of them? Yeah.
Ja, ich beobachte die noch, wie gesagt, die nächsten 450 Meter sind Ostkomponente. Okay? Cheers. Bye. Jo.
That looks interesting. Just another scan.
Give me a roll.
I don't know which one you want, but you got all. Okay, good. Do all three of them. Oh no. The collection. Thank you. I'll turn that right. Thank you.
There's no life over here. No life.
So much for dragons.
lander is going to pass under the ship and go on port side.
Okay. What depth is it at?
The lander is going to go on port side, so it will be separated.
Found a fissure. Found a crack in the earth. Look at sonar, trying to see where potential comes up. Fissure here. The giant rock. The sonar target I had is just a giant rock. Mm-hmm. Look at that coral. I don't know what kind it is. Is it a coral? It does?
Yeah.
I don't see the polyps. You sure it's not a sponge?
It's sponge? Sponges don't usually look like this. It looks like a sponge.
Okay, okay, there they are. Okay,
I
there
was
they are.
like, no, it's
Yeah.
not coral.
Well, no, I just at first I just saw spiny stuff.
Yeah, no, I see it.
RV Bridge.
Go ahead.
Hey, I think it would soon be a good time to start lander hunting when there's a good moment for you to come off bottom.
We're ready now.
Roger that.
Tell him we're on our way to him.
Yeah, we're on our way.
It kind of looks like it has
Alright, thank you.
a black skeleton in there
Yeah, it could be. Yeah, you can see on
on the little ones.
the little ones. Yeah, so it could be a black hole.
All right, I gotta.
Yeah. It's fun to say.
Pretty though.
I love these like fissures that you're nearby.
Is that also bottom?
Yeah,
I'm just, yeah, I'm chasing it.
Just for the milestone upload.
It's still pretty close to the ship though, right?
It just pops out.
Yeah,
to
let
the
me
side
know
now.
when it's good to start moving.
You can start moving now.
You're free to start moving now.
Alright, that'll be a move to the west for maybe 200 meters or something like that for starters.
Move to the west, that's where I'm at. Good.
Yeah. This is probably good trying to get out from under it. Yeah, no worries, thank you.
Well, I will hang out here till he comes towards me. That means I can look at this area some more. That
Yeah,
kind of
that's
looks
almost exactly where we're at.
Posture of active engagement now.
I know. I'm struggling. Thanks for laughing at me.
That's alright, anytime.
What's up, dude?
We've got about an hour streaming on Facebook left, so we can do that.
What?
When they're after the landers recovered, then we can do some leaching. Okay, gotcha. Is that right, fine? After lander recovery, we do some. Did you see them? Alright.
What's the area we're going to?
Couple of chimneys, couple of smokers, couple of smoker chimneys. We're going to do some side lighting of the Fiat Benzo transit.
So once you do 8 hours,
Sounds good to me.
start to do 8 hours.
Yeah, oh yeah, that's gorgeous. We just make sure it's 7:55. You're not going in there, are you?
It's not big enough for me.
It's not Sebastian size.
So it's not like just offline for that long.
I think Sebastian.
Like
Yeah,
just they
size.
cut off after 8 hours.
They wanted to be.
This isn't an Ant-Man situation.
Put yourself in there.
Review bridge, my apologies, I looked at the wrong transponder, we can just stay where we are for now.
Roger that.
That's alright.
So he's not coming to me.
Sounds like.
Is it how far away from the vessel on the port side now?
It's already 60 meters and it's continuing for another 150 with an easterly component. So
Okay, so I need to start going east now.
yeah.
Roger that. Back to going east mode.
Yep. We are currently 189 horizontally away from the vessel.
166 from my launch point.
Quite a nice mix of surface current there for the last 50 meters.
Another coral. Like a sea pen.
Did anyone lose a microphone cover?
always. There is nothing out here in the darkness. Oh, that's cool. Who are you? Is that a hydrogen?
Sorry?
Is that a hydrogen? Or a mineral? It has a tube.
Two entities.
There. Or am I? not a squat lobster.
Yes. Agreed.
We're good at this.
Yeah.
Isn't that what science is? Saying what it's not?
Sometimes.
Another fisher. They take you places that typically have hot water.
I don't think you're gonna fit in this one.
No, but you're right.
Unless you brought a shovel.
We do have a knife.
We'll be here a while.
I already went up. I went along this fissure already. Time to find more.
Who's the other station place? How station was? Let's go after this lander.
Whenever that happens. 141 meters. Still in a really good spot. Man, Mike, you were right. That's why we're gonna have to move it all.
Now it comes towards the ship. So perfect. Right spot. You
Where are those
backwards.
guys?
And that's it.
Where are those guys? Who are you?
Ruby Bridge. Now I'm actually moving a little bit to the west to get a bit more distance from the lander.
Roger that.
Heard that story before.
I'm not gonna poke that bear.
Who are you? Ah, I'm flying.
I'm not convinced of that.
I'm crashing.
That I believe.
Alright, come on. They're all over the place right here.
Are they?
I don't know. We have enough time to look, but let's try real quick. Let's try real quick. I think you might be right about a Xeno.
No, well, no, these are flat bugs.
Well, Xeno, I've seen flat Xenos.
Really? I thought they were like more like
they're like
spongy.
ear looking. I'll go with a sponge looking at that one over there.
That wasn't what I was referring to.
Oh, you were talking about these things that look like mushroom pores? That if you hit them, a bunch of black stuff comes. There, I did that.
Did you say you want to poke him?
No, I gotta get moving. Vesta's gonna run me over here in a second.
Maybe this might be one.
Oh yeah, yeah. Those that
look like a little like sponge.
Let me get a little bit ahead of the vessel real quick. Oh, look, another fisher. Oh no, same
And
one.
take you away from the ship. You're lucky.
That's one I already did. Which way's the surface going, Mike?
Last 50 meters in a mix, westerly windy.
Okay.
It comes from southwest all the way to northeast.
So I should probably
Sorry,
just,
southwest to northwest.
so I should probably get a head start
okay.
and get ways out away from them.
Yeah.
J-Rod, are
Yeah.
the lasers on?
I do not believe they are. Let's see. No, they are not.
The surface current is quite strong,
But
0.7, so.
they look like they got knocked. Those do not look like useful lasers at all. Lasers are not helpful today.
Just gonna quantify the like angle with it.
Yeah,
As you
the
get
further-
closer, it goes from below to above.
Look, I can make them touch. Lasers are not useful. Sorry.
Now they're a rangefinder. We actually have one for more than S3, and they are mounted.
I've heard about that, yeah. Hey. One is for measuring, one is for range, right?
I think they all kind of, like together.
Gotcha.
I mean, our standard lasers that are parallel, we actually have three. We have two that are 10 centimeters apart, and two that are at a right angle that are closer. And so sometimes you can't get the 10 centimeter ones on an object.
Yeah, that's cool.
Solid stations, then I should have surfaced approximately 100 meters, slight upwards on it.
Alright, what was the first target that he said he wanted to go to?
I didn't catch it. Dan?
Yeah.
Here.
Dan, what was the first location you want to go to? Just so we can get an idea of which direction to go.
Sir.
Roger that.
Are we sure that we want to
Oh, 27 on the way.
Well, now it's not. Now it's now.
This one shipping through.
Yeah, 3 is first.
On the surface. On the front.
Rat tail?
Chimera or beef?
It's a rat tail.
Not beef. Rat tail. These are
Why do you put it beef?
Bony-eared,
Sorry?
bony-eared
Bony-eared
fish.
ass fish.
I mean, yeah.
That's not a beef.
That
No,
is
not at
a
all.
beef.
Not even close.
That's a rat tail.
Yeah. Beefs do not.
Very good.
Yeah.
You won't take like this.
Right? How annoying is that? Like I had that just right. We shall try it again.
That's pretty.
That's beautiful.
Now you're just showing off, dude.
I know, right? That's what it feels like. Diva.
It's dancing for you.
RV bridge lander's about 50 meters just ahead of the port beam, so we're moving in to pick it up.
How's that?
Roger that.
We need to decrease the ISO one click.
Decrease ISO.
One click. Decrease.
One click.
One click.
Tell you something.
with his eyeball.
Of course I can. Why, you want to see the lights?
It's a bit like crocodile skin. Wow, and copper skin.
Alright. You see your eyeball. I see your eyeball. I do not want to see that either. Eyeball.
Is that why they call it the antidote?
No, that's not the
That's
right
a different fish. You're gonna crash.
This is the
You're gonna
front
crash.
tail, Dad.
You're gonna crash. Told you.
Oh.
Does everyone remember that fish that had a little copepod parasite?
I think he just ate that thing.
I didn't like that. I don't like parasites.
Oh, sorry.
I think he just ate
I
that
thought
thing.
it was cute.
They just give me the parasites give me the heebie-jeebies.
Are we filming at this point in time?
Yeah, we're just waiting for lamps to come on.
I'm just playing. This is J-Rock
I
time.
was just wondering. J-Rod.
So you give me a couple more meters?
Yep.
Focus.
Find us a beach.
There have been no beats there. We found like 1,000 at the other location.
I don't like his
Not as many.
scales.
The other sites there were tons of beeps. This
They're
site was not
freaking me
as many
out.
beeps were, if at all. I do not remember the beeps being present.
Yeah, it's too bad.
What's the difference between beef and Cusquille?
Well, Cusquille is Cusquille. Beeps.
Wait.
I think isn't
No,
beef a type of Cusquille?
Yeah.
Beef's a type of Cusquille.
Yeah, I think so. I think
They're
you're right.
gray fish. Can we all agree?
They are very fish.
Thank
You
you.
didn't care because you were such a
Oh, the bridge lander is out of the water and you have my full attention again.
Roger that, thank you.
All right.
And I lost your full attention.
Well,
We don't have
where am I
a
going?
expert. What? None of us know
Where?
fish.
What is the next location? The chimney? Chimney and
Chimney
smoker?
3.
Chimney 3.
If you look at that, that's yeah, right?
That's the one.
Yeah. What?
Mm-hmm.
Yeah. Much more sleep. Yeah. Different.
Very good.
Yeah.
Big eyes, tiny eyes.
This guy just like headbutts into the earth on the regular. Not so much.
Because his big eyes.
Who knows, really?
He's got some tiny eyes.
None of us are fish. See, a lot of you just annotated with fish.
You can find it?
Found one chimney 3 that's 243 kilometers away.
It's a little far.
Yeah.
My guess is
That's 243 kilometers away.
My guess is I need to go
Better get
south.
cracking then.
Right.
243 kilometers?
No, honey.
Correct.
Oh, honey. Did you just say, oh, honey?
It's a move on.
That's hilarious.
Yeah, come
That
on.
just made
See
my
a
day.
chimney
Time is ticking.
Chimlet 3?
No.
Chimney 3. Chimney 3.
Which
Chim,
also
chim.
might be
Oh, no.
smoker.
I can give you the.
You're
black
sure,
one.
Dan? We had this problem before.
It's chimney and smoker 3 are the same. So if you have a smoker 3, Give a chimney.
Smoker 3 is also 258
Smoker 3 and chimney 3 are the
kilometers.
same thing.
Yeah, they should be the same thing.
It's 258
Yeah,
kilometers.
well, no, just look in that
Don't be
area.
silly.
Yeah, look in this area. You're just looking at the waypoint.
It's, yeah, go up to the top, and it should be up here.
This one? Smoker 3?
Yeah.
That Smoker 3?
Yeah.
That's a different Smoker
That,
3.
we have too many smokers. We found it, don't worry. We're gonna take you 200 kilometers away. Roxanne's like, no.
This is Smoker 4 near to Smoker 3.
324 meters, 177.
Yeah, ish. Yep.
Let's start that vessel move.
There's chimney.
Chimney. Are you guys going to do a transect or what's the?
We're in a JRV. We've got an updated waypoint for you.
Updated waypoint.
Mini landers on deck, do you guys wanna be the leaders here? Oh, instead of the guy or somebody else would. I do want to do that, she needs help, we can get them in.
Still the big one.
Or you can go, and I can sit here, it doesn't really matter. You can go. Okay, if Jada's on it, I didn't know that, I just wanna make sure somebody.
No, I know she's up in lab, but I don't know if she.
Oh, yeah,
do that.
Yeah.
The land might want that.
I can't pay for it,
Pathopathes. Pathopathes. Yeah. Baby fish, toot, 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 toot.
Oh.
Evert's died again? Pathopathes.
Hey.
You did a great job.
Yeah. I'll do this away. And then it's gonna be that time again, and it's good. Yeah, sure. Of course.
Nothing. You said Smoker 3, right?
Chimney or smoke are 3, yes.
Yes.
Things should be the same.
Another thing to learn. Forever.
Forever. 200 kilometers. 200 billion bajillion.
200. Alright, I gotta wait for the dust to catch up a little bit.
Okay.
Okay. That map is like legitimately 30 feet long.
Good morning, folks. Hope you all have been enjoying the stream this morning.
The last place where we did the mapping was
The marine technicians have been covering for me as I had some outreach calls with some local schools in the Galapagos. But just popping back on here to tell you that we are about to take a little intermission for the stream because we're going to conduct some filming with the BBC, which we're very excited about. But that does mean we will be taking a little pause for a couple of hours, which is a great chance to grab a cup of coffee, take a nap, listen to some good tunes, and we will be back before you know it. Not cutting the stream right this second, but it will be in a little bit of time, but just wanted to give you all a heads up. Again, if you have any questions, let us know in the chat before we cut the stream. And yeah, enjoy.
I think it's just a wall. Unless it's probably just looking to see if there's another wall in that stuff. I always want to push things like that from the side. Always. It's like, I get it should fall over so easily. Just a little accidental look at that. That thing really wants to work. Oh, there's something looking up. Shortly. There's some diffuse stuff around here. You go ahead and put me in stuff. That way I can get set up to jump off this thing.
Yeah, of course.
That mountain peak. There should be that red peak right here. Or coming up to it. There's some diffuse right there.
Alright, if we're coming up to it, I think I'll probably pause the stream now.
What?
If we're coming up to it fairly close, right?
We're not.
Pretty far.
Oh no?
We're still
I was
yeah.
talking about another little seamount thing.
Oh.
He's just talking to himself.
Yeah, I've been trying to find out.
No, it's fine.
Normally she's not listening to me.
I'm listening to you, J-Rod.
That's great.
But I also like just got here, so. Hi.
We're going there.
Ooh, fun. I'll put that on.
What's Logger 2 doing? What is Logger 2 doing?
Logger 2.
Excellent question.
No, I'm just messing with you. I heard all kinds of scratching going on back there.
It's arts and crafts hour.
Is that a chimera? Yeah, it looks like a chimera.
Oh, a chimera, yes. I missed my chimera yesterday, so
Yeah,
I'm getting my chimera today.
probably the same
Hi,
one
buddy.
coming after me, right?
It's like you again.
Hi, buddy.
I don't have enough leash to get to him. Don't know, you start house. I guess you can,
Oh my god, hello.
There you go. I won't blind your big eye.